To explain in simple manner, what I'm going to do is I will take one presentation. Through presentation, I'm going to explain what is CDISC SDTM, ADAM, and TLF'S, and what might be your roles and responsibilities in your clinical sector. What is process of the clinical trial? Everything we are going to discuss today. Just introduction part. Okay, we are going to start with clinical SAS project training. Before we started with the clinical SAS project training, let us discuss about what is the role of SAS programmer in the clinical industry. Okay, so before going with that, why we always call clinical SAS programmer or clinical SAS programming and all. What is called clinical SAS programming and financial SAS programming? See, SAS programming is only one programming, guys. It's unique. You know what we learned: base SAS programming, advanced SAS programming. That is the one. If you use in the clinical sector, that is called clinical science training. If you use in the financial sector, that is called financial science. Okay, when you want to work SAS programming in clinical industry along with SAS programming, if you use CDISC developed standards called SDTM and standards to process clinical trial study data, okay, which is called clinical science programming. If you use base other than clinical sector like government sector, banking sector, or healthcare sector, or any other pharmacy sector, if you use SAS, no need any domain knowledge, guys. Just you can go with base and advanced SAS programming. That's enough. The usage of SAS programming is same, either banking sector or clinical sector. But whereas coming to the clinical sector, there, along with SAS programming knowledge, we need to learn CDISC standards. Okay, so because without these standards, we are not able to submit any data to the regulatory for the drugs approval. Standards are very important to know. So already we are good at SAS programming. Now, in this particular project training, we are going to discuss about SDTM ADAM standards. So these standards developed by CDISC organization, CDISC developed by FDA. Okay, so let me go with simple example, differences between financial SAS and clinical SAS. Okay, what is the difference between using SAS in other than clinical sector and clinical sector? Okay, so for example, let's come to the financial SAS. There is a bank one, guys. So when it comes to the financial SAS, it's not necessary to follow any specific terminology, guys. Okay, for example, so there is a bank one where you receive the data as customer ID and debit and credit. So for example, you have received data from your particular bank. You are working with the banking sector. There is a data from one bank you received. There is information. Just 3 examples I'm taking: customer ID and debit and credit. And suppose let's say you are handling with now another bank project. When you are working with service-based company, definitely you are going to work with multiple banks. There is a bank too. You receive data called ID, deposit, and withdrawal. So it is the same. So customer and ID, deposit and withdrawal, and credit. So deposit and debit, credit and withdrawal, almost same. So when it comes to the banking sector, it is not necessary to follow any specific terminology or specific standards, guys. Since then, no need to submit data to the any regulatory, no need to submit your data to for approval or something. No need, right? Okay, no need any regulatory approval. As for your client requirement, just analyze the data and generate the reports. Submit to your client. So no need to follow any particular standards. So no need to learn any additional knowledge if you are working with other than clinical sector. Any sector, just I'm taking about banking sector here. If you go with any other sector, no need to follow any standards, no need any approval. simply you can submit. But when it comes to the clinical, guys, it's not like that. I mean, when it comes to the clinical projects, whatever the data we received from the clinical domain, or else in the clinical sector, or else in the clinical trials research, it need to be submitted to the regulatory for approval. So there will be different kinds of approval. There is a different kinds of reviews that we are going to discuss, okay, as part of our clinical SAS project training. But whatever the clinical study data we always use, it should be submitted to the regulatory for review and approval purpose. So hence, there should be a standard that need to be followed while processing the clinical style study data. That is nothing but CDISC developed SDTM Adam standards. Okay, we are not able to submit data as it is. Okay, every company has its own internal terminology. If you send that particular data as it is, the particular regulatory authorities will not approve. So we should follow particular. See, they have developed some standards. These are the standards. All the standards we are going to learn one by one. We should follow these standards while submitting to the data to the regulator for approval. For suppose, let's say there is a pharma one. For example, let's say you receive data from this particular company. So patient ID and date of birth. So I'm taking simple example. I'm not going that much detail. I will keep it as simple as possible since it is very first session, right? Now, suppose to say when you receive data, when you are working in service-based company, so not necessarily you will go with only one project, right? Once you are done with one project, maybe you receive it for, or you are going to switch to the another project. So that is how rotates happen in the service-based companies. For example, say you are working with another company also, there you received subject ID and birth date, two different columns. The name itself, values will be same. Patient ID and subject ID values will be same, but names are different. Date of birth, birth date, the values will be same, but terminology is different. Whatever the data we received for a clinical project, it should be submitted to the regulatory for review and approval purpose. That's what we are discussing from the beginning. And whatever the naming convention that been used internally by the companies, either Pharma 1 or Pharma 2 or Pharma 3 or whatever, it should not be submitted to the regulators as it is. So now what we need to do here, if you want to submit this data, either Pharma 1 company or Pharma 2 company, or else there will be, see, guys, clinical trials are happen nowadays. Thousands of trials is going to happen. There are thousands of companies. They are going to do every day in a lot of clinical trials research nowadays. So each and every company has to submit their data to the regulatory. For example, if you are coming from United States, what is the regulatory for United States? FDA. If you are coming from Europe countries, EMAS. If you are coming from India, DCGI. So every country have its own regulatories. So as for for example, if you launch your drug in United States, definitely you need to get approved from the FDA. Okay. So whatever the naming convention rule that's been used internally by these companies, same naming convention rules, if you submit to the regulatory, they may feel difficult. It will be pressure on the regulators. Why? For example, if there is 100 pharma companies, they are using their data in their internal terminology as per their company standards. So if they submit their data to the regulatory for the product approval, now all the companies are using their own terminology. If there is no unique terminology, unique standards that's to be followed, then it will be pressure on the regulatory to understand the data first. Okay, first they need to understand your columns and then followed by your values. To understand the data only, it will take time. It will be pressure on the regulators. First of all, if you use your own terminology, to understand each and every organization, each and every company, internal terminology, they need a lot of time. So once understand, they are going to review. Once review, they are going to approve. So there is a lot of time consuming work as you have own terminology as for your company. So it's like a lot of pressure on the regulators too. So, and what being suggested by regulatory is, the organizations while submitting to the data to the regulatory, before go for the data submission, it should be standardized. It should be organized. It should be follow a unique standard for all the organizations, all the companies. Then it will be, if you have unique terminology, guys, data will be different. Every trial is unique purpose, guys. But whatever trial it is, the data while sending to the regulatory authorities, we should follow the same terminology, same column names. Values will change, not a problem, but the column name should be same. Okay. So as the unique terminology being followed by all the organizations, it will be easy for the regulatory to go for the data review and our approval purposes. For the example, how it gets standardized. For example, so there is a most popular standards that's been recommended by the regulator is nothing but CDISC, guys. See, not only CDISC standards, there are many standards available in industries, guys. There are so many, but remember, most popular standards is CDISC standards only. Around more than 180 countries, they are following just CDISC standards only. So that's the reason this is one of the most popular standards used in the clinical trial projects. Okay. So there are some other standards are existing, but no need to learn about all these. So this is the one most popular. We will be learning about these particular standards only as for our clinical project train. So as for the SDTM standards, suppose say, for example, patient ID and date of birth by this pharma company one should be submitted to the regulatory as U subject ID. See, patient ID, you need to create like a patient ID columns like a U subject ID. And guys, the data will be remain same. The names only it will change. Naming convention will be changing. Patient ID, instead of as for the SDTM terminology, you need to create column called U subject ID. And date of birth, as for the SDTM terminology, we need to create like birth DTC. In the same way, when the pharma company too also to submit data to the regulatory, their company information, their trials information, so they also should follow the same rules. Use subject ID, and birth date is as birth date. So now this terminology.
Yeah, sorry to interrupt. So which version of IG we are going to use, Malu? SDM IG? Like
3.2 IG we are going to learn.
Okay.
That is the one most popular. Nowadays, lot of the organizations are using 3.2. And there is no clear differences between 3.2, 3.3 and 3.4. So 3.3, they added few domains. 3.4, they added few more domains. So what are the domains added with 3.2 and 3.4? We'll discuss. We'll see the differences as well. Difference between 3.2, 3.3 and 3.4.
Yeah, sure, Balu.
Yeah. Okay. So this is the terminology we need to follow while sending to the data to the regulatory, guys. The naming conventions are the terminology only will change. The data is going to be remain same. Okay. So now it will be easy for the regulatory to understand your data, because all 100 companies, they sent their data, but in this particular format only. So there is no different terminologies. So subject ID like your subject ID, birth date should be birth date. So as they have unique format, so they can simply review your data and then they can simply either if it is as for the standards, if it is working fine, then they are going to approve. Otherwise, they are going to simply reject. So it will be easy for the regulatory to go through it and it will reduce the time consumption. So when it comes to the standards, it will follow. So now coming to the next concept. See guys, while converting this particular study data to this particular as for the standards, we are going to use SAS programming skills. That is nothing but your technical skills, programming part. Okay, either if it is a banking sector, just base and advanced SAS programming is enough. Other than clinical sector, guys, any sector, technical skills is enough. But whereas coming to the clinical sector, along with base and advanced SAS programming, we need to learn these standards. CDISC organization developed several standards. So to keep clinical trials research product data to standardize, these are the standards we need to follow to organize in a better way, systematic way, while sending the data to the regulator. Okay, CDISC is an organization which has developed several standards. We are going to learn those standards on as programmers. What are the standards that are unique and what are the important standards? We are going to learn in detail in our project training when we will start with clinical SAS project classes that are developed by CDISC organization. All the standards we are going to learn. Now, we'll discuss about briefly the standards once we start our work. Now we are going to discuss what is the role of SAS programmer in the clinical industry. So now coming to the SAS programmer role. Okay. So as I said, along with technical skills, you should learn standards as well, right? Where we are going to use those standards. Okay. When you go for the clinical sector, you have to learn standards called SDTM, ADAM, and TLF'S. Let us start with role of SAS programmer in a clinical project. As a SAS programmer, what will be doing in a clinical project? I will tell you. Okay, we will talk about the clinical project steps in detail before we get into the technical part in the project. So here is the example, just preview I will show you. Just focus here. Any questions? Make sure, let me know, or else note it down somewhere. Once we are done with session, maybe in last 5 to 10 minutes, we can spare for your doubts. Since this is a theory part, sometimes most of the questions, whatever down, when session is happening, most of the questions will be answered by the end of the session. Okay, and still if you have any questions left, I'm going to explain. Okay, so as a SAS programmer, we are responsible for processing the clinical study data. So processing the clinical style study data to the particular standards. How it should be processed? By using your technical skills like SAS programming, as well as along with SAS programming, we are going to use CDISC organization developer standards, SDTM standards. Okay. In order to process your clinical trial study data, we required SAS programming knowledge as well as CDISC developer standards, SDTM standards. Using these standards and using your technical skills called SAS programming, using SDTM standards, we are going to process your clinical study data. That is first responsibility as a SAS programmer. What is the first responsibility? So we are going to collect the data. There is a phase 1 trial, phase 2, phase 3, phase 4. There are a lot of phases. So each phase data we are going to collect. And so there will be a lot of teams also, guys, not only our work to collecting the data and everything. So what and all teams are involved, what is the workflow, we'll discuss, industry workflow, everything we'll discuss one by one. So the particular data, very first, we need to standardize, we need to organize. So which is nothing but extraction, guys. Did you remember extraction? We'll pick the data from different sources. We'll create a SAS understandable table called SAS table. Extraction. Same. So SDTM standards. So as you have raw data, as for your company standards, as for your internal terminology, as for the CDISC developer standards, CDISC recommended standards, FDA recommended standards, CDISC standards, we are going to convert your raw data into SDTM standard data. First step. Okay. Coming to the next one. Once we process the clinical study raw data, will it be sufficient? Does it yield any results, guys? Here, just we are creating the table only. Only extraction part is only done. Does it yield any results? No, right? So some kind of analysis work should be done with that particular study data. Okay. So what purpose we are going to conduct the trials? To prove your drug is safety and efficacy. So to prove that particular primary objective, definitely we need to do the analysis, data analysis. Some kind of data analytics work need to be done with your study data. So in order to do the data analysis work, whatever the analysis data should be developed, whatever analysis is required for that, along with to develop analysis data sets, again, along with SAS programming, CDISC developed organization developer standards, ADAM we are going to use. First, to process clinical trial study data, SDTM standards we are going to use, guys. For SDTM, we are going to discuss around 45 to 50 days classes. SDTM terminology we are going to discuss. Around 20 more domains we'll discuss. SDTM. Once your raw data is ready as for the SDTM standards, then we are going to perform analysis on it. We are going to create analysis data sets. Again, we are going to use our SAS programming skills along with SDTM ADAM implementation guide, ADAM standards. So once the analysis data set has been developed with the help of statistician, and we are going to run different kinds of analysis procedures in the SAS, and some kind of results will be produced, and the statistician is the one going to go through the results, and he will produce the insights of the results, and he will be the one, or there will be another team. Whatever the results interpreted by the statistician will be used in order to develop a report called CSR. Anyways, that is not part of our work. So clinical study report will be developed. So there will be separate teams. So whatever the analysis data has to be developed. Now final stage will be, in our work, when it comes to SAS programmer work, we are going to do the reporting, guys. You can call it as TLF'S. Tables, listings, figures, or else tables, listings, graphs, anyway you can call. As for the analysis requirement, whatever reports that need to be submitted, so some templates will be provided, mock cells will be provided, and model reports will be provided to us. As for the mock cell, as for the model report given to you, we are going to prepare the reports by using your study data, by using your analysis data sets. There we are going to call TLFs. So if you compare ETL process, here it is ETL process, guys. First one, extraction, creating tables as for the SDTM standards. Once table is ready, transformation, doing analysis with help of ADAM standards. Once analysis is ready, generating the reports by using mock cells. So this is the role of SAS programmer in the clinical industry. Processing the clinical trial study data, developing the analysis data sets, and doing reports by using TLFs, tables, listings. For doing processing the clinical trial study data, for development of analysis, we have to follow standards that's been developed by the CDISC organization, developed standards called SDTM and ADAM. In detail, as part of, as SAS programmer in the clinical project, when you are working for a clinical project, these are the different kinds of work we are responsible to handle. And it's not again, only SAS programmer will be do all these work, guys. There will be teams. The work is going to be split. Whatever will be discussed here, what we are going to generate the output going to be another team input. Okay, don't think you are the one take care about creating SDTM standards. You are the one creating about analysis data sets. You are the one creating reports. There will be separate teams. There will be hierarchy will be followed. There will be project lead. There will be the senior programmer. There will be junior programmers, intermediate level programmers. There will be biostatistician, principal statistical programmer, statistical programmer. There will be separate teams. There will be different teams to deal with all these work, guys. Okay, as for your experience, for example, you have 5 years experience, guy. in that particular case, you need to create, spec document, or you need to do the annotation, spec document, and everything we'll discuss coming classes. Unless you are the beginner level programmer, you have just 2 years experience. In that particular case, even if it is a 2 years or beginner or 3 years, up to 3 years experience, so they are going to provide some spec document. As for the spec document, we need to create the tables with the help of raw data. Very simple. Pay also, it will be like simple pay only. But you are the one have 5 years or more than 5 years experience. Your work is going to be bit odd or difficult. So you need to do the annotation. As for the annotation, we need to create the spec document. So then your work is going to be spread. For example, few of the programmers are very good at DM dataset. They are going to create DM tables. Or another one, adverse event, another one. Like that, there will be separate program. There will be separate teams. So senior level, junior level, and intermediate level. Okay? No need to worry. So what we are thinking, when that will be higher level. But when it comes to the industry, you may feel, oh, okay, this is my work. In that way, you may feel, I'm sure. Now, what kind of inputs we do require? So where we do get all input, everything we'll discuss as our part of our project training. Okay, so as I said, not only SAS programmers involved in the clinical trial project, there will be several teams. What and all teams involved? What is the workflow? We'll discuss. Hundreds of people they are going to involve in this particular project. Okay, so but we should know, as a programmer, don't left in a blind spot. Okay, where we are, what is our workflow? You need to learn all teams work as well. That is very important. Okay, when you clear about what is the clinical data management team work, when medical writing team work, statistician work, and CRF design team work, if you know each and every team, and what is your work going to be, you will come to know. It is always good to know what is what end to end in clinical trials project. Okay. See here, why? Because as I said, so there is a team one, guys. Team one generated specific report. That particular report will be team to input data. So team two once generate the reports by using that particular data, that output is going to be team three input. Okay. So one team, next another team, next another team, like that work is going to be spread in clinical sector. Clear? Now, we should know. So everything we discussed, okay, fine. But first thing, we need to know what is called clinical trial process. Sorry, one moment. Let me open another presentation pages. Guys, our regular classes will be not like with presentation. Okay, I will try to write by using Notepad, by using an Excel sheet, by using SAS tool. I'm going to explain everything. So as this is introduction session, I'm using this presentation to easily remember. So now coming to the next one, drug development process, guys. You can say clinical trial process or clinical trial phases, drug development process, anyway you can call. So drug is nothing but, like for example, medicine that are available in the pharmacy stores, either medicine or vaccine or syrup or tablet, whatever it is, we can call drug. Why drug development process is required? What purpose? Definitely, why guys? Drugs are to improve the quality of life, right? Huh? There should be always new drugs keep coming. Okay. I'm talking about not only drugs, guys, either it will be new therapies or new medical devices and new procedures, new vaccine, whatever it is. Okay, in order to improve someone's life, yes, new medicines. So drugs are very important. So in drug development process, how many phases will be there? How many phases it will happen? See, guys, to explain this particular drug development process, I'm going to use US clinical trials research as an example, because all the research data that we submit to the, yes, 4 phases, regulatory called FDA. So if you want to get approval in United States, we need FDA approval, Food and Drug Administration. So why I am using US as an example here, because US is the one of the popular country. They have done a lot of clinical trials research when compared with any country, guys. That is the reason I'm using US example here to explain clinical trial process. There will be, European countries also, when compared with India, European countries, they also did a lot of clinical trials research widely. When it comes to European countries, what is the regulatory? EMAS. And in India also, there are a lot of research. The India, what will be the regulatory? DCGI. So one of the most popular one is United States research. That's the reason I'm using here US research clinical trial phases, guys. So in general, how many phases will be there in the clinical trials research? So in general, there will be preclinical studies and phase 1, phase 2, phase 3, phase 4. 4 different phases will be there, guys, after clinical trials, preclinical trial. Okay, what is preclinical studies? Preclinical studies is nothing but when the new drug formula is developed, that new drug formula will be tested on first animals. Okay, so that we can call it as preclinical studies. Before testing into the humans, first they will test on the animals. Once the animal preclinical studies is proven safe, then drug will move into the next step. That is nothing but phase 1, human clinical trials, guys. Okay, very fast. Once drug is proven safe, there is no risk with animals. Then we'll start our human clinical trials phase 1. See, when it comes to the preclinical studies, we'll not be working on the preclinical studies data, guys. Just for prove safe. That's it. We'll not collect any data and all. Okay? We'll not work on the preclinical studies data. Now, once it's proven safe in preclinical studies, then human clinical trials will start. In human clinical trials, there will be 4 phases. Coming to the phase 1. So first in human. Okay? It's like first in human clinical trials. Very first time, newly developed drug formula being tested on humans. Safety is the only parameter that will be monitored in the phase 1, guys. See here, for example, the drug is for headache, curing headache. Okay, so very first, we'll take healthy volunteers only in the phase 1, guys. Listen, phase 1 trials will conducted on first in human trials. Whatever the drug is given, they will monitor. Their safety will be monitored. So here we'll take in all the see here mostly in phase 1. Remember, tens of people, like 10 to 50 people only it will take, or 10 to 30 people, not more than that. They will not go behind it because risk is very high, right? As it is very first phase in human trials, although it is proven safe on animals, It is not that animals, right? Animals are humans, we are not compare, isn't it? So that's the reason. Before we want to proceed with phase 2, we need to 200% ensure whatever the drug we are testing on humans, the drug is safe enough, right? If it is a hazardous drug, if it is not going to give the safety, either we should change the drug parameters, or we change the composition, or we change the whole the study drug as well, or we can stop in phase 1 itself. But without safety, we cannot proceed to the phase 1. So once proven safe in phase 1 on human clinical trials, then we can proceed to the phase 2. See, guys, once phase 1 clinical trials is done, we are going to collect the data and we'll submit the data to the regulatory. Once regulatory approves, then only we'll move to the next phase, phase 2. Phase 2 clinical trials will start. One moment. Now, we have completed phase 1. Once we understand that drug is safe enough, we will proceed with phase 2. So here, guys, coming to the phase 2, we are going to consider 3 different peoples, guys, here. Number 1, we are going to consider all healthy volunteers in phase 1. See, in phase 1, all healthy volunteers we are going to take. But when it's come to the phase 2, we are going to take 3 different peoples. One is, what purpose we are developing the drug. For example, curing the headache. Let's say we are going to recruit healthy volunteers. In phase 2, I am talking about healthy volunteers. Along with healthy volunteers, who is suffering with that particular disease, whatever disease maybe it is. What purpose your drug is developing? If it is a cancer patients. If it is a diabetes patients. If it is a headache patients. If it is a fever patients, like that. So some of the people we are going to take healthy volunteers. Some of the people we are going to take suffering with that particular disease. And some other people also we are going to take suffering with other diseases as well. For example, if you have headache, or else if you have fever, headache is common, right? Or else body pain. Let's consider the people who are having fever. Let's consider people who are having fall under the headache as well. So 3 kinds of people we are going to take here. Let me explain. One is healthy volunteers. Another type of people is suffering with that particular disease, and another people is suffering with any other disease. 3 kinds of people. So what we can do, what we are going to consider here. See, healthy people, because already we discussed in phase number 1, safety. Safety is already proven safe. Now, you want to prove efficacy here. So here we are going to take hundreds of papers, guys, hundreds of people, either 100 to 500. First phase 1, tens of the people, hundreds of the people in the phase 2. Okay. See here, why we are taking 3 different peoples in the phase 2, because already safety is proven. Now we need to prove the efficacy of the drug. Efficacy is nothing but, what purpose you are developing this particular drug. For example, curing headache. It should cure the headache, right? That's why we recruited headache patients. It should decrease the headache. So then only we can prove the efficacy of the drug in phase 2. Isn't it? As risk is lowered in the phase 1, now we are going to recruit the hundreds of the people. So different kinds of the people we are going to take in the phase 2 to prove safety as well as efficacy of the drug. What is the focus of the main thing in the phase 2? Primarily concentration on efficacy of the drug. how efficient this drug is working on that particular participant. Okay, say I have given that particular drug to the headache patients, it should cure the headache. After giving that particular drug, it should reduce. Then only I can say that, so okay, this is helping me. My drug is working, right? Safety will be monitored as well as parallelly efficacy also will be monitored in the phase 2. So what is the efficacy means? Every drug developed with specific purpose. What is the purpose? The purpose, what extent is being solved. So finding the efficacy is very important. Okay. So now once you prove in safety and efficacy in the phase 2, then we are going to send to the data to the regulatory. Phase 2 data, once get approval, then we'll go to the next phase, which is nothing but phase 3. As safety and efficacy proven in phase 2, here we are going to take more people, guys, thousands of people here this time. So tens are phase 1, hundreds phase 2, and thousands. The drug is going to be working efficiently. Now we have already checked the efficacy in phase 2. Now I'm going to extend my people. Now a lot of people I'm working with, different genders, different continent peoples, different country peoples. I'm going to check this particular one is working with all type of people, all type of genders, all country people. The size of the population is going to be increased in the phase 3. Larger scale, we are going to use thousands of the people. Okay, in phase 3 also same story, guys. Safety. So this time hundreds of the people we are going to identify safety and efficacy, and whereas coming to the phase 3, thousands of people and different continent people, different ethnicity people, different genders we are going to use. Again here we should prove safety and efficacy. Once safety and efficacy proved with thousands of people, then now drug is ready for commercial launch. So we are going to send data to the regulatory. Once approved, phase 3 regulatory, once approved, then drug is ready for the commercial launch. Commercial launch is nothing but gets manufactured and it will be available in the nearby pharmacy stores or medical stores. So once drug is available in the nearby pharmacy stores, then phase 4 will start, guys. Coming to the phase 4 trials, you can say forced marketing trials. Forced marketing trials is nothing but once the drug is available in the medical stores, some of the drug will be like prescribed medicines. However, when the drug is available in the market in initial stages or in the early stage, some kind of feedback or some kind of services will be conducted on that particular product. So drug is safe or not. Suppose say, once after using this particular drug, either in the short term or in the long term, after consuming that particular product, if there is any abnormalities faced by the consumers, whoever been using it, if the abnormalities are something which is life threatening, the drug will be called back to the market from the market, guys. Maybe for the reason, or it will be completely banned based upon the feedback, based upon the various results. We'll keep hearing in the real world, like some of the drugs will be available in the market. Suppose say 5 to 5 years down the line, after 10 years down the line, if you go for the same drug, sometimes the drug is not available. They will say like that. Drug is not available anymore in the market. That means whoever using that particular drug, whoever been consuming that particular, there is long term, maybe or short term, they are facing some life-threatening abnormalities. Regulators will always keep monitoring, guys. Once drug is available in the market, okay, they will not left like that. They will keep always an eye on that particular product, and they will check long-term safety for that particular product. who is consuming that particular product, how they are doing. Okay, that is nothing but quality of life, right? So phase 4 is nothing but post-marketing trial, you can say, or else long-term safety they are going to follow. These kind of phases will be there. 4 phases. Fourth phase is post-marketing trial. Phase 1, phase 2, phase 3, and phase 4. And so, see guys, in order to reach that each and every phase, what kind of data we are going to collect, what and all teams involved, what is the role of that particular team, what kind of outputs we are going to generate by each and every team. Everything we'll discuss in coming classes, guys. What and all teams involved, each team work, what might be our work. Done? So that's all about your introduction classes, guys. Clinical trial phases and what might be roles and responsibilities of this SAS developer. That's what. Just introduction we discuss. Just a preview. In detail, 2 to 3 months classes we'll discuss. Am I clear? Any questions here? Can I get a confirmation, all of you?
It's clear, Bala.
Clear, right? Okay then. See you guys. See you tomorrow, same time, same meeting link. We'll continue with Teams work. Okay, in previous session, we started discussing the difference between using SAS programmer in clinical sector as well as in financial domain or other than any other domain, other than any clinical domain. And also we discussed what is so special working in clinical project, isn't it? So as I said, when you are working for any other projects other than clinical, no need to submit data for any regulatory, right? No need any approval, no need to follow specific standards. So as for your vendor, or as for your client, as for your organization, what are the terminology and what are the naming convention rules, what are the procedures are required, what are the reports are required, there we are going to generate directly. But when it comes to the clinical study data should be submitted to the regulatory for approval and review purposes. Hence, the data need to be standardized and organized as recommended by the regulators. So data should be standardized and organized following the standards called CDISC. CDISC organization developed standards called SDTM. Okay, I found this on the web for organization development standards. And okay, there are few more standards are available, guys. Not only CDISC standards, but CDISC is the one most popular standards developed by FDA. And we will be talking and learning about CDISC standards. That's the reason why. Once we start our project training, once we are done with introduction, what and all teams, what type of documents required to create SAS tables or SAS domains as per the SDTM standards, what is complete workflow, everything. Once we discussed 3 to 5 days classes, complete introduction, then we'll enter to the SDTM classes, guys, SDTM project. Okay. And also, we discussed what is the role of SAS programmer in the clinical sector. So what is the role? We are going to working on the clinical trial study data, right? That particular study data, as for the SDTM standards, we are going to convert. Once convert the data as for the SDTM standards, then we are going to create analysis datasets as for the ADAM. Then we are going to generate the reports as for the mock sets. So for that, we focused on the learning what is called a clinical trial process, which is nothing but drug development process. Also, we discussed yesterday. So how many phases will be there and all. So I created one Excel sheet for that. Let me show you. Hmm. So clinical trial is our clinical trial process should start with preclinical study, guys. A preclinical study is which is nothing but primarily conducted on animals, right? The primary objective of this particular preclinical studies are ensure that safety of the subject. Once we understand that the study drug or vaccine or else any therapy or else any medical device or any equipment, if it is safe enough, then we'll continue to the regular clinical trials, which is nothing but human clinical trials. So very first phase, what we are going to monitor here, what is the parameter, what is the endpoint? Safety. So next, once safety is proven, then we'll enter to the human clinical trials, which is nothing but phase 1. In phase 1, we are going to take only healthy volunteers, guys. Most of the cases, healthy volunteers only we are going to recruit. As for inclusion and exclusion criteria, what is inclusion criteria, what is exclusion criteria, I will show you. Okay, while recruit the patients, while once we start the clinical trial study, guys, very first we are going to create a protocol. In protocol, we need to define everything, each and every information, how we are going to recruit the patients, what kind of age groups, what kind of gender, what kind of ethnicity, what and all inclusions are required, what kind of exclusions are there. Everything we are going to define in the protocol. Today, I'm going to explain about protocol document if time permits. Okay, so inclusion criteria, exclusion criteria means, for example, let me show you simple protocol. There you will come to know. Sometimes, guys, sometimes we are going to recruit only healthy volunteers. So most of the cases we are going to recruit healthy volunteers. Sometimes, some studies where trials are supposed to conduct on the patients only, such cases like, HIV, cancer, these kind of treatments, these kind of drugs while conducting trials, we will be take on the patients only. In the phase 1 trial also, we will work on the suffering with that particular disease. In all other cases, majority of the cases, we are going to take the only healthy volunteers based on the inclusion criteria and exclusion criteria. Let me show you what is inclusion and exclusion criteria. So definitely it is going to be defined in the protocol document, which is around 4.2 or something. It will show you. There you can see. Yeah. So subjects should eligible for all inclusion criteria, and subject who not exclude, he will not match with any exclusion criteria. Then only he will enter into the study. See, each subject must meet the following criteria to be enrolled into the study. Healthy male or female, and it should be age 18 to 25 years between only. And female must be non-childbearing potential. And again, you can see body mass index should be 18 to 30 kg for M square. With the minimum body weight is 50 kg only. And so this is called screening, guys. Screening. Okay, inclusion criteria, exclusion criteria. Before recruit the statements, we are going to very first we'll take the informed consent form. So an informed consent document should be signed by each and every subject. Informed consent form is nothing but, in general, when we go to the hospital, if any of our relatives or family members are admitted in the hospital, they are going to ask you before going to the surgery or something. They will ask you, right, sign this particular form and like that. What and all procedures going to be happen and everything. They will mention that particular form. Same like that. So once a subject entered to the study, what and all procedures is going to be done. everything they are going to mention that particular instant consent form. I will show you informed consent form also later. So very first, before stepping into the screening is nothing but inclusion criteria, exclusion criteria should match. So here we are going to check. So once informed consent form signed, that particular person only eligible to the screening, guys. So this is also should be finished before screening. Okay. And so here some of the inclusion criteria, exclusion criteria. For example, you can see pregnant or nursing females, they are not included. They are going to exclude. If the particular female is pregnant or nursing females, and history of febrile illness within 7 days prior to the first dose of this drug, and a positive urine drugs screening day 1. So there will be some exclusion criteria. Medical conditions or medical history presence should not be there. Then only they are going to include. If you have any medical history, cardiac arthritis and impaired renal function, thyroid disorders, these kind of medical histories, if you have, they are not going to include. So these are the, so it's not only these. So for this particular study, these are the inclusion criteria and exclusion criteria. So based on the study, inclusion criteria and exclusion criteria are going to be different, guys. Each and every study is unique. Each and every protocol is unique. But standard formats we are going to follow, but the matter is going to be different. Okay, we'll discuss in detail about protocol document. Okay. So before recruit the participants, we are going to check the inclusion criteria. See, first, informed consent form, guys. ICF sign. Once informed consent form signed, then he is eligible for screening. So once screening is completed, in the screening, they are going to check inclusion criteria, exclusion criteria. Once they are eligible for inclusion criteria and all inclusion criteria and exclusion criteria done, then only they are going to recruit to the clinical trial study, live trials. Okay. So who is eligible to clear the entire criteria? Then only the subject will enter to the study. Then only subject will go to the next phase. That is nothing but treatment. So informed consent form and screening, randomization and treatment, follow up. What and all stages will be there? We'll discuss later. There is separate flow. We'll discuss, guys. Okay, first concentrate here. Okay, and so once inclusion criteria, exclusion criteria are clear, then only subject into the study. That is the first step, guys. Next, once screening is completed, based on the inclusion criteria, exclusion criteria, we'll enter to the study phase 1. So phase 1 mostly around 20 to 30 or else 30 to 50. So mostly, guys, 10 of the peoples only, not greater than 100, below 100 peoples only. So some of the websites, they are going to show 30 to 50 people. Some of the websites going to show 10 to 50 people like that. Some websites like that, 20 to 80. But whatever, below 100 people only will participate in first phase. Why? Because the risk is very high. First in human trials, right? Okay. So what is the primary objective for phase 1? Safety. And is there any toxic effects? Okay. How? Drug is, distributing throughout the body, will access the toxicity. So main purpose is phase 1 is safety. Okay. So once phase 1 completed, so once is there any toxic effects are there, they are going to monitor, guys, in the phase 1, because very first in human clinical trials, right? So this is the main parameters in phase 1, safety. So once phase 1 is clear, we are going to send that particular data to the regulator for approval. Once approval, see phase 1, it will take several months, guys. Phase 1, and success rate will be around 70%. Phase 1 clinical trials. Okay. So once the subject is survived, if there is no side effects of that particular drug or medicine or that particular device, in that case, we will conclude that okay, subjects are safe enough. After taking the particular study drug, there is no toxic effects. Then we'll enter to the phase number 2. One moment, guys. In phase number 2, we'll increase the base. So this time, hundreds of the people. So there, whereas coming to the phase number 2, we'll go with 3 different peoples, right? 3 different volunteers, healthy volunteers and subjects suffering with same diseases or subjects suffering with other diseases most related to this particular disease. These kind of people, they are going to recruit them. Then here, the primary object is going to be efficacy of the drug. How efficient the drug is going to be work. So simply I can say that short time of safety is the main goal here. Okay, is the drug working or not? Overall object is going to be, let's say if I am going to take a COVID vaccine, for example, the object of that particular vaccine is going to be, it should be prevent or it should at least reduce the severity. Am I right? In some cases, it is going to prevent the person getting affected with that particular COVID or any other diseases. The severity of COVID on that particular human body at least restricted, or else the severity is less. So these kind of conditions we are going to able to meet. Can I say that? If I able to meet these conditions, okay, I can say that my drug is safe enough. In my first phase, I cleared with safety, and second phase, efficacy of the drug, how efficiently my drug is working. So efficacy. So short-term safety we are going to monitor in the module number 2. So phase number 2. One moment. 1 minute. Okay. So once if safety and efficacy proven safe in phase number 2, then we'll enter to the module number, phase number 3. We are going to extend this base again, thousands of the people. So here also, we are going to take healthy volunteers and patients with same diseases and patients with other related diseases. Okay. So here we are going to monitor both safety and efficacy in phase number 3 with a large number of the patients. So long-term safety also we are going to monitor in phase number 3. So once we are done with phase number 3, then once approved from the regulatory, drug is going to be commercial launch. It will be available for the public market, public people. Right? So here, phase number 2, guys, it will take up to 2 years. Phase number 2 in general, and phase number 3, it will take around 1 to 4 years. Long term, phase number 3 it will take, because efficacy to prove and safe efficacy, we are going to spend most of the time in phase number 3. And again, coming to the phase number 4, we are going to access the drug risk mainly. Is there any risk with this particular drug after using? See, because phase 1, phase 2, phase 3, based on the inclusion criteria, exclusion criteria, we are going to recruit the patients, right? Okay, fine. But when it comes to the phase number 4, it will be available for common public, right? All kinds of people, they are going to use that particular drug. So they maybe get some side effects, okay, after taking that particular drug. So here in the phase number 4, we are going to monitor, is there any risk with that particular drug? Is there any side effects? Is there any adverse events after taking that particular drug? And post-marketing surveillance, we can say that. What might be praised for this particular drug? What is the suitable price for this particular drug? What is the maximum retail price? What is the marketing strategies I need to go? Who am and all I link up with that particular uh, person with the penetrate in the market. All these things under the phase number 4. All these phases are done during the each and every phase. We are going to collect the data. Data collection is going to happen, right? What is going to be data? We are going to collect the data from the subjects. For example, initially phase 1. Let us come to the phase 1. We are getting the people information, that is, let's say, who is participating. We need to track them. What is the name of the person? What is the gender of the person? Which country he or she originated, and what is going to be race of that particular person? What is the ethnicity? What is the birth date? All this information we can say demographics, which is called DM. In this particular domain, we are going to store all the data. We'll collect the data. We'll create a table called DM. We'll discuss once start the project training. This is called demographics information. And so patients, so demographics data set is completely about patient information, guys. In and out about complete subject information. And then again, there are 4 phases, right? And first I'm going to do the screening. The screening in the sense, as for the inclusion criteria, exclusion criteria only, I'm going to recruit the patients, right? That is called screening. All the people, all the volunteers who have cleared the screening process, they will enter to the treatment. In between some of the phases, some of the people not able to clear the screening, guys. They may or may not, because they are not able to meet all the inclusion criteria, exclusion criteria. They will not enter into the treatment. Who clear the screening, that particular person only will enter into the treatment. All the screening details, my inclusion criteria, my exclusion criteria details also need to be collected. What is inclusion and what is exclusion, I need to take in. That is nothing but domain called inclusion and exclusion, TI. Domain is nothing but table, dataset, guys. Here we are going to discuss TI later. So I'm not explaining all the domains, what and all we are going to discuss. Just I'm giving the preview, what kind of data we are going to collect. After collecting, what is going to be our work. Okay. So demographics, see, all the patient's information we are going to store we are going to collect in a demographics data set. All the inclusion criteria, exclusion criteria information we are going to store in inclusion and exclusion criteria, TI. The people who not made that particular inclusion criteria, exclusion criteria during the study, there is another domain. Okay, IE, inclusion criteria, exclusion criteria not met. There is another domain we are going to discuss. So the domain is IE, not TI, just IE. Next, just example, guys, I am telling you here, not that much detail. And don't worry, all these we are going to discuss one by one, one by one. There is a order. Okay, otherwise we are going to discuss each and every domain. Once we are clear with this particular one, and we are also going to say, screening, when it comes to the screening, one moment. Your treatment will start. Once done with screening, your treatment is going to be start. After treatment, there is a follow-up. So you can say that there are 3 phases. First is screening, next is treatment, next follow-up. Okay? Each of every element is a screening, here. Sorry. Screening, next treatment, next follow-up. This is one element. This is one element. This is one element. Okay, all your elements information, all elements details, we are going to store in one table that is called trial elements or planned elements. Anyway, you can call TE dataset. We are going to discuss. Or else, this is planned one. Trial elements is a planned one, and actual append elements is going to be there. Another domain called subject elements, SE domain. Each and every element again, we'll say separate modules we are going to discuss. Okay, and again, so each and every visit there will be separate treatment. For example, first visit, so here treatment is not only for 1 week, maybe 2 weeks or 3 weeks or 4 weeks or 6 weeks or 10 weeks, depends on the study. Okay, so each and every visit information, visit details, we are going to store in a table called trial visits. TV. And subject visits, SV. Trial visits, we planned one. So maybe subject may or may not. He may come on the time or else before the day or after the day. So maybe he will come to that particular treatment. So we can call that particular one subject visits. SV. Understand what I'm trying to say here? All we are going to do is in general. So here, this is very lighter way I am explaining, guys. We are not discussing that much detail. Okay. First, inclusion, exclusion. We'll keep the data into one domain. Next, each and every information, every element information we are going to store in one domain. And all participant data we are going to store in one domain, one table. All visits information, scheduled visits and unscheduled visits information we are going to store in one domain. So, like this, we have different domains. Around 40 to 50 domains are there, guys, to collect the data during the study. Okay, once we started the drug to the given participant, study drug, your investigator has give some comments. So he is going to write comments on particular pad. That is called comments domain, CO. All the comments going to store somewhere in one table called CO. We are going to collect that like this. All the data we are going to store in different domains, different tables, guys. See, remember, instead of sharing around 50 to 100 pages of the document, if you create a simple table to summarize the table, if you share that particular table to the client, which is very easy, right? the tables only we are going to create, guys, to send your clinical trial study data to the regulatory for the product approval. While creating the tables, we should follow some standards. Those standards are nothing but SDTM standards. Every variable should have specific length, specific type of character. So like that, we have some rules. All the rules we are going to discuss, all the SDTM terminology and definitions we discuss once we start the SDTM project. We have different rules. As for the rules, we need to store the data in particular domain. Okay, so here your data collection is going to be happen. Whatever the data we have collected, so based on that particular data, we are going to prepare the report. We will prepare the report. The report will be submitted to that particular authorities, regulatory authorities based on the country. If it is a USA, FDA. If it is a European countries, UMAS. If it is India, DCGI. Once they approve, the drug will come to the market. If they are not approved, if they reject that particular drug, it will not come to the market. So each and every phase, once completed, should send for the review and approval purpose. Once approved, then only the trial is going to move to the next phase. Okay, once preclinical study is approved, then they are going to the phase 1. Once phase 1 approved, then phase 2. Phase 2 reviewed and approved, then phase 3. Phase 3 reviewed and approved, commercial launch. Drug is going to be commercial launch. So then phase 4, post-marketing surveillance, long-term safety, how the drug is working with all gender people. Is there any risk? Is there any side effects? If that particular side effects are life-threatening side effects, then we'll call back from that particular drug to from the market to the bank. They will ban that particular drug. This is complete clinical trial process happening. So this is in a nutshell, I can say that this is our complete clinical trial process and workflow. So for this, what and all teams are going to be involved to collect the data, to analyze the data, and to provide drug to that particular patient, to monitor the participants, to recruit the patients, to clear the protocol, to create the protocol document, to create the CRF document. So what and all teams will be involved in complete whole clinical trial project life cycle. Now we are going to discuss about teams. This is also very important, guys. See, there will be several teams we need to discuss. Sponsor, first team, then protocol team, then CRF design team, site management team. What is site, what is CRF, what is protocol, everything we are going to discuss. Don't worry. Clinical data management team, data analysis team, last but not the least, medical writing team. This particular team only, they are going to write synopsis based on your reports. Okay, so all these teams we are going to discuss coming 2 days or 3 days classes, guys. While discussing protocol team, we'll go with the protocol document. While discussing CRF design team, we'll go to the CRF document and all the things we'll discuss. Okay, so for all these clinical trial process, who is going to initiate first? There should be one of the person should initiate, right? Complete process. for either preclinical studies or as phase 1 or phase study and all these things. Who is going to initiate? Sponsor, guys. Sponsor is the one, he is going to initiate complete clinical trial study. So to do this particular study, we require a lot of money as well as a lot of time, lot of equipment, lot of resources. So someone need to be sponsor. Someone need to be spend the money, time, resources to start your clinical trial because it is a time-taking process. Generally, you know, sponsor is going to be initiate this particular whole thing. Who is that sponsor? Sponsor can be an individual person, or it can be CROs, contract research organizations, or else it will be group of sponsors, group of people. Hmm? Or it can be, government sector, government entity, or it can be private pharma companies. Or medical or pharma companies. Anyone can be. So these are the one who is going to provide or who is going to start this particular clinical trial. So these are the ones. Sometimes maybe combination of the government sector and private sector. Sometimes only private people, or sometimes individual people, or else any group of people. Okay, sometimes R&B, research and development department. So these are the one, right? Mostly they will concentrate on medicine or developing a vaccine or any medical equipment, right? So your R&B, other one can be part of your clinical trials research. Either pharma companies or medical institutes or else any government sector or government entity or else any individual person. Sponsor can be anything, guys. Any organization. So very first team should be formed, sponsors team. Sponsor is going to be funding for complete clinical trials research. It will take crores of money. Yes, Prithvi. So not only money. So to conduct the clinical trials, there would be place. There should be site, either health hub or hospital or else their home or else any medical center, clinical trial site. Everything they should provide. Okay, in terms of money or time or resources, everything legally. Whoever funds for the clinical trials, we can call them as sponsors. So sponsor is the key for your clinical trials, guys. Hold the clinical trials. He is authorized for all the pay participants, investigators, all the things. He is the one. Command the complete clinical trial process. How it should happen, how it should start, how it should end. And any approval for any trial, sponsor is the main person. He is going to take care. So very first team, it is going to be formed sponsors team. So once sponsors team is ready, so they are going to start the next thing, protocol, guys. They will form protocol team to create a protocol document. Protocol is nothing but planning. So like in order to start anything, there should be plan, right? So here, protocol is the team. They are going to plan, complete your study. Okay, so protocol team. So Francis is going to frame the protocol team, very keen to take the next step, which is forming the protocol team. I mean, they will recruiting the protocol team. What is the responsibility for the protocol team? In order to start a clinical trial starts, so there should be purpose, right? What purpose you are doing this particular trial? Every study has unique purpose, either to launching new drug or launching new medical device or launching new therapy or launching new vaccine or could be anything. So what is the purpose of this particular clinical trial should be defined very first in that particular protocol document. Protocol team generate protocol document. They will create. In protocol document, very first, they should mention purpose. Purpose of the study. And what is the primary objective and secondary objective? Hmm? All the objectives will be defined in the protocol document. Actually, protocol is nothing but complete planning. What is the main objective of the trial? What is the trial design? Huh? So all these keywords I am going to explain, guys. All keywords, complete terminology I am going to explain. So listen. So what and all we need to mention in the protocol document. Let me explain. If I write, it will take time. Anyways, we'll write later. As a SAS programmer, guys, if you want to work with clinical trial project, all these keywords, what we are going to discuss, you should be aware of this particular thing. You should learn. First, in a lighter manner, I am going to explain now. So keep on once you start your project training in detail, there I am going to explain. Okay. So here, what and all keywords you should be understand. That is what we are going to discuss now. Now, what is your study design? It is parallel study or a crossover study. What is called parallel? What is called crossover? Later I will explain. They should be justified in that particular protocol document. Okay, it is not only randomly designed. There should be proper justification what you are going to do based on which phase of clinical trial. Is this phase 1 clinical trial or a study plan? Yes, which is simply you can call study plan document. Could be. So what kind of phase it is? Phase 1 trial or phase 2 trial or phase 3 trial? So what is the sample size you are recruiting? One moment. If it is a phase 1, how many people you are recruiting? If it is a phase 2, how many number of people you are recruiting? How many, the new drug will be tested on how many number of the people? We can call it sample size. Why that particular sample size you are taking? You need to justify in that particular protocol document. And who will recruit it? What kind of patients you are recruiting? Males or females or only adults or only kids or mix of everyone? And why those kind of people? Again, you need to provide the justification for that. And again, is it randomized trial or single sequence? So randomized is nothing but treatment will switch, guys, but based on the participants. But single sequence means throughout the study we are going to use only one study drug only. I will explain later. So if it is a randomized study, why? What is the reason for the randomization? Huh? If it is a single sequence, why single sequence? Justification should be provided in that particular document. And from which ethnicity background the people are going to be recruited, or from which demographics or which geographic location people are recruiting, and again why you are recruiting those kind of people. And again, coming to the next one, what criteria will be followed? Inclusion criteria and exclusion criteria. I said, right? Why you are following that particular criteria? And again, recruiting the people, it is not that whoever come to the voluntary will be taken into the trial. Not like that, guys. So there will be certain criteria. We should follow the criteria before stepping into the study, right? If that particular person, if satisfies the inclusion and exclusion criteria, then only they are going to enroll. I said already. Otherwise, they cannot come into the trial. So what is that particular criteria will be followed and why? Again, there should be justification we need to provide in that particular document. And what is the sequence of procedures will be followed? Randomly, if you use the procedures on that particular paper, they will not agree. A clear picture of what and all procedures will be happening. The trial should be explained to the particular concerned person. I said, right, consent form, we are going to sign very first. What and all procedures. You should be explained that particular participant, whoever is the participating into the particular trial. Once they confirmed, they signed that particular form, then only they are stepping into the next phase, next step. That is nothing but screening. We'll discuss all the steps later. All the sequence of the steps and procedures need to be explained, right? And what kind of documents will be signed by them and why you are signing, that should be justified. And what kind of data you are going to collect from them. It's not that randomly the data can be collected from them. So there should be always a plan. So end-to-end plan, what kind of data you are collecting from the each and every participant. So and so data you are collecting. You need to justify in that particular thing in protocol document. And the collected data, where you are storing, it is going to store in a database, or it is going to store in a document, or it is going to store in an Excel file. Like how it will be stored, you should mention in that particular protocol document. And then privacy, the data integrity and data security, that is also very important, right? Data is nothing but it's person behind. It's an individual behind. How the data integrity and data security will be maintained, and what is your data privacy? How data privacy will be maintained? You should mention in that particular document. And what kind of analysis will be done with that particular data? For example, say if there is a phase 1 trial, what is the endpoint for the phase 1 trial? What you need to prove with phase 1 trial? You need to prove safety of the drug. Indeed. Efficacy of the drug. In phase 3, safety and efficacy of the drug we need to prove. So based on that particular phase, what are the endpoints? What kind of analysis should be done with that particular data? All the points, each and every point you need to mention in that particular document. End-to-end plan of your clinical trial. How it will start, how it should end, completely determined by the protocol document, guys. In case of there, if there is any deviation from the protocol, then what is the violation? Is the violation or deviation done by the management team or by the participants? If there is some violation or deviation happen, how that will be data is going to be captured? Suppose, say for example, that sometimes there will be some exceptional violations will be happen. If there is a participant or from the management side, if the violation is happen, so like if it is a something considerable violation, not that much effective. If it is a considerable violation, even though considerable violation happen, how that particular participant will be terminated from the trial? If the violation is something which can be exception, and what basis they will be continue on the trial. So all these points will be discussed in the particular protocol document, guys. Start to end. How a trial should be start? Yes, Thomas. Exactly. How a trial should start and how a trial should end. Completely determined by the protocol document. Once the protocol document is ready, once the protocol team is ready, it will be submitted to the regulatory guys. So very first, sponsor team and protocol team, they will sit both together and end to end their plan, what they are going to do with all the phases, what kind of patients they want to recruit, what kind of procedures they will start with that particular statements, what kind of analysis they want to do. Everything they are going to mention in that particular document. Once the document is ready, if they are satisfied with that particular document, they are going to send to the document to the regulatory. So regulatory reviews your document. If they are okay, if they are satisfied with your document and your plan of action and everything, then they once they approve, then you are going to start your live trial. Then only guys, once they approve the protocol, all your plan, then only we need to start your live trial. Suppose the protocol requires some kind of revisions, so they are not expecting, they are not approved. They set some recommendations. For example, you send version 1, they are recommended some versions, regulator recommended some revisions. So you need to modify your document again. So don't go with the modify original copy. Original copy will not be modified, guys. Let's say, for example, this is the protocol. For example, there is a ABC study. There is a protocol version 1 created. So they sent to the regulatory. They recommended some revisions. Then we need to copy to the main protocol, copy and paste, Control C and Control V. Then with the modifications, as the recommended modifications, we are going to create version 2, B2. Still, if they are not satisfied, if they some recommended some revisions still, then we are going to create. We'll not modify main versions. We'll copy and paste and we'll create the new versions. And each and every version we need to store somewhere. All the modifications will be done and based on the copies only, not with original versions. And then, okay, version 3, if they approved, okay, then your live trial is going to be started. Okay. So as a SAS programmer, we need to understand all these guys. We will be talking about, clinical trial study. What and all teams are involved. What will be each and every team work. What will be your work going to be? Where you will be part of this particular all the teams work. Everything we are going to discuss one by one. So first sponsor team we discussed and protocol team what they are going to do we discussed. Next, CRF design team we'll discuss later. Tomorrow we'll discuss protocol team guys. Yeah, sorry, tomorrow we'll discuss CRF design team and site management team and all. So that's all about today guys. We'll continue tomorrow. Here we are. Okay. So we are discussing about teams, right? Sponsor. The sponsor can be anyone, individual person, or contract research organizations, or else a group of people, combination of the government and private sector peoples, or else any pharma company and R&D department. Sponsor can be anyone, guys. He's the one going to take care about the end-to-end trial, payment, money and resources, teams recruitment, everything. He's the one. It will take. He's the one who's going to take care of. He's the one to submit the study data to the regulatory. So of course, through help of the teams only, and everything he is going to do with help of teams only. There are a lot of teams involvement is there in the clinical trials research. Okay. Next, protocol team. So protocol team, they are going to generate the protocol document. So here to generate the protocol, he is going to recruit some of the healthcare professionals and scientists. Okay, so some of the members he is going to recruit to generate the protocol. So protocol, while generating the protocol, uh, they are going to follow one standards, guys, called PRM standards to generate a protocol document. They are going to follow PRM standards. So yes, these standards are also generated by CDISC organization. Okay, so protocol is PRM means protocol representation model, which is developed by CDISC organization. What is the user protocol? Already I said. So here in detail about protocol document we'll discuss, guys. In detail, so as a developer, if you want to work with clinical sector, especially in the clinical domain, clinical trials research study data, if you want to work, you should thorough with protocol document. Some of the keywords you should know. Of course, we are not going to create protocol document and all, but through protocol document only we are going to work. We are going to create the SAS datasets and all. Okay, every clinical trial will be a unique purpose. So based upon that, they are going to create protocol document. Okay, now, so here, so as we are discussing about protocol team, I want to explain about protocol document, guys. Only to discuss about protocol document, it will take 2 day sessions because that much detail we need to learn. Some of the keywords, study design, what is the enrollment process, which type of study is it, open label or blinded label, one label, triple blinded or double blinded. Hmm? So everything, so crossover or parallel. So what and all keywords you should thorough. I'm going to explain here one by one. So this today's session is completely about protocol document, guys. So what and all we should include already we discussed just lighter way, right? So in detail we'll discuss here. So to generate the protocol document, sponsor is going to recruit some of the healthcare professionals and scientists and some of the statisticians. Our investigator, he is going to recruit and he is going to generate the protocol with all combination of the persons. Because so statisticians have their own plan. What is his plan? So definitely we need to include within the protocol document. Statistician, they are going to generate a SAP document. What kind of analysis is required to do that? So SAP, what and all we are going to do with SAP document? Definitely we need to include that one also in the protocol document. Everything end to end. How trial should start, how should trial end? What and all procedures we are going to do with from the beginning to until finish all phases with all the participants, with all the teams. Workflow we are going to assign here. Now, I will show you sample protocol document here. Hmm. Listen, again, I'm telling you, SAS programmers doesn't involve doing protocol document. We don't need to learn in detail about the protocol document. But for narrow purpose, I'm sharing all these. Okay, so some of the keywords are very important. We should know. One second. Hello. Hello. Yeah. Sorry, guys. There is a guy, and there are a lot of questions. Okay, I will show you protocol document. Very first document, guys, they are going to create protocol. Okay, without protocol approved, we are not able to start clinical trial live. Okay, so first document. See here protocol. For every protocol, I have unique protocol number or study number. Every phase of study, again, there is a unique ID will be provided. So this is study phase one study protocol document, and this is study number. So what is the product? This is the product. IND number, investigational new drug number. This is the one. And what trial of phase it is, who is going to be sponsor, sponsor details, contact details, and medical, who is going to monitor complete clinical trial site. Medical monitor. See guys, one more important point about protocol document. Each and every phase, whatever teams we are going to discuss, whatever work we are going to discuss, it will be carried out complete all phases, guys. Phase number 1, phase number 2, phase number 3, and phase number 4, everything. Okay. Study title, open label and everything I'm going to explain. Phase, primary objective, secondary objective, everything study design. Okay, we'll go with one by one. So this is simple protocol document, around 66 pages of the protocol document. So once we learn some of the definitions, then you can easily go through with this particular document. So this is content of protocol document, inclusion criteria, exclusion criteria. What is the access points? Is there any violations, deviations? Everything it will be there in this particular protocol document. Investigator signature, sponsor signature, is there any events, adverse events, all the information. Okay. Protocol definitions. So first one, clinical trial site. This is nothing but where the clinical trials are happening. The location. It could be hospitals or it could be, I will note down. Theory part, guys. There are a lot of theory parts. It will take time to write and everything. Anyways, important points I'm going to write. It could be hospitals and clinics, or else any health hubs, or else medical centers, or it could be home as well. Yes, Thomas, I'm going to provide running notes as well. Yeah. Once done with all introduction classes, I will share all running notes with you. I will update in the drive. No worries. No need to write anything, okay? Just listen. So clinical trial site. So this is the place where the clinical trials are going to happen. So, so here the person who monitor we can call principal investigator. I'm not talking about investigating here. Who monitors clinical trial site? We can call What is the investigator's work and everything, I will explain. Usually, there will be only one principal investigator, guys, in each site. Suppose if site is big, or else if it is divided into different zones, then may have principal investigators for each and every zone. Or else clinical trials, there are multi-site and multinational also available, guys. Multi-center, multinational. Some of the trials is going to happen multi-center, multinational. So multi-center means same location, same country, multiple locations. For example, if you are coming from India, different. One of few of participants is going to recruit from Bangalore, or few of the participants. So Bangalore, there is a one trial. So and again, Hyderabad, or else Chennai, or else Tamil Nadu, or Kerala, like that. Same country, different locations. If you conduct same trial, then that is called multi-center site. Different locations are different cities. Multination. So where some trials require diversity, guys. So different types, different geographic peoples also required in such cases. So different countries also we are going to do the clinical trials. Different countries people we are going to recruit. Okay, so clinical site can be either if it is a single. Hi, Kavi. So multi-center, multi-nation, or else just a single site. So completely depends on the time, guys. Okay, and again, um, one moment, guys. I need to drop it. Well, just one minute, not more than. Okay, and moving on to the next one. This is about clinical trial site. You know, there are the formalities. I said right. We always use the documents like informed consent, ICF form. Hmm? So informed consent, no need here actually about informed consent details. I will skip. One more important point I want to tell you about this particular study site. So clinical trial site or else study site, anyway you can call, guys, this one. Hmm? So as I said, we have single site or else multi-site. So multiple locations or multiple hospitals or different location style is going to happen. Hmm? Usually phase one trials, listen, so based on the phases, phase one trials, definitely it will be stick to one location, only one location. Because just, or 20 or 30 people like that, they are going to deal with phase one, right? So most of the phase, what and all domains we are going to discuss, what and all tables we are going to create all that particular data mostly for phase two and phase three information only, guys. Phase one just 10 to 20 people, the max 30 to below 100 people only we are going to do, right? Phase one trials. Because risk is very high, right? Phase one trials. Hmm. So whereas coming to the phase two and phase three, larger study population. Such scenarios, we need diversity. So such cases, we are going to depending on, different locations, different countries, people. So yes, so here people's also they are going to recruit based on location. Sponsor only, guys. Sponsor is the one going to take care about everything, recruitment and everything. There is separate team. So signing the informed consent form, finishing the formalities, and checking inclusion criteria, exclusion criteria, everything. There is separate team, site management team. They are going to take care. But every team, they have a relation with the sponsor, guys. Okay. So once coming to the clinical trial site, these are the examples. Next, objectives, study objective. This is also very important to know. So, which is nothing but what is the purpose of the clinical trial? Purpose of the trial. Every trial have any purpose, right? The purpose we are going to mention in this particular study objective column, guys. Like each and every value, trial site information, yes, there is a paragraph. Study objective information, there is a paragraph. All information, what and all we are going to discuss, all the information, it is going to be in a paragraph. Like, see here, there is a paragraph, right? And again, this data, we are going to create these paragraphs into table format. We have separate domain. See here, paragraph. Everywhere there is a paragraphs. Okay. Um, Purpose of the trial. So mainly, what are the primary objective for my trial and the secondary objective for trial? Primary objectives. Primary objective means main goal. What is the main goal for phase one? Any one of you? What are the endpoint for the phase one? We discussed already. If it is a phase one, let's assume you are creating a
Safety,
protocol. Yes. Phase one, safety. Phase two, what is the endpoint? Phase three,
safety and efficacy.
safety and efficacy. Phase two and phase three. Okay. Yes. Here we are going to assign in primary objective. This is the phase. So I want to prove. So these are the primary objective for my this particular study. Yes, we need to mention the primary objectives. Main goal of this study. And secondary objectives. Secondary objective means, additional goals. I can say that. Additional goals in the sense like observing the mechanism of the drug or behavior of the drug. Like that. What is the pattern of the drug once the drug is given to participant? How he is behaving. Guys, one more important point I want to tell you: participants, people, subjects, population, all keywords are same only. Okay, so some of the, as you are a programmer, so you are going to call participants as a subject. Sponsor is going to call people. Any other team they are going to call like participants, site management team. Okay, based on the person how they are calling, the names will be different, but subjects, patients, people, or study population all are same. Who is participating in the trial? Okay, additional goals. Examples: mechanism of the drug, pattern of the behavior. Once given that particular drug to the participant, is there any changes in that particular participant behavior? Pattern like, how drug is working, which we can call as mechanism of the drug. How the quality of life is impacted. For what reason generally we are going to generate or we are going to create these new drugs? Hmm? How it is impacting, how it is working. Is it improving quality or not? Because new drug means how the quality, I mean the person quality of the life, it's been improved, or else is there any adverse events? So additional goals, all additional goals. So assumptions, I can say that we need to mention here. Okay, primary objective means main goals, purpose of the study, purpose of the phase. If it is a phase 1 protocol, safety; phase 2 protocol, safety and efficacy; phase 3, safety and efficacy. Primary objective, and each and every primary objective there will be additional goals also will be there. So for example, if it is a phase 1, what will be the safety is the main thing. What will be the secondary objective? Phase 1.
Safety, sir.
Along with safety, we are going to check, is there any risk side effects which is cause death. Okay, so what kind of adverse events will be there? So that one also we are going to monitor. Secondary objective, not primary objective. First primary objective is going to be safety only. So like that, each phase there will be primary objective and secondary objective we need to mention. Next, coming to the next one. Usually the main reason, everything we need to justify all information in that particular protocol document. Everything very clearly. Okay, then only regulatory going to approve your protocol. Once you approve the protocol, you can start your live trial. Next one, come to the background. Okay, if you write something, for that you need to give the explanation. Okay, for example, you are using this particular 200 mg or 300 mg. Why 200 mg? Why 300 mg? Why you are increasing? You need to mention the reason for that. You're taking out of 300 participant total, just 50% one group and another 50% another group. How do you decide that particular group? Each and everything we need to mention. Okay, one by one we are going to discuss. Background. This is also we need to define in protocol document, protocol team, they are going to define about background. Background is nothing but always, this particular study. For example, this study is related to addict curing drug. Is there any medicine for this particular drug? Is there any trials happen in previously? If it is happen, what happened? But what happened with those trial results? Huh? Is there any clinical trials are conducted by any other organization in any other location in same product or similar product? If happen, we need to mention that what is the results for that particular study. Background also very important. And what is the difference between existing one and new one? Existing one means already, for example, let's assume there is a organization conducted trial on this particular product only. The particular product is rejected. Same product you are trying to do the research. What is the new formula you have? What is the difference between existing drug and your new drug? What is your formula? Okay. So 1 second. So background information we need to provide in background. One point I will write. I will not going to write everything about background. Simply I can say that same product with any organization. Same product in any organization. Fast trial. It happen. Guys, please ignore my spelling mistakes. What will be the result? And what is the difference between existing one and new one? Okay, existing, they are going to provide some of the information, right? These are the doses I'm going to use, these many people I'm going to recruit, complete. And once failed, again you are starting, but how we are starting? What are the changes you made? Everything, okay? And again, so when I come to the background, what type of drug it is, what type of form, or else it's like an injection, or else saline, or else tablet, or else vaccine, form of the drug, or else lotion, or else syrup, or any other forms if you have. How the doses will be given. So route of the administration, how long it will happen, how frequent it will happen. So all that we need to explain briefly in this particular background section, guys. Background. Next one, and very important one when compared with all these, as a developer, as a programmer, you should know about study design. Study design. There will be different types of study designs, guys. We'll discuss everything. There will be different branches, I can say that. We'll go with one by one. First one, what type of trial it is? Open label, single blinded, and double blinded, triple blinded. These 4 points we need to discuss. At the same time, single sequence, randomization. Again, under randomization, we need to discuss few more points here. Parallel study, crossover. Okay. So first coming to the open label. Open label is in the sense the new drug which is we are going to start to study on that particular drug. The drug is open to everyone on the site. I mean, whoever is taking the drug, participants, aware of that particular drug, and whoever is giving the drug to the participants, investigators. Yes, they are aware of drug, and whoever is monitoring the parties, that particular clinical trial site, clinical site management team, they are aware of drug, and who is responsible for collecting the data and doing analysis. Everyone, it will be known, guys. Everyone aware of that particular drug. I mean, the one who is working with the data, the one who is providing medicine to that particular participant, who is participating in that particular trial. So everyone, it is like an open label, open in the sense, it's like an open source. Anyone can know that particular product. It's like open to everyone, open to public. You can say that is the one open label. Everyone in the study aware of the drug, guys. Simply I can say that. All the teams. Open label. Single blinded. Where the participants will be blinded. Yeah. Okay, this information, in protocol document, it will display. So the study design is open label study. So everyone is aware of that particular drug. Single blinded. We are not revealing this particular drug information to the participants. Single blinded trial. So except participants, remaining everything, site management team, and investigators team, data management team, all teams aware of the drug except participants. Single blinded means only participants will be blinded. They don't know which drug they are taking. Okay, so participant will be blindfolded. What is the purpose of the drug given to that particular participants? Huh? They don't know anything except participants remaining everything aware of that particular drug. Site management team and investigator, whoever is managing that particular site, everyone. Next, coming to the double blinded. So double blinded is nothing but the study where the participants will be blindfolded. Participants, patient, that particular person, whoever is taking that particular drug. And investigators team also will be blindfolded. Participants and investigators will blinded. So they both participants and who is going to inject the drug to the participants, investigator. Huh? They both doesn't know anything about that particular drug information. Whoever is managing the site, they are going to just give some package to the investigators. And there will be some labels will be there in that particular packages. Labels will be provided. And based on the labels, they are going to provide so and so person this particular package is So they are going to give that particular package to the participant. They are going to give the medicine to that particular participant. Package only, it will be provided to the investigators. They don't know anything about that particular drug. Participants as well as investigators. So that's the reason we are calling double-blinded trial. So these two members not aware, but remaining everything like site management team and data management team, they are aware of that particular drug, guys. Double-blinded trial. You may ask a question: Why, Balu? Why this kind of techniques? What purpose? Yes, they are going to use these kind of single-blinded, double-blinded to make trial unbiased. Okay, so mainly for trial making unbiased, they are going to use different techniques. Quite common, all this. Now, coming to the, see, each and every protocol, guys, definitely you are going to find either open label or single blinded trial or a double blinded trial or triple blinded. Triple blinded. So yes, triple blinded means principal participants, investigators, as well as site management team. All blindfolded. Okay, here also same. So here site management. So here for site management team, they are going to get some package labeled like a code. Every subject have its own code, guys, subject ID. That particular package they are going to provide. So this is the for this particular patient. This is this package for this particular one. So site management team will get from the higher team like that. So site management team will provide to the investigators. Investigators they are going to provide that particular medicine to the participants, but they don't know anything about that particular drug. It will be kept in very confidential, highly confidential. Nothing will be revealed to the participants and investigators and site management team. Maybe at the time when the data submitted to the regulatory, you know, there in that particular higher level, it will be decoded and it will be submitted to the regulatory. That is how triple blinded it works. If the trial is triple blinded trial, no one will be having a single clue of what is that particular drug that's being given, either to the subjects who is monitoring that particular site, who is providing that particular drug to the participants, and all will be blindfolded. Okay. Next, coming to the single sequence and randomization. Single sequence means throughout trial, guys, only one group of people they are going to participate. Same drug. Throughout complete study, they are going to provide same drug to all the participants in that particular group. Single sequence. Randomization. See, randomization is nothing but randomly divided participant into different groups. Okay. So here there is a picture, guys. With this particular one, I can easily explain about study design. It's a parallel study design and crossover study design. Okay. We have few minutes only. I will explain later manner here. In detail, we'll discuss tomorrow. For example, there is a study drug, guys, A and B. For fever, we are conducting the trial. We are generating a particular drug. So study drug A and study drug B. So study drug A is why we are calling study drug and existing drug. Sometimes we need to prove the efficacy of the drug. So how efficiently working with when compared with existing drug. For example, once regulator may ask you a question. Hey, already this particular drug curing the headache, already drug is available in the market. Why I need to approve your drug? So in that particular case, you need to prove your efficacy, right? How efficiently your work is working fine. For example, existing drug, it's curing headache within 10 minutes or 15 minutes. If your drug is curing within 15 minutes or lesser than 15 minutes, then okay, fine, well and good. They are going to release that particular one. They are going to approve. Otherwise, if it's taking 1 hour time or more, then what is the need of that particular drug? There is no need, right? There is no use. So for that, some trials we are going to use along with study drug. Study drug means which is not available in the market. Existing drug means yes, it is available in the market, approved by the FDA, approved by the regulator. Okay. Parallel study means, for example, there is 100 people, guys. They are participating in the trial. So 100 people, they participate in the screening. There is a flow. Listen carefully. Screening first, then once screening is completed through inclusion criteria, exclusion criteria, and everything, then we are going to give the consent form, like formalities. Simply I can say that all the formalities. Hey, if you happen anything in this particular trial, this is your compensation, or else these are the side effects you can expect. You need to visit this particular site, this particular date to this particular date, before 3 days or after 3 days. These are the limitations. Everything, all the formalities we are going to provide in that particular instant consent form. Once participant is signed informed consent, then they are going to entering into the randomization, guys, based on the study. If it is a randomization trial, so randomization is going to happen. Randomization is the one biostatistician will do the randomization. So out of 100 people, 80 people clear the screening and signed informed consent, because the remaining people not met with all the inclusion criteria and exclusion criteria. Let's assume like that. 80 people only final they are participating in the trial. These 80 people, so they have divided into 3 different groups. Okay, biostatistician is the one going to, I will explain, guys, everything one more time in detail. So biostatistician is the one randomly these 80 people divided into 3 different groups. So R means treatment group, arm 1, arm 2, arm 3. So this here three actual 2 treatment groups are there. One is intervention group, another one is control group. So intervention, again, it will divide into multiple groups, guys, but whereas coming to the control group, they are going to provide only placebo drug only, nothing else. Fake pill, controlled drug, controlled treatment group. So whereas coming to the intervention group, intervention means all these. So who is participating in the trial, live trial? So that is called live treatment with study drug or else existing drug. That is called intervention. Who is participating in the fake drug? That is called control group. About control group and intervention group, everything I will explain once we are discussing about randomization process. There you can see. So this is control group. They are taking throughout study only placebo only. Day 1, week 2, week 3. And once done with your treatment, then follow up. And again, this is another group. So this is intervention group. These 2 groups. Drug A, and another group, drug B. So 20 people. So for control group, 30 people and 30 people. So complete 30 people throughout the study, they are going to take only one drug, which is called parallel study design, guys. Complete throughout study, if you take only one drug, if you are the participant, that is called parallel study design. But we have come to the crossover study. So switching will happen. See here, placebo, washout period. and drug A washout period, drug B. And again, A washout period means, for example, you are taking one drug, that particular drug effect should be nullified. It should be wash out from your body. Then only they are going to take another drug. Otherwise, it may cause any side effects. Okay. Washout period is nothing but existing drug effect should be nullified, should be washed out from the body. For example, there is anesthesia they have provided. So anesthesia, it will take hours of the time, sometimes it will take days. Okay, based on the anesthesia. If it is a 5 hours or 10 hours or else 24 hours, it will happen. That particular effect is available in the body. So after 24 hours only, they are going to take another drug. So washout period is will be there in the crossover only, not with parallel study. But because they are going to take same drug only, na? Why here washout period? So crossover is nothing but when switching will happen. So each and every group will switch to the treatment that is called crossover. So this is just a preview. In detail, we'll discuss tomorrow. I will explain one more time about parallel and crossover study. That's all about today. I have another session. Let me wind up the session for now. We'll continue tomorrow. I will repeat one more time what we discussed today. Thank you, guys. Okay, we are discussing about teams. So here we are still at protocol team, right? Protocol team. About protocol document we are discussing. Still we are at second phase only. Yeah, protocol document, what and all definitions we should learn. That's what we are discussing. So in protocol, there will be trial site information, and if it is a multi-center or multi-nation or single location, what will be the objective for your study, purpose of this trial. So what is the main goal of your study? If it's a phase 1, what is the main goal? Phase 2, what is the main goal? Phase 3 and all. What will be the secondary objectives? Background. So I said right, background. If any of our other organizations or other industries or other companies, if they've done trials about this particular one, what about the results? What formula they are using? What is the difference between existing formula and your formula? What type of drug it is? If it is a medicine or vaccine or a syrup or any medical therapy or any medical device. So complete background we need to put in background section. And study design, open label or single blinded or double blinded or triple blinded. Open label means open to everyone, whoever all the persons are participating in that particular trial, either participants, subjects or patients, anyway you can call, who are participating in the trial. Participants who are inducting, who are providing drug to the participants, investigators or doctors or clinicians, anyway you can call, all are same. Okay, investigators you can say, doctors, or else you can say RS clinician. Hmm? Anyway you can call. Yes, all of them, they are aware of that particular drug, and who are maintaining that particular site management team. So they are aware of this particular drug and who are analyzing that particular data, like us. Hmm? Analysis team, they are also aware of that particular drug. Open label, open to everyone. Whereas coming to the single blinded, where the participants will be blindfolded. They don't know anything about that particular drug. Remains everything, they are aware of that particular drug. So if it is a single blinded trial, so they don't reveal about drug information about the participants. Double blinded, along with the participant, doctors, investigators also will be blindfolded. So remaining people aware of that particular drug, they will be provided labeled, packaged, um, packaged with the labels they are going to provide to the investigators, your site management team. They are going to give that particular drug to the participants. And here triple blinded participants and investigators and site management team, all teams, they are not aware of that particular drug, all the people. Okay, even who are doing that particular analysis also. Hmm? You two also don't know about that particular drug. Triple blinded trial. Okay, so these kind of study designs will be there, and one more type of study design will be there. So study design in the sense, how many groups will be there? How many groups? If it is a single group, throughout the study or else multiple groups. Single sequence, only one group is only one group. They continuously use only one treatment. Same drug continuously. Or else if it is a randomization, parallel and crossover. Okay. To explain randomization, I want to tell you one point, guys. Here, the flow of your clinical trials once entered to the live trial. So this is very important. Just remember, first screen by using inclusion criteria, exclusion criteria. Okay. First screening. In screening is nothing but they will check your inclusion criteria, exclusion criteria. Every trial have inclusion criteria, exclusion criteria. If it is a cancer, guys, so they are going to recruit cancer patients. Patients should have cancer. That is inclusion criteria. Exclusion criteria. They don't, they don't drink. They don't use cigarettes. Like that. We have some exclusion. Just example, I'm telling you. Exclusion criteria means what and all things they are not able to. They are not do. So inclusion criteria means they must have that particular things, inclusion and exclusion. So like that, all inclusion criteria, exclusion criteria, they are going to check in screening whoever are matching. For example, if it is a 200 participants, all 200 participants may or may not. Yes, whereas coming to the participants, who is going to recruit? I will explain that one also. Site management team, they will conduct some seminars. They will explain about that particular drug to the peoples through public people. So some people they are going to join their own interest. Some people they are going to recruited by sponsor. Okay, so completely depends. Whoever voluntarily, if you want to join, you can join, but you need to meet inclusion criteria, exclusion criteria. Okay, if you meet inclusion criteria and exclusion criteria, then you need to, fill the in some informed consent form, ICF form. That is nothing but formalities, guys. Simply I can say that. Formalities, all the formalities, these and all procedures we are going to do with you. So these and all visits you need to perform. This visit or this visit, this many days gap will be there. And if you have any side effects or if you have any adverse events happen in this particular trial during this particular trial period, so this is your compensation. Like that, each and every information, right? Terms and conditions, I can say that all terms and conditions format. So I will show you informed consent format. So all the documents we'll discuss and I will show you later. So informed consent. First, once inclusion criteria, exclusion criteria completed, whoever all met with inclusion criteria, then they will enter to the ICF form. Same day, guys. It will happen same day, within a moment. They will confirm informed consent. They will agree with all the terms and conditions. They will sign the informed consent. Then they will enter to the randomization. Randomization. In randomization, either randomization is nothing but I said right. If it is a parallel study or crossover study, if it is a parallel study, throughout study, so randomly they are going to divide into multiple groups, guys, different groups. I mean, it could be one group or it could be two groups or it could be three groups. Depends upon trial requirement. What is the purpose of that particular trial? So randomization is but randomly. There will be no criteria, guys. There will be no criteria followed in order to divide the particular study population. Just randomly, blindly, randomly, people will be divided into different treatment groups, where each group will be receiving a different set of treatments. For example, if it is a single sequence, only one group, and they will go with only one treatment. For example, if it is a two different drugs, guys, two different drugs, sometimes we are going to use existing drug and study drugs. Sometimes we are going to use two study drugs. Such cases, for example, if it is a one group, same study drug in the mains, for example, A1, one group, and A2, one group. Sorry, A2, A1, one medicine, A2, one medicine. So then, only one group, first they will go with A1, followed by continuously A2. That's it. If you have two study drugs, first one drug, next one drug, only one group, that's why. There is no switching, there is no cross. But let us come to the randomization. Here, randomly divided into multiple groups. For example, arm 1, arm 2, arm 3. Three groups, guys. Okay. So each group, first, so there is a treatment period. Study day 1, or else under treatment I will say. Day 1, or else study day 1. What is called study day 1? I will explain. Which day we are going to expose to the first drug, that is called study day. So from there onwards, next visit will be maybe sometime they are going to use 1 week. Some studies they are going to use 2 weeks. For example, here 1 week, 2 week. Next visit is going to be 3rd week. Next visit going to be 4th week. All these are treatments only. And then last week, for example, after 2 weeks of the treatment, week 6, for example, there will be follow-up. Okay, randomly they divided into three groups, guys. This group, I can say that control group. Control group means throughout period, guys, they are going to use placebo. What is called placebo? I will explain later, but just introduction for purpose. Placebo is nothing but a fake drug, fake pill. It's like a sugar pill, but they don't know. So these particular persons, they are going to provide drug completely, entire study, placebo only. Why Balu? Why placebo? Why fake pill? Because yeah, guys, one moment. Okay, so why they are going to use fake pill? Because some of the people mentally they are going to prepare. Okay, I have taken drug, so it is going to be cured automatically. Huh? So mentality like that, even though if it is a fake pill, they are going to fix mental. They are going to prepare mentally. So I am going with treatment. They have provided medicine to me, so it is going to be cure. Like that, they will feel, and it will work sometimes. So how you are feeling? What is your mentality? Based on that, also drugs it will work, guys. Sometimes, last time in COVID, so most of the people they are not mentally stable, even though they are using medicine. So only they are feeling okay. So I'm infected with COVID. Maybe I'm going to die. So they, based on their mental condition, even though the vaccines are also provided, most of the people died because they mentally fixed. I'm going to die like that. So some of the people they will be confident. Yes, it's working fine. There is no need to hurry. So I'm using medicines like that. They will prepare mindset. If you prepare your mindset based on that, your drugs also going to work, guys. Your antibodies will fight with that particular viruses and all. Otherwise your antibodies doesn't fight, won't work. So that is also very important. If you want to prove your efficacy of the drug, huh? So drug is curing or else your antibodies are working. Huh? So to check that particular efficacy, to check that particular diversity, they are going to use set of the people only placebo, fake field they are going to use to eliminate. Huh? For example, you are over Nani, there are some people are overreacted. People are there. Those are not useful for that particular treatment, guys. Those are not useful for your study because they are overreacted. With overreacted people, you are not able to prove your efficacy of the drug. Drugs should work for all type of people, okay? Not only for mentally stable people, not only for confident people, not like that. It should work for all general people as well. Then only it is effective drug. So then set up people they are going to use control group. You can say placebo they are going to use throughout study, week 1, week 2, week 3, and everywhere. After that, no, we are going to deal with follow up. Follow up means, so after taking the treatment for 1 or 2 weeks, they will wait and they will check is there any adverse event, is there any side effects. And this is the last visit, guys. Any participant follow up is the last visit. First, they are go with screening and form consent form sign. And if it is a randomization trial, they are going to randomly divide into multiple groups. And statistician is the one randomly divided, and they will provide a code. So hey, this is your group. You need to go this particular group. You need to go this particular group. They are going to decide it. And another group is going to be, intervention group. Here, this is the one, guys. Intervention group. People are going to use study drug. These people are exposed to study drug. These so intervention group can be two groups or three groups or multiple groups based on the study. Okay, based on the treatment, based on the requirement. So two groups they can divide it intervention group. If it is a two study drugs, if it is a three study drugs, like three groups like that. Hmm? Depends completely. So treatment, and then follow up. So if it is a parallel and if it is a crossover, what is the difference? Here it is the presentation. Now you can easily understand this particular study design. So here there is a study drug A and existing drug B. If it is a parallel study, three groups are there. First group is a controlled group. Controlled group only fake pill, placebo, and then they will go with follow up. Next group, throughout study, complete study from the week day 1 to complete study, they will go. They are going to expose with only one study drug. And next next group, two intervention groups divided into two groups, guys. Intervention people. So study drug and existing drug. Follow up. This is called treatment period. This one treatment because this particular these are called elements, guys. Elements, placebo one element and drug A one element, drug B element. Elements one table we are going to create. What and all planned elements? How many elements subject participated? So that is called planned element, protocol planned element, trial element dataset, TE dataset, TE domain we'll discuss. Subject participant element, SC domain, subject elements we'll discuss one more. And how many visits? Trial visits. Trial visit dataset, how many visits you planned actually, but each participant, how many visits, how many times visited hospital, visited that particular clinical trial site. That is called subject visits, SV. TV planned, SV actually happen. Like that, guys. There is a comparison always. Through comparison, they are going to take a decision. They are going to take an analysis. Okay, this is parallel study design. So screening, informed consent, randomization. So either control group or else intervention group, whatever group it is, completely only one treatment. If it is a crossover, if it is a crossover study, switching will happen. Even for all groups, even for the control group also, placebo, then washout period, then drug A. What is this washout period? Washout period is nothing but, for example, here we are using different drugs, right? So first, what is the first drug you are using, that particular participant? The drug effect should be nullified, should be completely absorbed by the body, should be completely removed from the body. I mean, there is no residues, there is no particles should be left. For example, let's say there is anesthesia. Based on where they are providing anesthesia, based on that, it will take 4 hours sometimes, it will take 6 hours sometimes, it will take 24 hours time sometimes. For example, if it is a 24 hours time, the 24 hours time you can call washout period. To go with next drug, previous drug effect should be nullified, should be completely washed out from the body. That is called washout period. Washout period will be happen only in the crossover study. First one drug, washout period. Next another drug, washout period. Next another drug, then follow. Hmm? Okay. No treatment will be given to allow the effects. Completely previous drug effects should be dissipated, guys. In parallel study, not a problem, right? But because we are using only one drug. No need about washout period. But whereas coming to the crossover only, we need washout. So these kind of study designs will be there, guys. If you open any protocol, they're definitely either single blinded or triple blinded. I will show you protocol. Primary objective, secondary objective, study title, study number, study design. You can see here. Open label and single sequence. Open label single sequence means throughout study only one group. Open label, everyone is aware of that particular drug. This particular study will open any other. I think I saved somewhere other projects. Latest project. This is the one. It is SAP Document. And it is CRF. SDTM spec. Okay, I will show you tomorrow. Another protocol. So if you open any protocol, guys, there definitely what kind of study design it will be there. Okay, so this flow, don't forget guys. So screening, ICF, randomization, treatment and follow-up. So completely almost all domains we are going to discuss based on this particular concept only. Okay, so this is about study design. Next, one more important point. Inclusion criteria, exclusion criteria, guys. This is also important to know. What is that inclusive criteria and what is that exclusive criteria? Which is nothing but if a participant satisfies all criteria that is mentioned under the section inclusion, they can come into the trial. At the same time, whatever the criteria has been mentioned under the section exclusion criteria should not be satisfied with even one exclusive criteria. Should not be satisfied in the sense they don't drink, but they are drinking. So that is dissatisfied, right? So they are going to exclude from the trial. Female should be, like normal. It should not be pregnant ladies. So if that particular participant is pregnant, so they are not eligible into the trial. Okay. So where all the inclusion criteria should satisfy, even one exclusion criteria also should not be satisfied. So then that particular person, they are going to enter into that trial. They are going to consider into the trial. That is about inclusion criteria, exclusion criteria. Okay, person who says to simply say that yes to all inclusion criteria, a person who says no to all exclusion criteria, they are going to will be considered into trial. No means yes. Are you drinking? You said no, right? Are you pregnant? You said no. Hmm? Do you have any medical illness in first few years? You said no. Then only they are going to recruit. If you have any medical illness, if you have any medical history, they are not going to recruit. Your age should be, so coming to the inclusion criteria, a few examples. Your age should be 18 to 50 years. Yes. You should be gender male. Yes. Like that. All inclusion criteria, you need to say yes. All exclusion criteria, you need to say no. Then only you are going to consider in that particular trial. Inclusion criteria, exclusion criteria. Okay. Come to the next one. Study enrollment procedures. One second. study enrollment process is nothing but, see, particular participant, particular clinical trial to happen. Who is going to, yes guys, the enrollment process, simply I can say that. Who is going to enroll that particular study? They should be willing to participate into the trial. There is no force, guys. There is no force actually. Okay, participant have come forward voluntarily to participate, or else, so they should be willing to participate. They should be willing to accept the new drug, taking new drug. Then definitely they are going to enter into the study. Because phase 1 trial, especially phase 1 trial, we discussed right in previous classes. Phase 1 trial is very, very risk when compared with phase 2 and phase 3 trials. So preclinical studies, definitely it will be in animals before the phase 1. But even though there is a safety proven in the preclinical study, whereas coming to the first phase, human clinical trials, phase 1, we cannot compare, right, humans and animals, as risk is very high, as very risk is involved in that particular phase 1. So some of the participants may not interested to join that particular study. If the person or if the participant is willing to take the drug, although if it is aware of risk, then only they are going to enroll that particular clinical trial study, guys. Enrollment is nothing but taking the participant into the trial. Okay, if a person is who is willing to participate, it's not that person will be definitely considered into the trial. Okay, so who is willing to participate into the trial, they will not definitely going to be considered into the trial. So what is the first thing we need to confirm? All inclusion criteria and all exclusion criteria, they should match. If the checklist, all the checklists, inclusion criteria, exclusion criteria, if it is matching, then they are going to bring them into the clinical trial study. So once participant is considered into the trial, what and all procedures will be there? What type of what type of set of procedures we are going to provide that particular participant? Always there is a plan, right? All plan of actions we need to explain that particular participant by using which one? Yes, anyone if you tell me all the procedures, what and all things we are going to do with you, what and all side effects are available in this particular study, where we are going to explain that participant while doing informed consent form, right? I said right, formalities.
Yeah, yes, yeah, sir.
Yes, all the formalities. They are going to agree with terms and conditions. They definitely signed informed consent form. Then only they are going to participate in the trial. Okay, suppose if the trial study is randomization, so then usually at the time when the participants submit the instant consent, most of the time immediately they have to undergo the randomization process. But most of the clinical trials, once informed consent form is signed, they are going to enter to the randomization only, guys. If most of the trials randomization, if it is a not randomization trial, if it is a single sequence, then there is no concept of randomization. Within a moment, they are going to enter to the treatment only. Okay, so completely depends on the sponsor, guys, sponsor, and depends on the treatment, depends on the requirement. What is the techniques they are using based on that only.
Balu, sometimes that informed consent date will be like screening date, right? Like based on the sponsor, like how they
Exactly.
decide. Yeah.
That's why I said this. In screening and informed consent, it will be happening same day within a moment. Yep.
Yeah.
About dates also we'll discuss. Informed consent date and screening date and treatment start date, treatment end date. We have different dates. So once we enter to the project training, all the dates we'll discuss, and we may get a bit confused with a lot of dates, but I will try to compare while discussing new dates, existing dates, and what's the difference between new dates and old dates. Everything I will compare and I will try to explain in a simple manner. Because project training, as I said many times, when compared with technical training, bit difficult, guys. Not that much, bit difficult. If you do the practice, then it will be going to be easy. Okay. So whatever the group of the participants, if it is a single sequence or if it is a randomization, it's a complete set of procedures that is called study procedures, enrollment procedures. Okay. And study elements. Elements, you know that, right? I said elements can be anything, either if it is a screening, or else treatment, or else follow-up. All are. If it's a treatment, week 1, week 2, week 3, week 4, week 5, all are considered elements only. Treatment can be in multiple weeks, right? Sometimes months, sometimes it will be take years also. Completely depends on trial only. All under comes under elements. Okay, we'll talk about elements. We have separate domain there. I will explain. So here, screening is nothing but confirm the eligibility by checking inclusion criteria, exclusion criteria. Already we discussed all these. Okay, and elements is nothing but yes, how many elements we planned. So first screening, next treatment, in this treatment, you know, week 1, week 2, week 3, week 4, week 5. For example, week 5 there is an unexpected element is happen, adverse event is happen. Then that is also one of the element we can call 5.1, week 5.1. So element names we are going to explain here. For example, week 1 there is a drug A, week 2 there is a drug B, week 3 placebo, or else any other. What kind of treatment we are going to provide, we'll discuss, guys. Just these are called elements I want to tell you. Starting with the screening, ending with follow-up, what and all protocol defined, clinical trial plan, that is called element. Either screening one of the element, treatment one of the element, week 1, what and all drugs you are going to provide, that is one of the element. All these are elements. Elements we'll discuss. So elements and baseline assessment. This is also every programmer should know. Baseline assessment. Baseline is the term we should always aware of as a programmer, because we will be using this baseline concept while doing the programming part. Okay, so baseline is nothing but before taking the study drug. So any procedures that happen for a participant before taking a study drug, we can call such kind of procedures, such kind of visits done by the participant, we can call as baseline assessment before taking the drug. Sometimes, guys, before the taking the drug, they are going to, take some other drug. Study drug is different, and protocol define drug. Okay, so in the protocol we are going to define these many drugs I am going to use in this particular trial, like that. Before taking the study drug, if you take any procedure, anything, that is called baseline assessment. Okay, so baseline assessment also we'll discuss it along with examples later. But remember, baseline assessment is nothing but before taking the study drug, if any procedures happen, such kind of widgets are happen, we can call, that is called baseline. Okay, intervention, already I said, intervention is nothing but participants intervention control group, I said, right? I will provide notes, guys, no worries. Intervention is nothing but the time participant starts taking study drug. Who is going to take the study drug, that kind of people, they are going to enter into the intervention group. Who is taking fake drug, that kind of group we can call control group. Intervention group and a control group. And study visits, elements here we discussed, right? Visits also. For example, visit 1. First visit, you need to finish the screening. Next visit, you need to enter into the treatment. Treatment, this is visit 2. Next visit, they are going to provide after how many days you need to visit the site. Visit 3. Okay, elements information, visits information, both are very important. Visit 4. Total, how many visits will be there? Visit 5. Visit 6. This is 6.1, unexpected visit. Unexpected visit. And last visit, visit 7. Every clinical trial study.
Yeah, but like we will tell like unscheduled visits.
Yeah, unscheduled
like,
or unplanned, unexpected. Yes, whatever, all are same.
Yeah.
This is we are not able to plan, guys, because we don't know, right? The particular participant going with any side effects or not. Unexpected, unplanned visits we never plan, guys. So these are planned one, week 1, week 2, week 3, week 4, week 5. But this is unplanned, unexpected. If it is happen, so we are going to store in the one data set called, SV dataset. SV, subject visits. So what is actual visits? What is actually your planned visits? Is there any unexpected, unplanned visits also happen? We need to store somewhere. That is called unplanned visits. We'll discuss. So visits also very important to know. This will be there in your protocol document. How many visits are there? Each visit, what is your treatment? They will define. I will show you. I need to correct all these pictures because this is my second CDISC batch. Like this, you can see schedule of events. It is day 1, day 2, day 3, day 4, day 5, day 6, day 8. So if it is a day 1, what and all things they are going to happen? See here, informed consent, inclusion criteria, exclusion criteria. See, screening first, right? First screening, then it will enter to the treatment. Screening, informed consent, signed X means you need to do all these things. Inclusion criteria, exclusion criteria, protocol restrictions, demographics data. Demographics data means what is your gender, age, height, weight, BMI, everything, all information about participant subject. Here BMI and urine, alcohol, drug screening, all these based on the study. Safety lab evaluations and vital signs data, body temperature, ECG report, everything they are going to do while screening. Screening, you can see 28 to -2. So guys, screening always before starting the treatment, it should be either before 2 days or before 10 days or before 28 days, you need to finish the screening. Then only you will enter to the treatment. Okay, so like that, these are all examples. Okay, so day 6 means this is one of the visit, this is one of the visit, this is one of the visit. Different visits will be there. Visits it should display visit 1, visit 2 like that. In this protocol, it is not showing. Definitely, it will be there somewhere. I will check later. There is no time to discuss now. Tomorrow, I will show you for sure. Visits information. Okay, so widgets are also very important to know how many widgets are there and each widget, what is the treatment we need to mention in that particular protocol document. Okay, and what is the duration for each visit? Every visit has start date and end date, guys. We need to discuss everything about widgets information. We'll continue tomorrow, guys. I will note down here. Visit info. We'll continue from widgets tomorrow. Okay, all these are terminology we should know, guys. We should know before stepping into the real-time programming classes. Okay, all protocol definitions. One by one, we are learning. That's all about today. We'll continue tomorrow, guys. Okay. Still, we are at protocol team only. Now, protocol document we are discussing, right? What and all things we should know. Where we are. Okay, visits information to discuss. Yes, this is very important, guys, about visit information. You know, I think I have updated one picture. So when the participant does the visit, or does visit the site or a clinic, so that is called visit. So without visiting the clinic or without visiting the trial site, this procedure cannot happen, right? Like first doses or second doses or third doses or follow up, whatever. Whenever you visit the site, that is called one visit, guys. How many visits are there? Each visit, what and all procedures will be done? Everything interlinked, guys, correlated. You can see here, for example, I have given some example. How many visits are there? What are the procedures? Look at matrix. For example, visit 1. So after screening, after the screening, so in the screening, informed consent and demographics. Demographics means that, right? What we'll discuss about demographics in next coming classes. What is the age? What is the gender? Which ethnicity, location? Like all personal information about subject. Okay, height, weight, BMI, and everything. Here we are going to take demographics information. And is there any medical history in the past? So all this information. General physical exam. So they are going to conduct some on the participant while screening itself. Electrocardiogram form we need to sign, while test science form. So these and all, informed consent and inclusion criteria, exclusion criteria, everything in screening. Then visit 1, treatment, adverse event, and this one and this one. Visit 2, visit 3, visit 4, and last visit. The last visit definitely it will be follow up only. So some of the participants, so they are going to quit the trial, or else sponsor is going to quit them as they are not responding properly, as they are not attending on the time. So like that, there are many reasons, guys. We'll discuss all types of possibilities later. But every visit, these and all procedures will be done. They are going to provide complete schedule during the visit. Each patient, each participant, what will be the plan when you are in the study period? What in all clinical procedures happen during this particular visit to this particular visit? Okay. So, and again, so during the screening itself, they are going to explain, guys, each and every information to the participant. It's like a compulsory, mandatory. Okay. So maybe buffer will be given. Buffer means, for example, um, if the participant so on so date, you need to attend either plus or minus 3 days, plus or minus 5 days, like that buffer will be given. For example, there is a visit they have scheduled. They are scheduled either before 3 days or after 3 days. You should visit the trial. You should visit that site. Otherwise, if you attend later, it doesn't valid anymore. Okay. So like that. So visits plan, they are going to explain to the each and every participant during the screening only. For example, let's say I will go with example here how we are going to deal with the visits. Screening. After screening, treatment will start. For example, visit 1, visit 2, five visits, and follow up on last visit, for example. 1, 2, 3, 4, 5, and 6. Okay. Screening, usually, guys, they are going to, provide like this, minus 28 days to minus 1 day. Like that. So what is this minus 28 days to minus 1 day means, before treatment start. So here onwards, it will enter to this treatment, right? Treatment. I said flow already. Where it is? Yeah. Screening, informed consent form, both will be same day. So then randomization. So after that, we are entering into the treatment period. So once entered to the treatment period, you should be finished screening. Minus 28 to minus 1 day means definitely before starting treatment, before 1 day to prior to 28 days. For example, today, what is the date here today? December 23, right? December 23. If you want to participate today into the trial, first doses of the treatment, you need to finish screening, informed consent form and inclusion criteria, exclusion criteria, everything you need to finish. Screening is nothing but checking the eligibility, eligibility for the participant. You need to finish before 1 day, means minus 1 day means, tell me which day it is. Minus 1 day. Before 1 day. December 22. Minus 1 day means. 2 minus 28 days means. Minus 1 day before 1 day. Minus 28 before 28 days, guys. So means, December 23 to, I think, November 25, 26 around, right? Are you able to understand what I'm trying to say? If you want to participate this date, if you want to participate December 23, study day, if you want to participate to the treatment, first dose of the drug, you need to finish the screening either before 1 day to before 28 days. That means around November 25th to December 22, guys. You need to finish your screening. You may get out, Balu, in case if I am not able to finish this particular screening, if you finish, for example, November 20th, before November 20th, within this particular period, not. For example, I have finished my screening November 20th, what will happen? So if it is finished earlier of this particular period, that is not valid anymore. You need to do the screening again if you want to participate in the trial. So this is valid. Minus 28 days to minus 1 if you do the screening, if you finish the screening, that particular one only valid. So out of this particular range, if you done before the 28 days or after, after the treatment, no more valid. You need to do the screening again and again, that's it. So when it is valid screening, as they are going to provide, guys, they are going to give the information, hey, you need to finish the screening this particular period to this particular period, then only you are eligible to the participate in the trial. If you do before, you need to do the screening again, this particular range of the days. Are you able to understand, guys, what I'm trying to say here? Minus 88, minus 28, minus 1, sometimes minus 7, minus 3. Minus 7, minus 3 means? Minus 7 through minus 7 to minus 3 means? Tell me. This is the range you need to finish the screening, if they said.
3 days after the treatment.
Yeah, 3 days before or?
Before 7 days, like.
Before 7 days. 3 to 7 days before. You need to finish the screening. You want to participate in the trial. Okay, for example. So now coming to the guys, they never assign the dates in that particular, protocol document. Each and every visit, study day 1, for example. After 1 week, study day 2, visit 2, 7, 8 days, or else 15 days, or else, 22 days, 29 days. For example, if it is a follow-up after 1 month, next month 30, for example, or 50 days, 60 days, I'm going to use 30. So I said, right, buffer may be given. Buffer may be given means, for example, here, mostly from the second visit onwards, plus or minus, like this. Plus or minus 3 days, plus or minus 3 days, or plus or minus 5 days. What is the meaning for that? Plus or minus 3 days or 4 days or 5 days means before, plus means, guys, after 3 days. So they said once visit this particular date. I said again, remember, guys, they never assign the date. So because all the participants may or may not attend a particular date when it is going to start the trial. So always Not possible to schedule specific plan, guys. Days they can schedule 1 day. So 1 day to next 1 week, 8 days. So 15th day. So for example, if it is a trial started, you know, for example, January 10th, January 10th, whatever year it is. Hmm? So you should exit the next visit. For example, first visit January 10, visit 1. This is very important concept, guys. That's why I'm spending time here. So first visit January 10, then next visit 7 days, right? So 17, January 17. So based on that, based on the study days, we are going to calculate the dates. Huh? So here there is a buffer is given, right? So plus or minus 3 days they can attend. Either after 3 days like January 20th or else before 3 days. Huh? January 14, 15, 16 also he can attend this particular visit. If you are the participant, not possible to attend that particular trial site every visit. For example, there is some personal issue. Or else after the first visit there is an adverse event happen, you have some side effects. Until and unless if you are good, you are not able to participate the visit too. So maybe then the date will change. There is unexpected journey you need to travel, so may or may not attend that particular day, right? So that's why this buffer will be given, guys, for each and every visit for all the participants. Okay, so like this, so 10 and 17. So then next visit 3. So only number they are going to provide, study day 10 or study day 15 or study day 22, like that. Based on that, where you are attending from there onwards, they are going to connect the dates. 17 to 7, for example, January 24, like that. Next February 1 or something. So every visit like this will happen. If any visit between unplanned visit, if it's happened, for example, visit 1 to visit 2, there is an unplanned. Unplanned visit, for example, unplanned visit means, as I said, there is a side effect. So actually, January 10th, your first visit done. Next visit is going to be, you need to visit that site January 17th. So in between, for example, there is unplanned visit. Because adverse event is happen, there is some side effect. So if you have side effect, definitely they said, hey, in case if you have these kind of side effects, try visit the site and let us know. So there is unplanned. So there is adverse event happen and you visit the trial. So in between January 10 and January 17, for example, January 12th, you visit the site. That is called visit 1.1. First visit to second visit, there is a happen, right? 1.1. For example, unplanned visit again, January 14th also happen. He visit the trial with another side effect. So 1.2. Okay, actual visit, unplanned visit, like this we save in our data, guys. 1.1 means first visit, first unplanned visit. So first visit, second unplanned visit, like that we are going to store. They will provide the data with you. No need to identify anything your own. Next visit 2, visit 3, visit 4, like this will happen. For example, again visit 4. Here also, type in the baby. Okay, so here, for example, this is February. 1 hour second. So after third visit, there is unplanned visit. Then, for example, this is January 26, for example. Then this is third visit, right? They will provide 3.1 like that with this information. In general, visit 1, visit 2, visit 3. If you have any unplanned visit, these kind of versions they are going to provide. Numbering will be given like this only. But because unplanned visit, we are not able to plan, right? May or may not. May or may not. So unplanned visit will never plan, guys. Only actual visits only we are going to plan. They are going to plan the protocol document. Okay, when you look at the protocol document, you can easily understand how many visits are there they have planned, and everything. You are not able to find any unplanned information. So, but while screening itself, they are going to inform each and every participant. Okay, if it is any adverse event happen, just guys, one moment, 1 minute, I will be back. Okay. Okay. So unplanned visits always unplanned only, guys. How to deal with unplanned visits? There should be always a plan. You know, 1 second. When it comes to the practical work, so planned visits and each and every planned visit, all procedures will be happen. All this function, all these procedures will be explained to the participant while signed informed consent. Like this many number of visits being planned and each and every visit in case if you have any abnormalities, is there any side effects, you need to visit you need to visit the trial, you need to visit the site like that. Each and every information there, each and every procedures and each and every activities they are going to explain in a very clear manner to each and every participant. So it will call it as study visits. So following here, usually for every study, if you check any study, guys, there is no fixed date given to the participant for the studies. There is no specific date, as I said. Okay, so like this only, study day 1 or study day 8 or study day 10 or 15 or 20. So some number of days only they are going to provide. They will never perform, they will never say this date you need to perform, this date you need to visit the trial. Never happen. Okay, for an example, so you visited the clinic one fine day, they have asked to you, the team over there in the hospital or clinic, they have created an awareness program. They are doing an awareness program about clinical trial that's happening at their hospital. They were asking you for your willing, like, are you willing to participate in that clinical trial? And they are explaining all these risk factors and everything. What is the risk involved in that particular trial and how it will be compensated and everything. And all the clinical trial methodology, for what purpose they are doing this particular trial, why they have initiated this particular trial and research and everything. All that they are trying to create some awareness. Suppose say you have been convinced. Imagine yourself, from the awareness, whatever they are trying to create, you have been convinced and you are willing to participate in the trial as you satisfied all the criteria, all the procedures. Okay, for example, you are considered into the trial. So once you consider into the trial by them, as you satisfied all the criteria, now, so these dates cannot be fixed. So on these particular dates you have to do, it won't be like that, guys, in the real world, never. They will be telling you, so they are going to explain you about procedures and these many visits happen, these are the schedules for you, like that they are going to explain each and every manner. Which particular day you need to visit. Dates cannot given. This is just remember, because you never know like which particular date you will be starting the trial. Okay, sometimes trial will start earlier. Sometimes it will take time to start live trial, because after approving the protocol document only sponsor can start live trial, right? So how can they provide specific date? It is years of the time it will take in general. 1 to 2 years it will happen. Okay, so the next 10 day or next 15 days, next 10 days, next 20 days, like that only they are going to provide. Next, uh, one moment. Okay, so all this information they are going to provide in protocol document. And why I am stressing about this particular study day concept is, study day concept will be know, we should know as a programmer, guys. In our programming part, this is very, very, very important. Study this concept and visits concept. That's why I'm stressing here the visits and all the elements and every information. Okay, we are going to use study this concept in our programming part. Hence, it is important for a programmer. We need to understand the concept. Okay, we should be able to understand which visit they have planned. So, guys, actually, always planned information. See, so for example, they have planned 1, 8, 15, 22, study day 29, and study day 60, guys. They have planned. So they have mentioned in the protocol document all this information. Planned visits. And actual visits. As I said, they have planned, but all the participants may or may not visit the site as per their plan. The buffer will be given either 1 day before or 2 days before. So, actual visits. As a programmer, we need to derive by using informed consent date and study treatment date. There will be some date columns, guys, based on that actual visits. And planned visits and actual visits, what is the difference? We are going to calculate. What is your plan? Actually, what is happening? Okay. So when we are going to start our project training, there will be, we are going to discuss all this information. Okay. So planned study as per the protocol and actual study, we need to derive by using formulas. What and all formulas we are going to use, we are going to discuss. It should always compare, guys. Okay, based on the comparison, we'll get the results. Okay, in the real world, it's not possible actually. Every participant should do the visit on the time. You are the person participate in the trial. Just let's assume. So you need to travel somewhere. Either you will come day before or after the day. You may visit the site. Let's imagine always, guys, while doing the clinical trial, let's imagine yourself, you are the participant. All these circumstances will happen in the real world. Okay. So always each and every minute details we are going to enter into the protocol document. So all the planning information for that particular study. And one more important point in study days concept, guys, study days concept, we never use zero. There is no zero. Any project, any trial. Zero means that nothing. Either -1, -2, -3, -4 like that. Or else 1, 2, 3, 4. So always minus means before, guys. 1, 2, 3 positive means after, guys. You should remember. This is also very important. For example, I will show you in the protocol document. There you can see screening. Day minus 28 to -2. That means you need to finish the screening before 2 days prior to before 28 days. Okay. And check in day -1. Are you able to see here check in day -1? That means before 1 day you need to check in that particular clinical trial site. So like that, they are going to provide either negative days or positive days only. So follow-ups either day 4 or 5, 6, 7, 8, these are the hours they are going to do the follow-ups after 7 days or 5 days. There will be always positive value or else negative values only. We never use zeros concept in your study days concept. This is very important to know. Am I clear, guys? Study days concept. So each and every visit, there will be a day they are going to schedule. Based on that, we need to visit the plan. So this is about study days concept. I hope you understand this study days concept. Anyways, once we start project training, there also, TV dataset is there. Trial visits, TV. As for the protocol document, what and all visits you have planned? We are going to create a one table, guys. Trial visits, TV dataset, and SV dataset, subject visits. I said right, actual planned one. This is planned one. Here, actual happened. Actual visits. Okay, researcher, they are going to compare. So what is the actual planned document visits and each subject, how they visit the participant, how many adverse event happen, unplanned visits happen. So everything they are going to compare. Always there is a comparison. So like this, every information. So, elements information, what is actually planned elements and what is the real elements. TE dataset. TE, trial elements. Actually, we are going to plan some elements, right? And what is the actually happen? Subject elements, SC dataset. Like that, demographics information, age, sex, height, weight, and everything. DM dataset. I will show you. SDTM implementation guide. How many datasets we are going to learn? If you observe carefully here, you can see here is what I am saying here. Trial design domains, trial arms, trial visits, trial elements, trial summary. All these, what we are going to define in the protocol document. So your protocol document, it's like, like this, right? This is protocol document. Like paragraphs, it will be there. Each and every paragraph information we are going to convert into table, guys. We are going to convert into table. The table we are going to learn in our project training. How to create the table. What and all variables will be there. For example, you can see trial visit, right? Just go to the TV dataset where it is TV. Trial arms, trial elements, trial visits. You can see one record per planned visit. Each visit one record for subject. Study ID. Every study have one unique ID. Domain. Domain means TV. Trial visit. This is your domain name. Visit number. What is the visit? How many visits happen for that particular subject? Okay. So visit number and visit. So visit number and visit. Visit day. I said right, visit day, planned study day. So study day 1, study day 10, study day 15 or 20, like that. ARM CD. So ARM CD means ARM is nothing but treatment group. Which treatment group? This particular person. Screening, or else treatment, or else follow-up, whatever. ARM CD. And ARM. So description in detail. So here we are going to use code. For example, treatment A. Just we are going to use code A only. Here we'll explain description, treatment A, like that in detail. Just here we are going to enter the code A. What is A? Sorry, description here. So TVSTRL, visit start rule. So every visit we have some rules, right? So I said some of the rules we should follow. So what is the start rule and what is the end rule? So these are the columns you should define to create a TV domain, TV dataset. Now come to the SC dataset. So this is planned one. As per the protocol document, this is planned one. See, you can see here, protocol defined description of the clinical encounter, and visit is nothing but any clinical encounter happen as per the protocol document. That is called one visit. So as for the protocol define means, everything they are going to define in the protocol document, right? So first screening, next treatment, first visit treatment A, next visit treatment B, next visit like placebo, next visit something something. So for every event, definitely every clinical encounter, you are going to visit the trial, right? You are going to visit the site. That is called one visit. Okay, protocol defined description of clinical encounter, that is called visit. We'll discuss later about TV dataset and everything. This is, as I said, yes, a TV dataset is planned one. So protocol document, what we are going to define, say protocol define visits. Are you able to see here? Protocol define visits in order to describe assessments and procedures. This is the particular domain we are going to use. Next, coming to the SV. VM domain, SC subject elements, SV domain. See here, subject elements. The subject visits dataset provides viewers the summary of the subject visits. Say subject visits, summary subject visits. So actually happen one. There you can see study ID. Every study there is unique ID, right? You are going to get from sponsor, study ID, and domain. So which domain it is? ESV, right? ESV. And again, so use subject ID, unique subject identifier. Every subject they are going to provide one ID, guys. That is called unique subject ID. Unique subject identifier they are going to provide. We need to fill this particular column also. See, if you want to create a SV data set, see these and all columns you need to include. And values you need to derive. They are going to provide. See, for example, if you want to create a SV data set, SV spec document they are going to provide. I will show you. SV raw data set also they are going to provide. Raw data and spec document. For example, special purpose domain, study data, spec document for SV, subject visits, and study data also. There you can see study data derive SC and SV domain. SV domain, spec document. Guys, are able to see here? Study day 1, baseline, study day 15 plus or minus 3 days, study day 29, visit number. To derive the visit number, okay, as for your spec document, if visit DT equals to screening day minus 21 to minus 1. This particular study may be minus 21 to minus day 1. They have provided that is visit number 1. If it is a visit DT in study day 1, visit number 2, 15 and plus or minus 3, visit number 3. Like that, based on your conditional process. This is your condition. You are going to create a visit number variable. This is visit number. This is visit. See, this is just number. This is just visit 1, visit 2, visit 3, visit 4, like that. We need to enter the complete name. Study day. Which state study day is it? 1 and 15 and 29, 43. There is a study days. You can see here. As I said, you will never get dates. Just study day 1, 15, like that only. Yeah. Study day 71, like that. Visit day. SVSTDTC. Start date of visits. How to calculate? SVENDTC. What is the end date for the particular visit? Start study day, end study day. So like that. See, for example, this particular domain, to derive this particular data set, as I said, they are going to provide raw data also. See here. Visit table. In the visit table, you need to use all these columns to derive these columns. Source file, visit. I will show you where it is. To derive, study data. Visit table. There you can see. Visit DT. This is the one they are showing. Visit DT. If visit DT value is this particular one, then visit number equals to 1. Very simple, you need to derive. So here, this is your visit DT values. Slide 1, slide 15, 29. This data, no need to enter your own. You are going to get from your client. You are going to get. And the spec document also they are going to provide to you. So you are the beginner, no need to take care about spec document, annotation and everything, guys. But you are experienced professional, for example, you are applying for the job with 3 to 5 years experience level, then in that particular case, you need to do this particular spec document as well. Otherwise, minimum 2 years or 3 years experience if you will for the jobs, no need to create the spec document, no need to create the annotation. Just simply, they are going to provide spec document and the raw data and everything. As for your programming knowledge, just we need to create the SAS datasets only, guys. As for the with the help of SDTM implementation guide. Okay, so for example, if you want to derive the visit number, you should know what is called visit number, how to derive. SVSTDTC, what is called SVSTDTC, where it will be there, what kind of format I need to follow. Like that. ISO 8601 format. What is called ISO 8601 format? I will explain separate day. Okay, you should thorough with SDTM implementation guide, guys. Along with your project, you should confident with SDTM implementation guide. Then only you can derive any data set. Until and unless if you don't know what is called subject ID, what is called SVSTDTC and visit number and visit, you are not able to derive. What kind of variable it is, how to derive this particular variable. That is what we are going to learn. If you learn, you can deal with any project. So guys, whatever project it is, project can be anything. But if you want to create a SV dataset, these and all variables only we need to follow. Nothing else. So I said right very first, whenever you want to submit your study data to the regulators, we should follow some terminology. We should follow some rules. Just that one is nothing but your SDTM implementation guide. As for the SDTM implementation guide, we need to submit. As for the SDTM implementation guide, domains. These are the domains. So we have different domains. Domain is nothing but a data set, guys. Different tables. Visits information one table, elements information one table, side effects, adverse events one table. Is there any past medical history? Separate table, demography information separate table. All these tables we have separate data sets, how to create and how to derive them, all the variables. Okay. So actually around 40 to 50 are there, 50 data sets. No need to learn all these. Around 20, 25 enough mostly. Because if the study data is able to create all these data sets, okay, then you can create. Otherwise, for example, in your study, there is no information about death details. So such cases, no need to create a DD dataset. Okay, you have 48 for all types of study. They have provided these 48 and 50 datasets. How many datasets are able to create as for your raw data, as for your trial? For example, if it is a 20 or 25 or 30, yes, those datasets only we are going to create and we are going to send that particular to the regulator. Not mandatory to create all the datasets. But if you have collected the data, definitely you need to provide that particular dataset to the regulatory for approval. Okay, so regularly and mostly I can say that around 20 to 25 domains we are going to create 25 different data sets. Those data sets we are see here. Let's come to the special purpose domain. So these four important: comments, demographics, subject elements, and subject visits. Trial domain here. TA dataset, and except TD, remaining everything are important. All five. So whereas come to the interventions, general observation class. Here around 3 to 4. Here 3 to 4. So every domain, every class we are going to discuss around 4 guys. Actually we have 5 for the 20 or 22, 23 domains we are going to discuss. Those are enough. You can deal with any project. Okay. So this is just introduction about what we discussed today. Just study test concept only we discussed, nothing else. Okay. So tomorrow I am going to finish protocol document, protocol team information. Then we'll enter to the next team, CRF. I think. Yeah, CRF design. What is called CRF? how it looks like, everything we'll discuss tomorrow. Okay, guys, that's all about today. See you. Okay. So still we are at protocol team and regarding protocol document, what and all keywords every programmer should know. That's what we are discussing, visit concept and what is called intervention, what are the study enrollment procedure, what is inclusion criteria, exclusion criteria, what kind of study design, what is called parallel study design and crossover study design, what is called washed-out period, what is called open label and single label, double-blinded and triple-blinded. All these things we discussed, right? So now coming to the next concept here in protocol document. So we are going to finish protocol document. I will enter into the next team, CRF design team, safety assessment. Safety TY. So when we are going with, yesterday we discussed, right, subject visits and trial visits. Trial visits are planned one, subject visits are actual visits by participants. So while doing the actual visits, there will be some unplanned visits also will happen, guys. Unplanned visits in the sense adverse events. If there is any adverse event happen, visit to visit, you can visit suggest, you can visit site. So all these formalities, while doing the screening itself, they are going to explain these and all procedures are there for you, and these many visits are you need to participate. Huh? In between the visits, if you have any adverse event, these are expected adverse event. Adverse event is nothing but side effects. If you have any severe side effects, you need to visit site like that. All the information they are going to provide while doing the screening itself, while signing the informed consent form. Okay. So initially they are going to explain, but they will not mention in the TV dataset. Okay, planning. We never plan, right? Unplanned visits. Unplanned. Why we are calling unplanned and unscheduled visits is nothing but it's just imagination. May or may not. Okay, so side effects will come or may not come. How can we define in protocol document? It's okay. So whereas come to the subject visits here. So once live trial starts, once you approve the protocol document, once the live trial starts, if any of the side effects are happen, so side effects information also we are going to store in that particular table SV. So here actual visits as well as unplanned visits also. Okay, any adverse event happen, serious adverse events. Adverse events is nothing but, I said right, side effects. Severe illness or severe side effect if they have. Okay, that is called unplanned visit. So which is nothing but it is falls under safety assessment, guys. In safety assessment, any expected or unexpected side effects will be there. Both will be mentioned in this particular, expected and unexpected side effects or else adverse events. Information we need to define in this particular section, safety assessment. Okay, let us come to the real-time scenario. For example, say you have visited doctor, guys. For example, not necessarily you are taking the new drug. In case, just in case, suppose you visit the doctor for some reason, you have a headache or you have body pains or you have something, blindness or something. You visit the doctor and he prescribes some medication to you. Okay, when the doctor prescribes medication to you, he will let us know, hey, while doing these kind of medicines, you may expect these kind of side effects. For example, say like bloating or nausea or some kind of dizziness. So they will say, hey, while doing this particular medication, you can expect all these side effects. Once you consume that particular medication, maybe next few hours, or next maybe 1 hour or 2 hours, or next 3 hours, you will be having such kind of illness, such kind of uneasiness. But later on, it should go down. But in case if it is got worse, okay, if it is going to be severe, then they will ask you to visit the clinic, right? Hey, while doing this particular medication, these kind of accepts, these kind of side effects may come. In case if it is get worse, just visit the site and seek the medical attention in the same way. So safety assessment is nothing but expected side effects will be there and unexpected side effects. So accepted side effect will be always they are going to inform to the participants. That is common. Okay, while signing the informed consent, they are going to inform to the participant these are side effects. And unexpected side effects it will happen. What about unexpected side effects? Everything after one start the live trial, there in another domain they are going to define in the dataset. Definitely, we need to send all the information, expected side effects as well as unexpected side effects information also. Okay, so that's the reason I am telling you guys always. Imagine yourself if you are the participant, you are taking the drug. Okay, once drug is taken by, not possible to take yourself, right? Definitely, doctor is the one. We need to prescribe the drug, right? So, or else investigator will be prescribing the drug. So investigators are doctors or clinicians, anyway you can say all are same. Okay, so they are going to provide the drug for you. Drug is nothing but medication for you, either study drug or protocol define any other drug, whatever. So once you take the medication, maybe these are the side effects, these are the expected side effects, but in case they get worse, if you don't go away its own, so sometimes some side effects it will go on its own. Okay, otherwise if it is get worse, just make sure always visit the site, always visit the clinic. Okay, so we are going to take care about that particular adverse events. Okay, investor is the one, he is going to inform to all the participants. Okay, if any participant have adverse event, okay, adverse event information not listed in your actual trial visits information, but if it is a happen, we are going to say somewhere once live trial starts. That is called, adverse event dataset, guys. We are going to discuss adverse event, AE domain. We have separate domain. We have separate dataset. Okay. So because guys, they will always ask the participant to seek the medical attention. So they will usually, especially when it comes to the phase one trials, right? Very first in human trials, phase one. All the investigators very closely monitor the participants because phase one is very risky phase because first in human trials. So what is the phase one end parameter? What is the primary objective for the phase one? Safety is the parameter, right? Safety is the main parameter. So once phase one is approved, then only we are entering into the phase two. Okay. So when it comes to phase two, already safety has approved. Okay, not a problem with phase two somehow. But when it's coming to the phase one, very first we are going to prove the safety, right? Okay, so here they are going to they are closely observe participant in minute to minute, guys, whereas come to the phase one, honestly speaking, because that is first in human. First time that particular new drug is being tested on humans. Risk factor is very high. So each and every participant need to be monitored very closely by the sponsor and investigators. Okay. Investigator is nothing but, as I said, doctor. Okay, you can say doctors, or else you can say investigators, or else you can say clinicians, or else medical team. Anyway, you can call. There are different names in each, so different websites, they are going to provide different names. Don't confuse, all are same. Okay, who give the drug to the participant? Who administrated the drug to the participant? Who will be maintain the patient safety? That is called, investigators. Okay. So this is about safety assessment, guys. In the safety assessment, all these data we need to put. And again, one more section we need to know: intervention, unexpected, discontinuation. Intervention, already you know that we discussed. Any intervention called study drug treatment. Who is participating in that study treatment? That is called intervention. Intervention group and control group we discussed, right? Intervention, discontinuation. Intervention is nothing but treatment. For example, some unexpected circumstances, the treatment may be discontinued by the participant, guys. See, discontinuation of the treatment can be done because of any reasons. There are several reasons are there. Like sponsor. Sponsor, he may not be willing to continue to the participant into the trial anymore. Or participant voluntarily, they will withdraw from the trial. So there will be certain reasons. What is the reason for the discontinuation of the particular participant? In what and all the circumstances, a particular participant voluntarily or like willingly they want to discontinue the trial, or sponsor want him to exist from the trial. If you want him to exist from the trial, what is the reason behind that? Our investigator wants him to exist from the trial. It all depends. Either participant their own, they can discontinue the trial, or else sponsor they can exist from the trial. Investigator wants him to exist from the trial. Okay, all the information they are going to mention in the intervention, discontinuation. All other formalities will be there, right? So if you, while entering the trial, we have a lot of formalities, right? Informed consent form, and next inclusion criteria, exclusion criteria, all these things once confirmed, then only you are entering into the trial. At the same time, if you want to discontinue the trial also, okay, what purpose you are discontinuing? What is the reason for that? Who is discontinuing from the trial? So all the information. Okay, intervention, discontinuation. Intervention, discontinuation. Next, statistical and analysis considerations. Analytical. Considerations. Okay. Statistical and analysis. Statistical or else analytical. So, guys, what and all trials are happening, what and all visits are happening, each and what and all phases is happening. So every phase we are going to collect the data. So once we start screening, once we start inclusion criteria, exclusion criteria, there onwards, once you enter into the trial, you are going to form the sign, right? Informed consent form. There onwards, we are going to collect data. When you enter into the trial, what is your start date? What is your end date? How many visits happen? What is this particular visit start date? What is this particular visit date and date? What is the dosage? How many elements? Each elements, how they are participants. So complete data we are going to store, right? Because without store the data, how can you prove your safety? How can you prove your efficacy of the drug? If it is a phase 1, we need to prove the safety, right? So if you want to prove the safety through mathematical calculations in front of you, in case if you have proven facts in front of you, either it is in a table format, or it is in a graph format, or it is in a plot format. then only you can prove. So if you want to generate the graph, or if you want to generate the tables, or if you want to generate your report, definitely there should be a table. Table means patient information. Okay. So all the data they are going to collect. For example, now you are the participant, starting from, several procedures, as I said, like screening, enrollment, assessments, treatment, randomization, treatment, follow-up. Huh? Is there any adverse event happen? All the adverse event information. The participant will go into the several stages of procedures. Huh? Many procedures are there, right? All the data will be recorded. All the activities will be recorded. Okay, all the activities, all the procedures. So that will call usually data. That is called data. All the data need to be reviewed. All the data need to be submitted to the regulatory, and then only we'll get the approval. So in order to send this particular data to the regulatory, to review the regulatory your data, to approve your data regulatory, definitely we need to collect the data. We need to standardize the data as per the SDTM standards, as per the CDISC. So then, as for your regulatories, then only we are going to send. How data should be submitted? What and all process should happen? What kind of analysis you did with this particular data? Okay, that is called, role of SAS programmer, guys. SAS programmers will always be working with very closely with the statistician, biostatistician. These two combination. I will explain about statistician team and all, but listen. Biostatistician and SAS programmers both work very closely, because biostatistician, he's coming with his plan as per the sponsor requirement. Okay, he will create one plan, SAP Statistical Analysis Plan. He is going to create one SAP document. As in that particular document, what kind of analysis they requirement, what kind of reports there is he need to provide to the regulatory, all the plan of action. Simply, I can say that they are going to create in one document. As for that particular document, the SAS programmers, they are going to work as for your analytical skills and programming skills you are going to use to generate the graphs and tables and reports. Okay, so statistical analysis and what and all statistical insights, what and all statistical results we need to produce. All the based on the statistical results, all the interpretation will be done by the statistician, all the insights will be provided by the statistician, and all the data, analysis happen with the SAS programmers. We are going to produce that particular data to the regulator to review and approval purpose. Whatever the process of the work should be done with that particular data. Okay, when the live trial happens, all the data gets recorded. So all data will be carried out by that particular team. So about teams, I will explain. First, we are discussing about sponsor team and protocol team. Protocol team, what they are going to do, and this what and all documents we should know. That's what we are discussing, guys, here. So flow from which team to which team the data is going to flow. We'll discuss later. One workflow sort. Okay, so all the data carried by one team, that particular team will send to the another team, and then another team, like that. So once we reach to the SAS programmer statistical analysis team, so we are the one. So as for your SAP document, as for your statistical considerations, we are going to perform analysis on it, and that particular one will send to the regulator. So SAS is one of the prominent tool, guys. SAS is one of the popular tool we are going to use to process your clinical style raw data. That's why we are That's why we learn SAS programming, and that programming how to use to deal with the clinical trial study data we are learning. Complete flow. Okay. So here in this particular section only, what kind of data you are going to collect, data collection information. At the same time, once you collect the data, what kind of quality assurance. There is nothing but privacy assurance. The main thing is because when SAS programmers looking for the data, what we receive the data, okay. So this particular data, how we are going to save. So data is nothing but it's like it's an person behind, right? Who is participating in the clinical trial. It's all their data. It's an individual data. So data we need to secure. Suppose imagine yourself, you are the participant. Now it's you have all your data, obviously. What is your age, gender, is there any medical past history? Huh? So complete your information, each and every birth rate information, all the information you are going to provide to you, right? You are going to provide that particular person, they are going to store somewhere. So like that, all participant data. So we need to secure the data. We need to provide the privacy for the particular data, privacy, data quality, data integrity. Okay, how you are providing? Because we keep always using data analytics. Data analytics is nothing but we are going to deal with the data. So whatever domain it is, there we are going to use the data. Any project. Okay, so we always keep hearing terms like data integrity, data quality, data privacy, all these. Because behind the term data, it's like a person. It's a person, right? For example, let's say one real-time use case we'll discuss. You have health insurance. You have health insurance data. Nowadays, we are all into the health insurance, right? So which means that all our data only. So basic requirement that we always seek is definitely data privacy. Okay, we never wanted to our data to be exposed to the public, right? So quality assurance, data privacy, data integrity, what kind of committees are there to do this particular data privacy and data integrity and all, what and all teams will be involved. Okay, so complete information under this particular section we are going to provide. Okay, and again, participant rights also. You can find participant rights. Guys, all these words or sentences, you can find in any document, any protocol document. Okay, participant right. Definitely, I said participant is nothing but the person, who is participating in the trial. What is your rights? Okay, each and every information. What are your limitations? What are all your limitations? They are going to enter into this particular section. Okay, and what kind of different committees are there? What kind of different boards are there? And what is the investigator sign? What is the investigator address? All this information we are going to find in this in your protocol document. So most of the sections, I think I have covered here in the protocol section, all necessary information only. Okay, as for our programming part, if you want to start, once we start the programming part, so there again we'll discuss visits information, event information, okay, all these things. That's why all the information we are discussing in detail. As a programmer, what and all keywords, what and all sections you should thorough with protocol document while doing the programming. So that's what we learned all. So what is the plan and what is actual data? Like how a clinical trial planned and actually how the clinical trials happened. There will be always a comparison, right? So plant data from where we do get? So plant data always will get data from, guys, anyone if you tell me plant data where we'll get? All the planning information where we are going to enter? Plant data. From protocol only, right?
Protocol team. Yeah.
Yeah, protocol only. In the protocol document only, all your planning information will be there. How should start? How should style end? What and all procedures? What and all widgets? What and all elements? How we are going to recruit the participant? On which geographic location? Why that particular geographic location? Are you recruiting only females? Are you recruiting only males? Are you recruiting only kids? Why? All this information, right? What and all discuss last 2 to 3 days classes about protocol document, everything we should mention complete. That is called plan information. Hey, I want to do this particular thing. So I want to start this trial from here. I want to end this particular trial from here. So guys, protocol means before entering into the live trial, what and all your plan sponsor need to mention in this particular document. That document we can call protocol document, plan of action about your complete trial information. So once your protocol is ready, so then you are going to submit this protocol to the regulatory. Once they approve protocol, then only you are entering into the live trial. Okay. So here, what is our work? Already we discussed. What is the SAS programmer work? SAS programmer work is going to be processing the clinical trial study data to the as for the standards we are going to use SDTM implementation guide. Okay, processing clinical trial study data into standards as for the regulatory standards. We are going to convert the data. To convert, we are going to follow some guides like SDTM implementation guide, IZ implementation, ADAM implementation guide. We'll discuss. Okay, so that's the reason why we have good understanding about protocol document, guys. I'm not saying I have covered entire protocol document, but we need to understand. But what I said, as I mentioned, some kind of key points here, this should be understood clearly, guys. So that we'll be able to process the planet data. So how to process your actual data when the trial start? I mean, whatever the protocol being designed by the sponsor, protocol team will be submitted to the regulatory. So once they approve the protocol document, the sponsor gets approval for the submit. Once sponsor get the approval about your protocol, then sponsor can start the live trial. When live trial starts, then actual study data going to be recorded. Okay. For example, version 1, they submitted to protocol version 1. They submitted to the regulator. So regulatory recommended some reasons. Hey, these and all procedures not acceptable. So try to do some changes. So they recommended some reasons. Then, so next, again, same document we'll create like version 1.1. Okay, existing protocol we are going to save. At the same time, copy and paste and create one more. And as for the regulatory recommendations, create new protocol document and send to the regulator. If they recommended again and again version 1.2, something like that. So like this, all the versions we need to store. Okay, so from the first version to till approve the version, all the protocol documents you need to store somewhere, guys. Say, hey, this is not valid version, they are not approved. I'm going to delete like this one. No, never do that. Each and every version we need to store. Okay, so this is about your protocol document, guys. Because most of the clinical trials will be happening for the certain period. So sometimes it's like a month, 6 months, or 12 months, or sometimes it will take years, 2 years, 5 years, 10 years, sometimes 10 years more. Okay, so each and every document we are going to store somewhere. All the data, all the documents. Okay, from the participant data we are going to collect, right? That kind of data will be stored somewhere. How the data collected, and so if you want to collect the data, you need some document. Again, not as usual, not like you. Okay, so this kind of data. So what kind of data we are going to collect? Already we mentioned in the protocol document, right? As per the protocol document, we are going to create one more document, guys. That is called CRF. CRF document. Case report form. CRF means. So based on protocol document, CRF design team, they are going to generate CRF document. So based on the CRF document, we are going to collect the data from the participants once live trial starts. Okay. So once you collect the data, where you are going to store? There is a database management team. Once data is collected, once data is validated, they are going to log the database. Once they log the database, that particular data they will send to the statistical analysis team. Once analysis team analyze their data, they are going to send that particular reports to the medical writing team. So medical writing team, they are going to write a synopsis on your data, and that they are going to generate final report that is called CSR report. That particular report we send to the regulatory for the protocol approval, so for the product approval. So there will be flow. There will be separate teams, guys. Okay. So here we are going to discuss about next team coming to the picture is CRF design team. Once a protocol is ready, once approve the protocol by the regulatory, we'll entering into the CRF document. Now, what is CRF document? Who is going to design this particular document? Who is responsible for designing CRF document, and how it gets designed? What are the basic points we should learn about CRF document? How data collected by using CRF document? And what will be the final log for that particular database? And all these points we are going to discuss in coming classes, guys. Okay. So now, so protocol ready. You submit it to the regulatory approved. Finally, version 1.2, regulatory approved. Let's assume. Then very next thing, CRF design. This is also very important, guys. Okay, now if it is a beginner, in beginner case, CRF no need to learn that much detail. But if you are an experienced programmer, if you want to apply for the jobs with 2 to 3 years or 3 years more, if you want to apply for the jobs, you should thorough with the CRF document as well, because based on the CRF document only we are going to collect the data. Okay, CRF is the main role it is going to play, guys. Based on the CRF document, we'll collect the data, we'll create the raw data tables. That particular data we'll get. And based on the CRF only, we are going to create the spec document. Okay, this table is available in this column is available in this particular table. These variables you need to derive from this particular table. So each and every information we need to do the annotation. So what is called annotation, everything I'm going to explain. First, let me explain about CRF. Annotation spec document. We have separate classes, guys. In detail, we are discussing. Now come to the CRF design. CRF is the one document sponsors should get ready based on the protocol. Because every day, every study, there is a unique requirement, right? So based on that particular study, you are going to generate your protocol document. According to your protocol document, you are going to generate your CRF design also. CRF should get designed. Okay, sponsor can do this particular one. There are some other organizations also, guys, who are, if it is, for example, if it is entire project in a single site, in single house happen within that particular organization, some kind of person sponsor is going to recruit to design the CRF. That is called CRF team. Okay, who will be able to do the design the CRF document as per the protocol. Okay, so in general, we have separate organizations, guys, clinical research organizations. So they should design CRF as per the protocol document. Otherwise, if it is a big, big clinical trial, everything. So sponsor is spending a lot of money. All the teams they are maintaining in the same site. In that particular case, CRF design team, they will recruit and they will design the CRF document within the one second. They will do the same site itself. Otherwise, some of the organizations to generate the CRF design, they are going to hire other peoples from other organizations. Okay. So coming to the CRF, CRF is nothing but case report form. Nowadays, case report form is the play vital role to collect the data. Like for example, simple example, what is the use of case report form? Suppose say, whenever you go to hospital, you will be provided with a form, right? Like maybe if you go to the hospital to get the procedures or else any treatment or else any surgery or else anything, you will be asked to fill up the forms, isn't it? Such kind of forms you can compare with the case report forms, guys. Like form filling basically. In the past, it is used to be paper-based forms. It's like manual entry. There used to be data entry team who enters the data, paper-based data into the systems in the past. But nowadays, it changed, right? Nowadays, it's mostly like EDC, electronic data capture, electronic data entry. I mean, electronic ECRF. Electronic case report form. Electronic case report forms for easy understand. Now you can compare today, like digital form filling, right? Even if you want to apply for the loans, or even if you want to apply for the cards, or even if you want to apply for the anything in general, even if you want to apply for any other. So once you enter into the hospital also, there also, they are going to enter your data in the form only. That form, so it's like a form in front end. It's like a form only, but back end it is going to store like a table, guys. They will do some programming. That is some different kind of story. No need to learn about that particular one. Okay, in front line it's like a table. It's like a paper. It appears only. But whenever we keep filling that particular values, that particular columns, it is going to be stored backend like a table. Okay, so that is called digital form filling. Whenever we fill that form, usually it will be like, uh, so right, document how it looks like. There will be no columns, there is no variables, there is no rows, right? It's like I will open CRF document. How it looks like. Hmm. You can see here. What is the inclusion criteria? So inclusion criteria, exclusion criteria, what is the date of birth, gender, race, ethnicity, age. It's like a form only, na? It's like a form only. But each and every information is going to store in table format. So for that, there is separate teams, guys, database management team. They will take care about that particular coding. No need to do your end as a SAS programmer. No need to learn how this data is going to store in a table format and everything. No need to worry about that particular one. But you can see everything. This is called simple CRF form. Mostly in today's market, guys, in today's clinical trials, EDC only using. There is no paper-based form, electronic data capture only. Well, because what is the advantage of electronic data capture? So if you store your data in paper, if you store your data in Google Sheet or Excel sheet, there is a difference, right? If you store your data in a cloud, you can share that particular data to any person all over the world, which is easy. But if you save your data in a paper, how you can send the particular data? If it is always all documents, it should be like 400 pages, 300 pages, and 500 pages documents. How you are going to submit? If it is a paper-based data, but if it is a electronic data, if it is saved somewhere in the server, somewhere in the cloud, in the table, so if you want to approve from anywhere, if you want to approve from India, or if you want to approve from, for example, some people, they are going to conduct the trial in India, but they are going to approve from the FDA, United States approval. So if you want to send the data, if you send all the documents into courier or else something, it should take a lot of time, right? We'll never do that. So they are going to say through servers only, through cloud only, which is very easy actually, if you save your data. That's why initial days there is a paper-based forms and all, but nowadays we have EDC only, electronic data capture only. Okay, so about case repeat form also, as a SAS programmer, we need to understand, guys. And I said again, I'm telling you again, so we really don't need what is programming behind this particular CRF document. How design this particular CRF document? How they are going to store the data? No need. We need to understand how to do the programming, how to derive the variables based on your raw data. Okay, how to do the specifications, how to do the annotation. These kind of things we need to take care. Okay, so designing the protocol document, designing the CRF document. There is some standards, guys, should follow, like designing case report form. As I earlier said, protocol. If you want to design, we have some standards, guys. I think I said already. PRM, so which is developed by CDISC only. PRM standards, protocol representation model. While designing the protocol document, If you want to submit the protocol document, CDISC developer standards called PRM standards we need to follow while designing the protocol document. Okay. So yes, definitely you should follow the PRM standards, guys. Protocol representation model, as for the CDISC. But whereas coming to the CRF, there is no specific obligation. There is no such obligation like that. You need to follow these standards like that. Okay. But if you want to follow some standards for CRF also, there, CDISC standards, guys. CDISC, DASH. Not compulsory, but we have standards to generate the CRF document, but not mandatory. Okay, so there is no rules and regulations to follow, just CDISC standards to generate the CRF document. Okay, because CRF document usually will not submit to the regulator, guys. CRF document will not send. Protocol document we are going to send. CRF document, by using CRF document, we'll collect the data. That data we are going to standardize and will send to the regulator as per the SDTM standards. Okay, CRF document just for to collect the data, to store the data. We are going to use CRF document. But when it's coming to the protocol document, yes, you should follow PRM standards. Need to be followed. Protocol representation model. While sending to the protocol document to the regulator. So CRF document, no need to submit. Just remember, it's up to the sponsor's choice. Okay, if they would like to follow CDISC standards for getting the CRF design, or else they can skip it. There is no such obligations about CRF document. Okay, I will show you CRF document, sample CRF document, and what and all things we should know. Everything we'll discuss next session, guys, about CRF. This is just introduction about CRF only. So what is the use of CRF? So once protocol document is ready, once sent to the regulator, once you get the approval, very first time, next they are going to recruit CRF to design the CRF. So within the site itself, there will be separate teams will be there. RLs, there is a lot of organizations they are doing nowadays CRF document, guys. Okay, based on the study, they are going to design the CRF. CRF design. What is the main base? CRF design main base is protocol document only, guys. Based on the protocol document only, so in the protocol document you are going to mention, right, all the data, these kind of data this kind of analysis I'm going to do, and these kind of patients I'm going to recruit, everything, each and every plan of action we are going to enter into the protocol document. As for the protocol document, what kind of data we need to collect from the patients, so we are going to design the CRF document. CRF document, if you want to design, protocol document is mandatory. Okay, about CRF, we'll discuss tomorrow. Sorry, not tomorrow. We'll discuss about CRF Team and CRF document later classes. Like Friday, we'll continue the session. Okay, then. That's all about today, guys. I will update video in the time. Thank you. So just we recap what we discussed in the first few sessions. Then we continue with today's session. In the first few sessions, we discussed about SAS programmer role in the clinical sector, in the clinical project. What is going to be our work, how we are going to deal with clinical study raw data to convert SDTM standard data, and all the process we discussed. And we learned about clinical trial phases. Phase 1, what is the end parameter? Phase 2, what is the end parameter? Phase 3 and phase 4. How many phases total? All the phases we discussed. And then followed by what and all teams involved. Once a clinical trial initiated, what and all teams should be involved as part of the clinical trial. So we discussed about sponsors. Sponsor is the nothing but who is the one fund for the clinical trial, which is like you can always say as for the source of the clinical trial. So fund or source or site, whatever, the person who funds for the complete clinical trials. So it can be a person or it can be organization or it can be any entity. Okay. So as we discussed about the sponsors, so sponsor team responsible for all. So once sponsor team is formed, followed by what is the next team? Protocol team, right? Protocol team. I mean, first team sponsored will be looking for forward to recruit the protocol team guys. And we also discussed our protocol team, the responsibilities of the protocol team, and as well as we discussed protocol document, what and all keywords you should learn as a programmer. 1 second. Okay, so we have discussed all the protocol definitions. What and all keywords are very important as a programmer. What and all keywords we are going to do the programming. We should know all the keywords. We discussed all the key points from that particular document. And moving to the next one. What is the next thing we discussed? Once the protocol is ready. Protocol is nothing but what is protocol, guys? Any one of you? What is the use of protocol document?
Protocol document will have like complete information about the trial, what are the inclusion, exclusion, like all detail, end-to-end
Yeah,
details.
where things start and how it should end. What and all procedures we are going to do. End to end, all the information. Protocol document. Okay, this is my plan I want to do with all these participants. These are the participants I want to recruit. These participants I am recruiting from this particular geographic location. So this kind of age group, then these many procedures I'm going to do. So all the information we are going to mention in the protocol document. That protocol document, once we submit to the regulatory, so regulatory review the protocol document, they approve the protocol document. So once they approve the protocol document, then only we are going to enter to the live trials, right? So once protocol approved, what is the next team, guys? Within a moment, after once protocol is approved, we are going to start live trial. So to start the live trial, which document will help you?
CRF next.
Yes, CRF team. CRF design team. So sponsors should recruit the CRF design team. CRF is the document, creates a report form as per the protocol requirement. Sponsors should always hire CRF design team to create CRF. How the CRF looks like and CRF document uses. What is the use of CRF document, guys? How we are going to generate the CRF document? So complete protocol, what and all list of procedures and information you are going to mention in the protocol document. As for the protocol document, we are going to, okay, what kind of data I need to collect from the patients or participants or subjects, whatever. So all the information, we are going to create a form. So based on that particular CRF, we are going to collect the data. CRF is the main form to pick the data. Once the document is created, sponsor will be recruiting two more teams. One is site management team, and other one is clinical data management team. One moment. Sorry, guys. Yeah. So coming to the site management team. Site management team always responsible for the participants, like creating awareness of the people. So who are willing to participate in the trial. So they are going to recruit the participant. They will explain each and everything about the clinical trial. So these are all procedures, so and so procedures we are going to happen once you participate in the trial, right? And site management team is responsible for to fill all the forms. Okay, and then once recruit the participants based on your inclusion criteria, exclusion criteria. Then data management team based on CRF document. Based on CRF document, database management team, they are going to design database structure. Okay, database management, clinical data management is but CDM guys, CDM team. Database will be designed. Okay, once all the background setup is done, so which is particular location, sponsor has interest to conduct the trial, the particular site. We can call it as clinical trial site location or clinical trial site. For which site there will be a management team, site management team, we can call them. They will create awareness. Site management team, they will create awareness for your clinical trial that are happening their location. And then if someone willing to participate, they will explain all the procedures like what will be done to the participant, and still if they are volunteer to join the study freely, yes, they can join without any compulsion or without any force. Okay, and whatever that CRF form is there, so that particular form will get filled by each and every participant and continue into the trial. And so more data gets filled as for the particular form. All data gets collected from the participants. Okay, so we are going to store which date they have collected data from the participant, which date it is started trial, which date it is ended trial. Each and every information, every piece of information we are going to store, guys, in the form of the tables. CDM team is nothing but what they do is like mainly for the database design. Once usually trial period will be, so it will be 3 months or it will be 6 months, certain time period. I think there will be some of the trials which will be happening for the years also, running very long time. Otherwise, most of the clinical trials, it will be 3 months or 6 months or 9 months, max 1 year in the site. Suppose a particular person has entered into the trial 1st January. Then most likely by end of the March or by end of the April, they should be able to finish the, study, a 4-month period or 3 months period or 8 months maximum like that. So all the data gets collected at the same time. CDM team, as said, so they will responsible for the database design, database architect. All the data got entered into the, so we discussed, right? I show you some of the Excel sheets. So if CRF form, it's look like a form, but backend it is going to store like a table. I said, right? To store the table, actually all the time not possible to go with Excel. Sometimes they are going to store, databases also, database table. So once trial is finished live, once live trial is finished, then clinical data management team will be validate all the tables of the data, guys. This team, so what and all data will be entered. So once you fill the CRF form data, it is going to store some of the tables in the backend. The table of the data is clinical data. How it is going to store the structure, the design, yes, clinical data management team only, they will take care. No need to learn that particular one, how they are going to design that particular programming and all, which tool they are going to use. That is not our work. So whatever the tables of the data you collected, running the trial, before once you start the trial till end, whatever the data we collected, that particular data they are going to check. In the clinical data management, there are different teams, guys, like a validation team, database lock team. So they will lock the final database. Okay, once data is collected, once a trial is finished, they are going to lock the database. Once lock the database, you are not able to update anything. You are not able to enter anything. Okay, so how the validation process is going to be done? Usually, in the most of the cases, in the site itself, they are going to validate. Once validation is done, they will to validation they are going to use their own validation techniques or own programming codes or some kind of tools they are going to use, some kind of applications they are going to use, clinical data management team. Once data is validated, so once validation is done, then they are going to be lock the database, guys. So we can call it as final lock. Once the final lock is being done, now sometimes original source like the database will be shared with the following teams. So once the database is locked and clinical data management team, it will send that particular database to the next team, either data analysis team or else any other. We'll discuss next team also. Sometimes what they will do is this clinical data management team, they will convert all the tables, what and all tables are generated during the clinical trial study, all the tables they will convert into SAS datasets, guys. Sometimes they are going to do clinical data management team. So in such cases, you will get SAS datasets itself as your raw data. It depends upon the sponsor, guys. It depends on sponsor and CDM team, but don't expect all the time. In few cases, in very few cases, you can expect that raw data tables converted into SAS tables, and that particular SAS tables will be shared with you. The following team, that is nothing but next team, data analysis team, nothing but our work only, SAS programmers. It will be less burden, right? Yes, already SAS datasets are there with you. You can play with your dataset. So because the dataset is ready, either you can use set statement or you can use SQL, either to combining or else if you want to apply the conditions or if you want to derive something, if you want to create any kinds of the reports, everything you can perform. So otherwise, to import the data, we need to spend time. So once the dataset is ready, your 20% of work is going to be less. One step less, simply I can say that. Suppose say that, for example, data is stored in Oracle database. Now that particular program is provided access to that particular Oracle database. You need to import the tables from the Oracle table to your SAS environment. Then you are going to write your SQL fast through facility. We discussed, right? SQL fast through facility, or else you can use libname method, implicit method. You are going to import the tables of the data. So all the tables once you import, then you can start your work. Suppose if it is an Excel, so some kind of studies they are going to send ready data in Excel format. What we need to do, if it is Excel data, if you receive, either you can use proc import procedure to import all the tables into SAS environment to create the SAS dataset. Correct? So based on the input data, so if it is databases data, we have one kind of procedure. If it is a Excel, we have. If it is a text file, we have another concept. All methods already we discussed as part of our technical training. Sometimes what CDM team will do is such a nice team actually. Sometimes they will convert these Excel tables also into SAS datasets, guys. Those datasets they are going to send to the next team. Will be said to the SAS programmers team. Okay, so some persons, guys, they are going to workaholic or something they will call that particular people. They are going to do as much as possible, how much time If they have more time, they are going to help for the other peoples also. Don't expect all the organizations, all the people will be like some of the persons, helping persons, right? Rarely we can find that kind of persons nowadays because everyone is selfish fellows, any sector, but few of them are still exist, guys, in this world. Okay, so they are going to help with you like that. And that SAS datasets they will save to the class programmers, hence importing the data to the SAS environment. So there is one step is reducing to the programmers. That is all nothing, but that is not a big thing actually. Okay. So, but for beginners, some of the beginners with the basic level, they are going to apply for the jobs with some of the family reference or some of the friends reference. Hey, Maradina? No. 1 second. Okay. Now, so once clinical data management team validation done, they are going to log the database and they are going to send that particular database information sometimes, or they are going to create a SAS table. SAS tables you are going to get. So what is the next team? The following team is nothing but data analysis team, guys, here. So with data analysis team, combination of data analysis is nothing but not only SAS programmers, biostatistician and SAS programmers. So what biostatistician is responsible for? So biostatistician will design SAP plan, guys, statistical analysis plan. Okay, while preparing the protocol document, also biostatistician is going to be involved, guys. Okay, so protocol team, I said right, protocol team. Yes, in the protocol team also biostatistician they will involve and they will explain. So, in the protocol document, they should mention briefly, it should be brief in the protocol document, like what kind of analysis will be done with that particular data. In order to analyze what will be the end parameters, like safety and efficacy, or only safety or efficacy, or anything else. What will be the complete analysis plan? Will be briefed in the protocol document in detail, like step by step. In detail, what kind of analysis will be done with all will be documented in the SAP document, guys. Okay, SAP is nothing but statistical analysis plan. What and all statistical considerations you are going to perform with that particular data. So all the SAP plan, SAP document information, very first we need to mention the protocol document only. So I said, right, protocol document is nothing but end-to-end plan. All the planning information, what kind of analysis you are going to perform, what kind of data you are going to collect, everything. Even with SAP document information also we are going to mention the protocol document, because SAP document, no need to send to the regulator for the approval purpose, guys. But protocol, mandatory. Okay? It's like internal thing. SAP document is internal thing. Biostatistician is the responsible for statistical analysis plan and followed by so once biostatistician is created SAP plan, that particular as per the SAP plan document. So once we receive the data from the database management team, clinical data management team, your work is going to be start, guys. Statistician is the responsible for reviewing the SAS programmers' work. As we discussed, I discussed the SAS program work in the beginning, right? Like processing the study data. So I will write one more time. Processing clinical trial study data to CDISC organization developed standards. SDTM TDAM. Okay, processing clinical trial study data to CDISC organized developer standards. As for the standards, once process our data, that particular data we are going to send to the regulatory for the approval purpose. Am I right? Okay, statistical analysis requirement by the statistician. So here, first we'll convert the raw data as for the SDTM guide. First, once convert, then see it doesn't yield any reports, right? So first we'll convert the raw data into SDTM standard data. So then we'll do the analysis as for the SDTM. As for the add-on, analysis dataset is going to be created. Yeah, coming. 1 second, guys. Okay, CDISC organization developer standards Adams. Adams is nothing but analysis datasets we are going to create. So once analysis datasets is created, then we are going to generate the reports as for the mock cells, TLFs. TLFs will be created as for the template, and we are going to generate the reports, figures, listings, and graphs, right? That is in more detail we are going to talk once we start our live classes, guys, project classes. So today we have covered, as of now, so data management team and data analysis team. Now, coming to the next one. So here only SAS programmers work. What might be SAS programmers work? Already we discussed in detail. Let me talk about few more points here. In general, we always say, other than the clinical, like statisticians, when it comes to the statistician, as for the technical aspects, statistician is common, guys, in general, any sector. But when it comes to the clinical, we are going to call biostatistician. When working with clinical data, the person, for example, who is working with banking data, banking data means we will always talk about profits and loss and like this, right? So like this quarter, this half of the year, what kind of profits we are getting, what kind of loss. Let us implement this kind of particular model, this kind of prediction model, this kind of risk analysis model. So these kind of information we are going to perform, right? How much profit it was made, or how many customers are retained, how many new customers I have attracted when compared with the previous month. So in these terms, you will be talking about out of clinical project, I am telling you, in general, guys. Especially when it comes to the banking sector, retail sector, other sector, it will be target towards sales, right? How much percentage of the sales been increased due to how much revenue has been incremented, how much profit it was, how many new customers been attracted, how many existing customers been retained due to this performance, what is the growth of the organization, what kind of expanding, usually we can call growth. The particular growth is demanding how much we need to expand, how forward to it can expand. So these are the different aspects that we can talk when we compare with other than clinical, the retail market or any other. But when it comes to the clinical project, it's all about the survival, right? It's a new drug. Once the new drug is launched into the market, as we discussed before, then it becomes in the retail chain. Okay, retail chain in the sense it will available in the public to the market for all the pharmacy stores. But before drug is launched into the market, I mean before it gets commercialized, it always should be survival, safety and efficacy. Safety and efficacy is nothing but, in simple terms, you can say that like survival, improving the quality of life, right? So biostatistician is although similar when compared with the statistician like other, in order to analyze the clinical trial study data, still they have to follow. I mean, they have to analyze the data, incline towards the survival rate and the survival estimates. Okay, that is the one of the prominent analysis that needs to be done, survival estimates. And looking at the data, so one, the person should be, the statistician should be able to explain how safe this drug is based on their analysis only. From the first visit to second, I mean, say like the visit to the subject, from taking drug to the next visit, first visit they are going to take some drug, right? From the next visit, what is the difference? What is the performance? What is the difference in the performance? That is why, so one of the most popular analysis that we can call statistician carry out in the clinical project, guys. Calculating change from the baseline. So what is the calculating change from the baseline? For example, let's talk about the baseline. Like one subject, the participant is scheduled with 5 different visits, guys. Visit 1, two, 3, 4, 5. 5 visits. So first visit, all the formalities like screening and enrollment and all the process. Leave it first visit. Subject eligibility confirmation. The particular subject fit for the trial or not. Everything it will happen in general in the first visit. Once subject eligibility is confirmed, so once subject is enrolled, the next other formalities will be done. So when we start the live trial, coming to the visit 2, he is taking the first dosage of the drug as per the plan. Study drug. Okay. So for example, if it is a weekly dose or if it is a monthly dose, it will continue the plan. So visit 1, visit 2, visit 3, visit 4, visit 5. So the study days already we discussed. Some studies it will take 1 week, some studies it will take 2 days gap between the visit to visit. So it depends. Some unexpected visit will be there. Is there any adverse events? Adverse events means is there any side effects? So you can visit the site. All the study visits information already we discussed. I'm not going that much detail here. So here my intention is I want to explain. For example, so what is the baseline after the visit? What will be is there any increment or else it is still stable? We need to check, right? For that, for example, once enter the screening, it's a simple example I'm taking. I'm not going that much detail. I don't know to go that much detail. For example, that particular participant sugar level or else BP level or else, for example, fever, for let's say it will be around 102 degrees Fahrenheit, guys. for example, while screening. So once first visit done, around 50 mg doses, hmm, it should decrease or else it should be same. Okay, so decrease like 100 or else 101. Next increased to 100 mg. Okay, so it is came to 100. For example, next visit 250 mg. 98 for next visit. Simple example I am telling you. 5 mg or 600 mg. 97 normal temperature, for example, 98.5 or something. Huh? So from the baseline, so this is your baseline. Baseline means before entering into the study, before starting the study. So what is the difference? What is the change? How much percentage it is changing? The change will be calculating, guys. That one gives you idea. Yes, am I able to continue this particular study or not? Okay, baseline is nothing but a visit that is done by any subject before taking the first doses of the study drug, guys. That is called baseline visit. Or else you can say subsequent visit. Baseline visit to the subsequent visit is change from the baseline to regular visits. What is the change? How much percentage there is a change? Statistician will be planning for the different other analysis too, like survival estimates. Huh? Different analysis methods will be used to like ANOVA analysis of variance, multivariate analysis, regression models they are going to suggest or advised. Like some of the recommendations by the statistician. As per the statistician recommendation, programmers will work. Okay, so what kind of analysis can be carried out from the based on your data and all the results at the same time? Different parameters also will be calculated, like coefficient of the differences, coefficient of the variance, basic statistics like minimum, maximum, mean values. All these they are going to plan the particular SAP document. As for the SAP document, we are going to work, guys. They will suggest you. As we already have learn SAS programming, throughout the procedures, we can do your analysis. What kind of analysis their requirement as for statistician requirement? What kind of procedures we should be able to do? Basic statistic calculations. Throughout the procedures, we can do always the analysis. So as a SAS programmer, we know like what kind of procedures should be used and what kind of coding should be done as SAS programmer, right? You know that, right? If you want to generate the statistics like minimum, maximum, mean and all, what kind of procedures we need to put? If you want to generate the summary statistics, what kind of? If you want to generate the tabular statistics, what kind of procedures we need to put? Here already we learned. So main work will be carried out by this biostatistician is based on the trial requirement only, guys. Every trial is unique. Based on the particular trial requirement, statistician is going to plan their SAP document. Because every trial, every clinical research or every clinical study will be unique, will be done for unique purpose. So by understanding that unique purpose of the clinical trial for the statistical analysis, what kind of analysis should be carried out the data. So when it comes to the statistical analysis that will be carried out the data, we generally do not call the data. We can call as sample size, guys. Sample size. Sample size is nothing but, how much size of the data we have to carry out the study analysis, because sample size is very important, guys. For example, suppose say you have data with 10 points. Listen, sample size you should know, guys, as a programmer. So you need to calculate the average of the 10 points value. You also have the data 50 points. So based on the 10 points, okay, let me compare. You have 50 points data. 50 data values. You have to carry out the same analysis with 50 data points and 10 data points. Both are constant data, reliable data. For example, in order to check the consistency and reliability of the data, we also have different methods, different approaches. We always have your programming approaches to find the data consistency and data reliability, and all the things. For example, if you have 10 data values, 10 points. So simply I can say that, guys, if you want to purchase a product either in Amazon or Flipkart or Azia or Myntra, so there is a product with 10 reviews, 4.5. Only 10 people reviewed that particular project. 10.5, sorry, 4.5 is the rating. And around 1,000 people are reviewed, just 4.4. So 4.4. Which one you are going to take? 1,000 people, more people, 4.4 review is going to take, or as 10 points, 10 people, just 4.5 it is going to take. Definitely we are going to take more people, even if it is just 0.1 less rating, but around 1,000 people are saying this is good project, right? Am I right? That is comparison, guys. Okay. So what will be the trustworthy point? Is it enough 10 points to calculate this kind of analysis? Or I need to pick more? Got it, guys? What I'm trying to explain? Sample size. So sample size also should be justified in the protocol document initially. Okay. So 10 points also same, 50 points data also same, even if the group also same. But reliable the data, some kind of analysis, some kind of justification. Based on more points only, you can justify easily. Okay. So most of the people are changing. For example, around 100 participants, out of 100 participants, 80 participants are improved their health. Okay, so based on that, I will take my decision. Okay, this is safety. This drug is safe enough. Just go to the next phase, like that. Okay, sample size justification is always important, guys. Sample size is nothing but how much data will be used in order to carry out the data analysis. That we can always call it as sample size. Biostatistician usually put all the analysis plan in the form of the document, which is called SAP document, statistical analysis plan document. Usually the statistical analysis plan document, as I said, it is internal thing, guys. Okay, no need to submit to the regulatory. But briefly about the statistical analysis plan will be placed in the protocol document initially. That protocol document will send to the regulatory for the approval. So no need to send some trials, guys, some sponsors along with protocol document also they are going to send SAP document, but not mandatory. Sometimes it needs to be submitted to the regulatory, but not all the time. It depends upon the study, guys. Simply I can say that. How intense the analysis is going to be. So it depends upon the different factors, like whether the sponsor has to get the approval from the regulatory or not. But most of the times, SAP document, this statistical analysis plan remains internal only. It always a statistician plan only to briefly explain what kind of analysis I need to do to perform to identify end parameters. No need to submit this data. Okay, some of the references will be carried out from the protocol only, like if you want to generate the SAP document, or like what is the study title, what kind of versions they are using. So all this information they are going to mention the SAP document, but no need to send. It's like completely statistician thing, guys. Now, next. See, regarding SAP document, no need to learn that much detail. Actually, we will be not involved in that particular doing work, preparing this SAP document, preparing CRF document, preparing protocol document. So that's why I'm explaining all these things, guys. Preparing protocol document, there will be separate team. Preparing CRF document, as for the protocol document, there will be separate team. Preparing the SAP document, he is the one going to take care about. Statistician is the one. Okay, how much you understand about all these documents. So how much you have knowledge, guys, all these documents, based on that you can organize your work. That's what I believe. Simply I can say that. If you are good with all these documents, if you are thorough with all these documents, okay, where you are working, what is your work is going to be, you will be left in the blind spot. Okay, you will always should know what is the flow is happening. So then it will be easy for you. What needs to be done? When we are doing our own work, why we are doing, it will never be a question mark for us, right? So we can easily understand that this is the reason why I am doing this particular work. At the same time, if something is assigned to me, also you can easily relate to the complete streamline. This is all streamline. This is my work. This entire streamline. This point I am working. So this is my work to finish. This is from this work I am receiving from this particular team. Throughout all process, from the way we are getting input data, after finishing this work, where to send this particular work, for which team we need to submit this work, what is the next process of work will be carried out, who will be carrying out this particular work. When we have this complete streamline of idea, work pipeline, if you know that, it will be easy for us. That's why even if you start 1 week more classes, still we are at basic introduction classes only. Okay, each and every document what we are discussing, all documents are very important to know, guys. So that will gives you a very clear idea about what's happening, what and all teams will be there, what will be that particular team work, what might be my work. So you can completely understand. So usually different teams will be there, guys, not only SAS Programmers team. So as of now, as we discussed around 1 week or classes like that, so you should understand, right? So in clinical trials, there will be a lot of teams going to be involved. It will be a lot of time consumption, and it will be a lot of money investment also a requirement. All this. Okay. Next. At the same time, so different people, like people who've been involved in the creating the statistical analysis plan. So to create the statistical analysis plan, not only biostatistician, guys, along with the principal investigator and statistician, biostatistician, sponsor, all these three combination, because statistician should explain to the sponsor, right? So by doing these kind of analysis, I am gonna prove this drug is safe enough or efficacy or not. These kind of. So investigator will help you. Investigator is nothing but doctor. So doctors and biostatisticians, sponsors. So there will be ethic committee, which will always will be there. Committees, different teams. They are going to involve while doing all this. Okay. Every time depends upon the study how intense the data analysis is going to be, depending on the different internal or external factors. Sometimes regulator also need to approve the SAP document also, guys. Sometimes, rare cases, I said, right? So what is the brief of outline that particular document? What is there in the document? What purpose is being created this particular document? What and all will be explained in this particular document? that kind of acknowledgement we need to put. Acknowledgement is nothing but acknowledging the people who have contributed to generate this particular document in this particular organization. So all the information, all the collaborated work can be done. It is not an individual work, guys. If you are working in the real time, it will always a collaborated work. Sometimes, sponsor, he may collaborate with some other external vendors, some external vendors teams. So it's just like an acknowledgement, acknowledging all other contributes, like disclaimers. What kind of copyrights will be there? So all this information, very important, guys. Table of the contents, list of the observations, everything we are going to discuss once we open the SDTM SDTM implementation guide and ADAM implementation guide and all these things. But as of now, we discussed what and all teams involved in this particular trial. All the teams information we discussed, guys. Up to this clear, any questions? So what clinical data management team will done, what data analysis team will done, what site management team work, what is the CRF design, what is the protocol. So along with teams work, we discussed protocol document, how protocol document looks like, what and all information we need to put, and CRF document. So what is the CRF document? Once I enter the data in the CRF document, what is going to happen? All these things we discussed. Next, we are going to discuss SDTM guide only, guys. Workflow, industry workflow, I will explain. And then we'll go to the SDTM standards. So as of now, we discuss the teams work only. So once all teams work is done, what is going to be next? Complete flow, industry workflow, we are going to discuss in the next session, guys. This is also very important. We'll connect all teams work again. Done. So that's all about today. We'll continue tomorrow, guys. Tomorrow also we have class, guys. There is no more holidays, okay? Yeah. Thank you, guys. See you tomorrow. Same time, same invitation. Yeah. So here we are discussing data analysis team. Coming to the data analysis team, biostatistician and programmers both are going to work collaborated work, and biostatistician will create the SAP document as per the protocol requirement. So as per the study requirement, he is going to create a statistical analysis plan. Based on the plan, SAS programmers are going to work. Okay, so we should know what is called SAP and all. Already we discussed. Today, what I'm going to show is, what I'm going to explain is, I will explain few of the important keywords in the SAP document as well. Okay, so about protocol document, what are the important definitions we should know, we discussed. About CRF document, what and all things important we discussed. At the same time, SAP document also. Okay, so no need to involve to create SAP document, CRF document, protocol document, but you should know as a programmer what and all keywords we are going to use, right? So, and guys, after that, once we discuss about SAP document keywords, and then, so once we are done with our work, the last team we are going to discuss today, medical writing team. Okay, so what will be the medical writing team work we are going to discuss? Okay, see here, let me show you some of this SAP document. I will update all these documents in the drive, guys. See here, so in this particular statistical analysis plan, it's a complete, just I will show you briefly, and each and every keyword I will explain once we discuss with the document. See here, some of the reference number. This is about your study title, which phase it is, and who is the sponsor, where site is located, sponsor signature, principal investigator details, and phone number. So which version it is? I said right versions we are going to create. This is first version. So signatures, approval signatures, table of the contents, and abbreviations. AE means NO means analysis of variance. So all these abbreviations we'll discuss later. And next, introduction. What are the primary objectives and secondary objectives? What is the study design? Single sequence or open label? What trial phase it is, and schedules, schema. What is the schema you are going to use? Treatments and sample size justification, definitions of analysis populations, safety population, PK analysis, everything I'm going to explain. Pharmacokinetic assistant, demographic subject disposition, safety and tolerability assessments. We are going to discuss, is there any adverse events, expected adverse events, vital signs, electrocardiogram, data presentation, how to present the data, what kind of interim analysis. Interim analysis is also very important, guys. So all the studies may or may not possible interim analysis. Some expensive studies only. We are going to perform interim analysis. What is called interim analysis? All these things we'll discuss. So data presentation. Okay. So about SAP document, all these important keywords we are going to discuss. Okay. So because as I said yesterday, you should know about all the documents, guys. So then only if you know the unit analysis, if you don't know the what is the complete streamline, you may feel what you are doing, what is going to be your work, why you are doing. You may get confused. Okay, once if you know, it will be easy for us. Okay, will not be left in a blind spot. Basically, when you are working, that gives you very clear idea. Okay, what's happening, why I am doing this particular work, why should I do this? Okay, so I'm trying to keep simplify here, guys, as much as possible in introduction part, as we are still at introduction part only. Okay, once we start the programming classes, so how much knowledge you have in the programming, based on that the programming classes you can understand. If you are good at basis programming and procedures, SQL and macros, then okay, it will be easy. You can understand how we are importing the data, how we are deriving the variables, what kind of statements we are using, what kind of attributes we are setting the variables, and everything. That is not a big deal. Okay, only we need to spend the time. What is the variable, what are the variable attributes, and how to derive that particular variable. What is the use of that particular variable? Okay, so all names, it looks like familiar, guys, similar names. Just one or two characters only, it will change based on the domains. So you may get confused. Okay, yes, RF SDTC, RF ENDTC, like that, RF XST DTC, RF XEN DTC. Just one character only change, but completely different uses that particular variable. How to derive RF SDTC, RF ENDTC, how to derive. What is the meaning of RF SDTC? RF SDTC mostly we are going to use, that is the column, that is DTC means date and time in character format. Okay, when first doses of the drug is going to given to the participant, that is called RF SDTC. Like that, just you need to spend the time about the variable definitions only. No need to spend time about the programming. Okay, so definitions only we need to understand there only. So why I'm saying like that? Because already you learned technical training, right? Most of the people, what they are going to do is, without prior knowledge of technical training, they are going to directly enter to the project training, guys. Okay, maybe they can understand theory part, may not understand once enter to the domains SDTM guide. Hmm? Through SDTM implementation guide, if you want to import the data, if you want to standardize the data for SDTM implementation guide, with help of a lot of conditional processing, with help of a lot of options, lot of functions, lot of procedures, there you may get confused. Okay, there again we need to spend time to learn that particular procedure, why this procedure is generating this particular output. Okay, for example, by using proc summary, what kind of summary statistics you can generate? If you don't know, how can you use that particular procedure, right? Like that. Okay, so now coming to the SAP document. Usually different people will be involved, guys, to create a statistical analysis plan. So principal investigator, statistician, sponsor, all these they are going to involve to prepare this particular SAP document. So especially statistician is the one is going to give the suggestions. So these kind of, because statistician work is going to be, to generate the statistics only. Now, what kinds of statistics are required for this particular study to prove drug is safety and efficacy, like that? Okay. So as I already said, SAP document is completely internal. No need to submit to the regulatory. Depends. Okay, depending on the different internal external factors. Sometimes only around 10 to 20% of the case only we need to send SAP document also to approve the. We are going to send SAP document also to the regulatory to approve. Otherwise, most of the cases we never send. Okay. Now, so very first we are going to use purpose of the document. What purpose? What is the purpose of SAP document? Complete plan of action. What kind of analysis is required through this particular study data? See, for example, guys, here, as of now, what we discussed around, 6 teams we discussed, right? Let us come to the protocol team. Which document we received? Protocol. So based on the protocol, CRF team, they are going to generate CRF, case report form. Okay. Site management team, they are going to recruit the subjects. While recruiting, they are going to collect some data. Data collection will happen here, like inclusion criteria, exclusion criteria, informed consent form sign, all the demographics information like name, age, date of birth, geographic location, ethnicity, and everything. Is there any adverse event happened in the past? All this information. So data collection will happen here. So clinical data management team, what they are going to do, guys? They will validate the data. So based on the CRF document, they will create some database structure to store the data in a table format, right? They will log the database. They will, once validation is done, they are going to log the database. So here only, mostly in the clinical data management team, they will deal with your study data. So or else raw, you can say raw study data, either in table format. So table can be, of course, after raw study data, it will be definitely in the table format, either Excel table or else, CSV or else database file, database table. So there will be different sources. So as I said, some of the times they are going to create directly SAS Dataset itself. Okay, protocol document is ready, CRF document is ready, data collection is done. So once data is collected based on the CRF document, so CRF document site management team, they are going to collect it, and clinical data management team, they will design the database structure, database architecture. Then, so here tables got created. So here they are going to check the validation. So validation is nothing but, guys, so there is a gender variable. Gender variable, it should either male value or female value only should be there. Otherwise, male and female, if you have any other related information, they are going to delete that particular value. Data cleanings, data validation, and everything, it will be done. Data management team. Okay, and again, so they will create the structure. For example, simple example I am going to explain. If you enter your Gmail in your Google, if it is a valid Gmail ID, then only it will allow you, right? Otherwise, this particular Gmail is not there. Okay, or else once enter your Gmail ID, definitely it will ask you password. So if you enter exactly what password you set for your Gmail, then only it will allow you, right? That means already your mail ID and password saved somewhere in the database. Somewhere. So once match, then only it will allow you to log in your mail ID, right? Same, this clinical data management team, while generating the structure, they are going to set some conditions like this only. So if there is a gender column, if you enter either M or F or male or female, then only it will allow you. Otherwise it will, so there is invalid data like that. It will not allow you, right? So just example I'm telling you, like that they are going to set some structure to fill the data. Okay. So even though they are setting all the things, still they are going to check the validation. Once validation is done, they are going to log. So raw data is ready. So protocol document is ready. CRF document is ready. Raw data is ready. So all these documents. So now come to the data analysis team. Here in the data analysis team, SAP document is ready. So biostatistician is the one who is going to create SAP document as for the study requirement. Once SAP document is ready, all these documents will share to the SAS Programmers guys. Protocol document, CRF document, raw study data, and SAP document. Yes, these are more than enough to work, guys. Okay, so based on the experience, so for example, you are the 5 years experience guy, then I know I said right many times, there will be certain hierarchy, guys. Different teams will be involved in this particular thing. Not only one programmer doing this particular thing. Not only one programmer, 2 programmers, or 10 programmers. Okay, so there will be junior level programmers, or senior level programmers, and intermediate level programmers. There will be a lot of hierarchy. So now coming to the, see here, whereas coming to the, for example, let's assume you are the guy, you have 5 years experience working with that particular organization. You need to take care about CRF annotation. So what is called CRF annotation? We are not done yet. I'm going to explain. CRF annotation means based on your SDTM guide. Okay. So based on SDTM guide means, for example, for example, study ID. Study ID is the variable. It should be unique study identifier where it will be there in the CRF document. Or else, one second, guys. Section 5. Okay. Demographics. This is the one easy for understand as a beginner. For example, age. In the CRF document, where age is there. So this is dm.age variable. So that particular variable I need to save in the dm table. Okay, domain is nothing but SAS Dataset, guys. Okay, So like that annotation will happen. Annotation means just mapping. Okay, so that is completely, that is like a form. I will show you annotated CRF, which will be there somewhere. You can see date of informed consent date, which variable value it is. DS SDTC, DS domain, disposition domain, SDTC is the variable. Okay, so like that, for example, so here, so inclusion criteria, exclusion criteria data not met, IE dataset. Here category IE cat, inclusion exclusion criteria number, IE test CD code. So these values, it will be stored like a column, right? Which column it is and which data set it is. This is called mapping. This mapping. See, coming to the DM dataset. In the DM dataset, so this is birth etc in DM domain. So sex, this is the variable we need to save in the DM domain. And race is the variable we need to save in the DM domain. So all these ethnic, age, and all complete. This is called annotation, guys. So basically, this is the one. Like this only we are going to do the annotation. But not you. No need to worry. So who is have more than 5 years experience or 7 years or 10 years experience, guy people. So mostly senior level programmers only, they are able to do this particular annotation. But you are the one, if you're able to do this, this is really wonderful. You can, can demand. So not that much big deal, guys, to do this particular annotation. If you are good at SDTM implementation guide, if you are good at all the variables, then you can easily mapping your CRF document. You can do the annotation. Okay, annotation is nothing but just a set mapping. As for say SDTM implementation guide, what and all columns will be there. Okay, so CRF annotation will be happen. Once annotation is done, then there is another team, guys. Specification document. Specifications means like this. Yes, annotation also I am gonna teach you. At least 1 day session or 2 day sessions will discuss annotation, Thomas. Okay. Where we are, what I'm trying to say here. Yeah, spec document. So based on the CRF document, specifications document is going to be ready. So there is separate team. So once CRF document is ready, spec document they are going to create, like spec document, how it looks like. Let me show you. SDTM specification document like this. See here. I will open any one of the real-time spec document. Yeah. For example, you can see. So these are the variables, right? For this particular data set, for this particular domain. How to derive? Which table this particular variable value is there? So for example, RFSTDTC, this is the column. This is the column. If you want to derive, you need to import the raw data set rand. By using rand table, you can import the data and you can create the table. So what is the length? What kind of variable it is? What kind of format we need to apply? So this is all this table. So this is already available in, up to this, it is available in SDTM implementation guide. Here it is, you can see till core. See here, variable name, variable label, type, role, CDISC node, score. This table is copied from the SDTM implementation guide only. So specifications means like this. So is this data is collected or not? Is it necessary to maintain this particular variable in the data set? Yes, data is collected. We need to maintain. So what is the length? So here, what is the value for this particular variable? Some variables are direct variables, guys. No need to derive. Directly you can set the values. In the specification document, they will provide the values. Simply you can assign the values. How to create this specification document? Guys, this specification document based on the annotated CRF only we are going to create it. Once CRF is annotated, CRF is annotation is done. So annotation means simply you will map. Okay, this is the column for this particular table, this particular table, this particular table. Once annotation is done, so all the table how to bring to create a simple data set. From which table, which variable is there. In DM table, subject value is there. So just create. So I said, right, we are going to collect the study data here. Raw study data, tables. So this is the raw study data. And by using annotated CRF, by using raw study data, spec document, all these documents they are going to provide. So you are going to work, guys. SAS programmers, work with start. One second. Okay, about documents, I will take separate session, no worries. Just which team, what kind of documents they are going to provide. I want to explain that particular concept only. So protocol team, protocol, CRF, this is the flow. Okay, so data management team, they are going to collect the raw study data. And here data analysis team, they are going to send a biostatistician SAP document. All these documents are, they are going to provide, guys. So may or may not, for example, all documents access, for example, if it is a beginner level programmer, they are going to send, okay, so they are going to send only like, for example, I am dealing with only two domains. I am dealing with DM and sub DM. So this particular domain spec document only they are going to send. This particular domain spec document, and see here, study data to derive DM and sub DM. Study data and spec document. These two enough, guys. These two along with the SDTM implementation guide, you can derive the data set. Simple. Study data is there. Mapping specification document is there. Which variable, which table is there. So what else required? Nothing else required, right? By using your programming skills, simply you can create a data set. Got it? So come on to the SAP document. So purpose of the document, what is the purpose? What purpose will being created this particular one? All that will be explained this purpose for document section. Next, acknowledgement. What and all people, who control this particular clinical trial site, who is, principal investigator. Okay. So complete information acknowledgement. Usually, in order to get this document done, it will be always a collaborated work, I said, right? Okay. So it cannot be done by using individual person. When we are working in the real time, it always teams work. There will be certain hierarchy. Sometimes, sponsor, he may collaborate with some other external teams or external vendors to create this particular document. Okay, it's just all like acknowledgement. All the acknowledgement it will be done. All the contributors information and disclaimer. And this CODIMER something. Please do ignore my spelling mistakes, guys. It's like we can say what kind of limitations and what are the copyrights. So copyrights, guys, simply I can say that disclaimer is nothing but. And table of contents. If it is a 15 pages of the document, so definitely you should mention, right, which page, what concept is there. Table of contents. So list of observations, abbreviations. Okay, you can, guys, you can use these documents in the real time. Okay, SDTM implementation guide you can carry with you. SDTM implementation guide, the SAP document you can carry, and all these you can carry. So only interview, while doing the interview, you are not able to carry these documents, but otherwise, when you are working with the real time, all the documents you can carry. You can check if you have any questions. Okay, because in the SDTM implementation guide, so each and every column, what is the use of that particular column and what kind of variable it is, everything it will be there in detail. Okay. Assumptions is nothing but what is the purpose of this particular domain data set. By using DM domain, what kind of data we are going to say. Okay, there are a lot of examples it will provide. See here, how much knowledge you have in the implementation guide. That much easy you can do the programming, guys. That much easy you can derive the data sets as well. But no need to learn all 450 pages. See here, around 400 pages of the document. No need to learn. And what kind of assumptions you need, I will guide you guys. So this is about, I'm talking about SAP document, all this. All the observations. For example, MI means what is the, until unless if you don't know MIN. So for example, in the SAP document, I want MIN, I want MAX, I want SVM, like that. If they are providing some keywords, so the detailed information, list of the observations, there you can check. The full form will be explained, whatever observations is there being throughout the document. Okay, throughout the document, they are going to use sort names. The sort names abbreviation, there you can find corresponding section called list of abbreviations. Okay, like coefficient of variance, CL is nothing but CL like a confidence limit, like that. What kind of constant limit being used for the considering for the data analysis, all these things. Okay, whatever the abbreviations throughout the document will be explained. All the abbreviations information it will be there in the list of abbreviations, guys. Okay, and again, so as I said, study objectives. Study design. Okay, actually, what is the study design? Already we discussed. Where we discussed, guys? Where we need to put the study design very first? Protocol. Okay, so as we mentioned already in the protocol document, here also we are going to mention what kind of study design it is. 1 second. Okay, sample size justification. Sample size is nothing but, this is about data. What calculations will be done? Okay, how many number of subjects I need to enroll into the study? Okay, if it is a less member of the study, if it is a less data, usually some results will not be reliable, right? We cannot rely on that if it is a less data. Based upon what kind of analysis will be carried out, the number of participants need to be decided, guys. For an example, say based on the data analysis requirement, statisticians say the target is 100 people. Okay, so it is not that only 100 people will be recruited. Always a buffer should be considered, because not necessary that all 100 people will be completed the trial. So if someone exists in the trial, in middle of the trial, that is possible, right? They can exist from trial voluntarily, or else maybe investigator trick them, or else some of the participants will die. All these are expectations. Okay, so but at the end of the day, 100 people's target is there to do this kind of analysis. So because no one will be forcing the participant to continue the trial. At the same time, suppose say during the trial, if any participant is suffering from the adverse events, serious adverse events, which is like life-threatening adverse events, because of that, because of study drug is something which is not safe. So if the person is dying because of the study drug, or if the person is died because of some accident, For example, he is participating in the trial, but he died somewhere because of some other factors. Okay, not related to the study drug. Maybe he died with some other, maybe there is a tsunami, or else maybe a earthquake, or else accident, or whatever it is. It maybe there is a corona. Okay, so still, that person will not be available for the trial anymore. So target is 100. So if you want to get 100 papers completers, we can call completers, guys. Okay, so if you want to get 100 people completers, we need to recruit at least 120 or more than 120 papers. 1 second, guys, 1 second. If you recruit 120 people, at least in order to achieve 90 completers, or at least 100 completers. If it is 120, you recruited 120 in case if it is a completed, which is really wonderful. Not happen regularly. If it is happen, that is great. Not happen, not possible actually. Okay, so then, for example, guys, they recruited 100 participants or 110 participants, and 90 completers are done. So, but here the sample size is at least 100 participants is required. So then again, no replacement will be considered, guys. They are looking forward for the new people. Of course, trial period becomes little lengthy when we go through the real protocol, and we can see that why that particular point would have been mentioned there. Definitely keeping this kind of data analysis, I am recruiting these people like that. Okay, so analysis data is very important. What kind of sample size? If it is a less data, there is no point. So that's the reason why we keep always in mind. Okay, mainly statistician will always recommend a specific number, guys, to do this kind of analysis. We should be doing this kind of analysis for this particular study. Okay, so I need this particular number of subjects. That is why even in the protocol document also, why this particular many number of participants? We need to give the justification, right? Already we discussed. There will be justification section. Sample size should be justified. So there will be different methodology involving in analysis. Okay, so sample size justification, of course, even though you mentioned in the protocol document, in SAP document, as for the statistician main requirement, they are going to mention. And again, analysis population flags. Flag is nothing but yes or no. Usually when we completed the screening, what is the next thing it will happen? Enrollment. And other formalities. Then we are going to wait for the treatment. Okay, very first visit, screening will happen, inclusion criteria, exclusion criteria they are going to check. So, and all the forms, what and all forms and formalities are we need to finish. Then first visit that is going to happen. Then next visit, we are going to enter in the treatment. So not all the people, but who is confirmed, who is eligible for that particular study, they are going to treat, right? Okay, so the group of the people who are participating in the trial, we can call ITT population. Okay, intensive treat population. And again, safety population. Safety population is the group of the participants where received the at least one dosage of the drug. Huh? So not necessary to take complete all the doses of the drug, but at least one dosage of the drug they are going to take. They should give in, but there is no effect. There is no adverse events. So that is called safety population. Okay, so I have finished all my formalities, and finishing all formalities and intended to treat. But say like for some of the reason, I have stepped back out. I don't want to participate in the trial anymore. Okay, that is called, guys. That is called comes under safety population. Okay, because I don't want to continue the trial. Haven't taken the treatment. I have, so even though I have filled formalities and then I have received my first dose of the drug as well. It's like there is no effect. So that is called safety population. Okay, so here the falling into the group who taken the first doses of the drug we can call safety population, guys. And per protocol population, these and all information it will be there. Per protocol population is in the way there will be always some set of the people who will follow the instructions as it is. Like there will be always some set of the people who follow the instructions. Like for example, while participating in the study, you will not allowed to smoke. While participating in the study, you are not able to drink. Okay, so there will be few participants who didn't smoke, like follow the instructions exactly. There are the few participants. They always violate the rules. Some of the participants. Say like the one participant, he smoked, had drank. That means the participant is not as per the protocol. He violated the rules, isn't it? Right? Okay, so the participant who follows the protocol is nothing but plan of the action, complete planning. The plan, the instructions, the procedures, the person who follow the protocol as per the plan, like without any deviations or without any violations, violations of the study rules as per the protocol document. That is called per protocol population, we can call, guys. Safety population. Okay, so first doses of the drug and per protocol population who is maintaining exactly all the rules, who are following all the rules as per the protocol, per protocol population. Like this, there are few more definitions too, but most popularly used definitions we are going to discussing here. Okay, next, statistical methodology. So here in the statistical methodology section, very detailed explanation will be there by who is the one biostatistician. Like in general, complete information they are going to provide in this particular statistical methodology. Okay, statistical methodology is nothing but what kind of descriptive statistics will be calculated, what kind of inferential statistics will be calculated. Inferential statistics is nothing but like um, regression analysis, multivariate analysis. These kind of analysis will be carried out. Okay, so the complete information, statistical methodology section they are going to mention. And next, data imputation. This is also very important to know as a programmer. Data imputation is nothing but submitting the missing data with a value, previous value or next value. For an example, say, let me take a single example. There is a participant, say one of the participant done 5 different visits. So which particular dates I have done visit being recorded somewhere, right? Hmm? For example, there is a visit 4. So visit for this particular date, data is not recorded. For example, I said every visit information we are going to record it, every information, every piece of the data once you enter that study. So where you entered first visit, where you finish the screening, where you entered the first 2 doses of the drug, next doses of the drug, next visit, all the visits information, there will be separate table called subject visits. We have one domain. All elements information, like what kind of treatments they are going to provide, elements information, like screening and treatment and follow-up, that is another domain. All subject information, another domain, demographics. Huh? So like that, we have separate tables. Based on the particular table, we are going to send the data to the regulator. They will simply analyze, because when going with big document tables and graphs will gives you comfort, right? While analyzing the data. Same thing. Okay. Now here, some dates are not recorded. Usually that this doesn't happen. Just for example, I'm telling you, the date you need to be recorded actually. That is very important and essential information. Okay, I'm telling you just for example. So whatever the data that's been not recorded, it should not be left like a missing data, guys. It should be updated with some other data. That is called, like data imputation. So data imputation is something, but that is the technique to fill the missing data. Okay, there will be different methods replacing missing data values. So, to replacing missing value with mean values or median values or, mode values, or else with front values, forward values, or else backward values, back values. So forward and backward values, they are going to use to replace the missing values, either date or whatever. Okay, replace the visit date for 4th visit date, either minimum date of the this particular visit date or else worst date value. Okay, sometimes whatever the worst value, replace with that particular one. Okay, replacing LOCS. This is also you can find, guys. LOCF, last observation carry forward, which is backward or forward field. Like previous observation data will be replaced. BOCF. Baseline data. Okay, so replace with missing data with baseline data, which is BOCF. OOCF, worst observation. Like among the data values, all the variables, whatever the, data you consider the worst as per the particular parameter, just replace with the worst value. Or sometimes we follow the regression imputation, which is like, multi-imputation technique we follow. But, either worst value or else previous value or else back value, or else mean value or median value. So there will be certain standards we are going to follow, guys. So that the technique will follow. So that is called data imputation. Data imputation information also we are going to mention there. It will be always biostatistician call, guys. He is the one, take care. Okay, he will mention in the SAP document what kind of imputation. So as a statistician and SAS programmer should be working together. They are going to work, and the work will be done combination, right? So he will suggest your statistician, hey, just replace with the mean value if you have any missing value, or else just replace with the LOC of last observation carry forward value, just use that. So like that, he will suggest you. Okay, so moving on to the next one, assessments. Like I said, mainly safety and efficacy. Safety and efficacy are the parameters that should be evaluated. You need to prove. So what are the primary and secondary endpoints? And what kind of subgroup analysis? Like, what kind of confidence intervals? Which software will be used? How to import the data? How to generate the results? Okay, different kinds of assessment will be explained in this particular section. What kind of data analysis in generally, happen? It will be explained in the analysis section. It will be elaborated what kind of formulas, what kind of classifications you need to follow, which is nothing but statistical parameters and all definitions, calculations, all the information. And the safety and tolerability assessments. It is very important. Safety assessments, like what kind of risks were involved in the treatment. If the treatment is effective, safe, okay, fine. If it is not safe, okay, participants are not safe, then what kind of assessments will be there? Tolerability assessments is nothing but some cases, mainly in the phase one, guys, mainly in the phase one, first in the human trial, right? So each participant should be monitored very closely, like what kind of side effects usually happen in daily basis. They will be asked to fill their log, okay? And so so doctor or investigator, he's the one, hey, every day, if you feel any uneasiness, if you feel any adverse events, just fill up here. This is the log, all the activities they are going to follow up, guys. What kind of side effects they have, in case of that particular subject or participant is experiencing any adverse event, any side effects, what kind of responses should be considered, what kind should be dose is reduced or should be dose is increased, it should be terminated from the trial. All the dose adjustments and terminating from the trial and all the tolerability assessments will be done, will be mentioned here. Usually in the phase one, almost like a day-to-day basis, each and every person need to be monitored so very clearly to their soft safety and tolerability. Such kind of assessments will be done. Adverse events, clinical parameters, other assessments, all these vital signs, laboratory results, already, all this information will be there. And again, coming to the interim analysis, last topic for today's session. So interim analysis is nothing but it will be carried out for all the studies, but in general, where the interim analysis comes to the picture is because for the doing interim analysis, it's like very expensive trials only. Honestly, I can say that because it involves like, so every clinical trial study, it will take time and money and resources and a lot of teams involvement. Everything it will be happen, right? Okay. So whatever the analysis, whatever the statistical analysis and statistical evaluations need to be done, so will be done in different intervals. Like after 20% of the phase, or 20% of the data collected, or 50% of the data collected, or 40% of the data collected. So we are going to do the analysis. For example, let's say sponsor is aiming for the 100 participants. Let's say like once 40 participants have completed the trial, which means around 40% of the trial is done. So once 40% of the trial is done, the data analysis will be carried out, guys. Just they are going to check the trial is going to growing in same direction what I'm expected direction is going or not. If it is going as per their expectation, then they will carry it out. Otherwise, the rest 60% people are 60% or less. So at this point, usually sponsor can make a decision like whether like to continue the trial or terminate the trial based upon the results. With the 40% of the results, as you have already 40% of the data you collected, you did some analysis, and based on the analysis, either continue the remaining for 60% of the people, or else I should terminate this particular trial. So 40% sometimes. So no, something like like maybe in 20%, 60%. So once 60% data is available, they are going to run again analysis, reporting all the data. They will do the analysis again. So because every clinical trial study, it will involves a lot of cost. At the same time, a lot of teams involvement. So mainly cost is involved, right? So interim analysis, usually we cannot expect for all the studies, but usually where the studies are very expensive, where billions and trillions of the money need to spend in the clinical trials. It's like a lot of money gets invested into the clinical trials. So in such cases, if for example, you set some limit, guys, you set $10 million, okay, $200 million. Once cross the limit to just 40% of the data, once collected, 40% of the trial is done. So you cross the limit, you are crossing the expected one. So such kind of scenarios, sponsor may go for the interim analysis. Okay. So then at the same time, even safety also can be accessed, safety and efficacy. So one of the crucial reasons, definitely if the treatment is not effective, not up to the mark, then it can be stopped early. To avoiding waste of the resources, waste of the money, waste of the time. So we are going to perform interim analysis, guys. Okay. So saving the money, mostly I can say that. And again, after 40% or after 50% of the data, he will get conclusion. Okay, so this study, as I'm expecting, it is going same way. So he will get confident. He is going to spend more, right? So this is about interim analysis. Okay, that's all about today. A few more things we need to discuss about SAP Document data presentation and insufficient data. If you have, what we need to do, all these things we are going to discuss next session, guys. So next session is last session. There I'm going to finish SAP Document with medical writing team. Then we'll enter to the industry workflow, guys. Okay, done. That's all about today's session, guys. Let's wind up the session for today. We'll continue tomorrow. Thank you, guys. Nagaras, see, actually, introduction, 2 days classes only, but we are still, it is, I think, 8th day or 9th day, but still we are in introduction only, na? Only introduction, it is taking 10 days. So once enter to the SDTM and all, I'm not sure it will take 3 months more. 3 months more it will take. And if you feel if you don't have that much time, let me know, guys, any one of you. If you feel if we are going very detailed and if you feel like waste of time and all, I will send existing batch videos. Just go through them, go through the videos while doing the practice with videos. If you have any questions, then you can ask me, and simultaneously you can attend these classes as well. Both options are available. Okay, existing batch videos you will get, and you can practice per day 2 to 3 hours, 2 to 3 classes like that. And while doing the practice, if you're stuck somewhere, if you feel uncomfortable there, you are not able to understand, even though video also, then let me know. I will try to explain. Okay, just swing it through WhatsApp like that. If any one of you want to finish complete project training like 1 month or 45 days, just like that. Okay. Yeah. Because programming training, when we started actually programming training, we started and actually August. 3 months course only actually, but it's take around 4 months more, right? August, September, October, November, December, 5 months we discussed. Actually 60 to 65 classes only, but around including today class, we discussed around 75 classes we discussed. 90 days, if you skip the all days and all, So around 60 to 65 classes only, but we discuss detail, right? So it's taking time, that's why. So buffer will be there always. I'm not able to say specific time I'm going to finish this particular training, guys. Okay. So like other institutes, they are going to, they will schedule the plan. So this particular day, this topic. So next day, next topic, next day, next topic. So because after finishing the 45 days or 60 days training, they are going to start new batch. Okay, so they will explain the concept, but they will not check with the students. All the students are good with that particular concept or not. But in our institute, not like that, right? If I explain something, if you are good with that particular concept, then only we are moving to the next concept. That's why it is taking time. Okay, so all in all, we discussed last session. This is all about your SAP document, statistical analysis plan document. So this is completely created by the biostatistician and biostatistician and team. So once the SAP document is done, followed by its once SAP document is done, it will send to the programmer. So then it is going to be SAS programmers work. Okay, so there we are going to responsible for your work. What is your work is going to be, right? Submitting the study data as for the SDTM standards. So where we get study data, already we said, from CRF document, right? So based on the CRF document, data gets collected and stored in form of the tables in the database, which is completely validated by CDM team, clinical data management team, and CRF design team and site management team. All three together involved into this particular study data. Okay. And from there, we'll get the raw data or study data. Already I said yesterday. So based on the CRF document, all the study data should be sent to the regulator. Am I right? One second. Who is this? Okay. So, but cannot submit it to as it is, right? The data should not be processed, should be processed following the document called SDTM. SDTM, Study Data Tabulation Model. So it's the name itself telling, Study Data Tabulation Model, which is developed by CDISC organization. Okay? CDISC organization developed standards. Guys, CDISC is an organization. CDISC is not a standard, okay? CDISC is an organization. That particular organization generated several standards. Okay, so not only SDTM, madam, guys, few more standards are there. Okay, see, to generate the protocol, PRM, protocol representation model, PRM standards are there. Not mandated to follow PRM and all this. So for every document, there will be certain standards will be there, guys. But as SAS programmers work, so we should thorough with only SDTM implementation guide, ADAM and only ADAM, and also dataset model. So these three very important. These two. TLF'S means there will be, this complete training. You can say CDISC SDTM. You may find in internet like this. ADAM TLF'S. Tables, listings, figures. So SDTM ADAM standards, guys, developed by CDISC organization. We are going to discuss first SDTM, next ADAM. SDTM just will convert study data into SAS datasets as per the SDTM standards. Once dataset is ready, did you remember, guys, we discussed ETL process, extraction, transformation, and loading. Same. SDTM is nothing but extraction. Convert your raw study data into input data as per the SDTM standards. Once raw input data is ready, do the analysis as per the SAP document, as per the statistical plan, ADAM. Analysis happen, guys, here. So transformation. Once transformation is done, loading or reporting. What kind of graphs? What kind of tables? What kind of figures we need to create? TLFs. Very beginning I said, right? ETL process. SAS is best ETL too. Same thing only we are going. So our work is related to ETL only, no only na. That is also I said roles and responsibilities of the SAS developer. Either we are going to work with the extraction team, or transformation team, or loading team. So SDTM is nothing but which is equals to extraction team, guys. ADAM, transformation data analysis team, and TLF'S, tables listing figures generating, reporting, or loading team. Okay, so some of the jobs related to SDTM, guys, nowadays. Some of the jobs, most of the jobs, SDTM only. So some of the jobs, ADAM and combination of the SDTM ADAM, if you are good with all, so you can apply for any role, either TLFs or SDTM ADAM and all these.
But sir, we need to have a like full clarity about all three standards, no sir?
Yeah, exactly. As you SAS programmer, you should thorough with all these things. So because initially no need ADAM and TLFs. So for example, as a beginner or 2 years experience guy, so SDTM is enough. You can enter into the field. But later on, you need to learn later also. But if you learn initially only, then it will be easy for us, easy for you, maybe in future. Or else simply you can say, I am familiar with SDTM. So while preparing resume itself, yes, I'm done with SDTM, ADAM and TLFs. I'm very good at all these concepts. Hmm? You will get bit high, right, in the resume. Yes, of course. That's why we are learning all this. You should learn. Every programmer should learn, guys. We don't know which team we are going to work, and sometimes the combination of the work also we are going to perform. So you should learn all these three. When compared with ADAM and TLFs, only SDTM only it will take around 48 domains will be there. So no need to learn all 48 domains. Like procedures around 300 procedures more there. Around 30 procedures we are going to use regularly, right? So 48 and 50 domains will be there. So mostly we are going to use 50% of the domains based on the study. All the domains not required to learn. So regular most used one only we are going to discuss. SDTM. Okay, domain is nothing but data set, guys. How to create the SAS dataset only. We are going to discuss that. So ADAM around 10 to 15 we'll discuss, and TLFs, different tables and graphs, how to create and everything we'll discuss. TLF is nothing but, so ADAM, guys. See, if you observe carefully here, to generate the SAS dataset, we have certain standards called SDTM. To do the analysis, you have document called SAP. Okay, so everywhere you will get some suggestions, you will get some documents based on that only. No need to do anything your manually, right? So once your analysis is done, so you are going to get TLFs, TLF mock cell. That is called template, guys. So these kind of statistics, these kind of tables I want. I will show you if time permits to a TLF document. I will show you these kind of figures I want, these kind of plots I want, these kind of graphs I want. They are going to send. So based on that, we are going to generate the graphs. Based on that, we are going to generate the tables. Got it? Okay. Yeah. So first SDTM. Based on that, next step will be statistical analysis plan. That is nothing but analysis and creating data set approximately as for the analysis requirement. And once analysis data sets is ready, follow by using CDISC organization standards, which is nothing but ADAM. ADAM is nothing but analysis dataset model, and followed by the reporting needs to be done. What kind of reports we need to be done? So based on the TLFs only. Okay. So guys, whereas coming to the SDTM ADAM guides, you can simply download from CDISC organization. These two documents. So of course, you will get from your client. They are going to share with you. But if you are the one individual person, if you want to learn your own, so some of the people through YouTube tutorials, through Google, they are going to learn new technologies. Okay. So in that particular case, yes, this is completely free of cost. Anyone can download from the CDISC website, guys. Simply. CDISC watch. This is an official website. There here you can download. Just click on sign in. If there is no account, you can create account. See here, sign up now. You can simply your mail ID, you will get some verification code. Once verification code is received, so password, confirm the password, what is the name, first name and all. Just agree on terms and conditions and all. That's it. Once you create the profile, missing required element. What is the problem? Which mail ID I created here? Forgot actually. Now you can see I logged in. Standards, you can see here. Just go to the standard tab. There you can see SDTM, SDTM IC, SDTM, SDTM IC, and ADAM. Okay, just click on SDTM. You can see here there is a versions. See here SDTM 2.1, 2.0. So these are very old versions. Now we come to the here 2024. This is the one. What is this? 1 second, guys. SDTM IC. Yeah. So this is the one. SDTM IC. You can see here 3.4 version. This is the one latest version. They released 3.4. So 3.3 and most popular. It's been around 10 years more we are using. So most of the organizations, they are requesting to use 3.2 only, guys. So 3.2, 3.3, 3.4. So you can simply download like this. Once click here. So files and links. That's it. Start download open. So here 2.5 section, there you can find all the data as you can see. So that is nothing but, guys, what and all data we collected, all the data we need to submit in a table format, guys. To create the table, these are the standards we should follow. So how many? See, for example, so first we are going to discuss trial design domain, guys. Trial design. So this is the one based on the protocol document. See here, as for your requirement, how many treatment groups are required for that particular study, trial arms, all the treatment information, all the treatment groups information, we are going to create the table, planned one. And again, trial elements, what and all elements will be there. Elements means, for example, screening, treatment, follow up, all these. Is there any run in? So as for the protocol document, how many elements you planned? What is the order? Okay. So complete elements information we are going to send to this particular data set, guys. And visits. So how many visits you scheduled? Visits information, completely trial visit dataset. Trial summary, complete summary. Nani, one moment, guys. Okay. Yeah. So like this, every domain have unique purpose, guys. Trial information, visits information, inclusion and exclusion criteria information. There is a table. Okay. So disease assessments. There is a table. So complete planned one. The trial arm one is a planned one. So this is the one special purpose domain actually happened one. This is you planned, but as for the plan, not possible to visit the trial site all the participants. Okay, so there will be buffer. So first visit to this particular day. After 1 week, plus or 3 minus days. So plus 3 days or minus 3 days. Minus 3 days means before 3 days, any one day you can visit, or else after 3 days. So buffer will be given. So based on that, you plan one particular day, but when they visit the site, trial site, when element is happen. See here, subject visits and subject elements. Here trial visits and trial elements. So this is actually when it is happened. So here this particular one complete subject information, special purpose domain, demographics. So most commonly used data sets guys, demographics and subject elements and subject visits. These three are popular from this particular special purpose domain. Whereas come to the trial design, so except TD, remaining everything is popular. TA, TE, TV, trial summary and inclusion, exclusion criteria. All these we are going to discuss. Let's come to the interventions. So this is complete subject information. So comes this particular domain, special purpose domain. So I'm not going that much detail SDTM implementation guide. Just for you, I'm explaining here. So detail, once we start SDTM classes there, I will explain. Just what I'm trying to explain is here. So the complete data, what we collected, the data as per these standards, we need to like these kind of table formats, we are going to send to the regulatory guys. Okay. So DM is nothing but demographics data, height, weight, BMI, age, sex, gender, like which kind of ethnicity, race. So when he visited, when if signed the informed consent, when screening started, when doses is taken, complete information like this. Okay. So all these data, this particular domain. So interventions, complete medication information. That particular participant have any prior medication or therapies are using before entering into the study or else during the study. Is there any substances uses, any alcohol habit or any coffee and tea drinking habit are there? So what kind of procedures will be done? So complete medications information and procedures information. Here you can see interventions. Adverse events. Adverse events means side effects, guys. So this is completely medical history. So all this information and findings. Here, this is test information, ECG and vital signs, death details. So like this. So we have separate classes, guys. Separate domains. Each domain unique purpose. So usually we are going to use shortcut names only. For instead of demographics, we'll call DM, TA, SE, SE, CM, CE, EC, EX, PR, like that. So all these domain detailed information, you can see around 400 pages will be there, right? So simply you can navigate 6.1 section, you can find interventions 6.1 onwards and 6.2 events, 6.3 findings, 6.4 findings about and 7th section you can find these data sets and relationship data sets, section 8. So remaining section just introduction only, and what kind of variables will be there. So what is the differences between previous implementation guide and this implementation guide? Okay, so how to derive the variables, what are the standards, and all these. See here, what are the key variables for each data set, each domain, what are the key variables, all the information it will be there. We'll discuss, guys, everything about SDTM. Simply just we discuss. See what I said here. So if you want to download, just sign up your account and just go to the standards, SDTM IZ. There you can see files and links. There you can simply. Okay. So here all the versions will be there. Versions. First, once you open the SDTM IZ, it will display the versions. So mostly, guys, we are using 3.2 only. So don't worry, 3.2 and 3.2, 3.4. So there will be some added domains only. Okay, so existing domains it will keep as it is. So few more data sets they added. So no need to learn. Okay. So what we learned, that is more than enough. SDTM implementation guide 3.2 version, guys. So but you should know differences between 3.2 and 3.3 and 3.4. What are the differences between 3.2 and 3.3. We'll discuss also. We have separate session for that. Okay. So same like that, Adam versions, files and links. You can simply download here. Atomize. And just when compare with the SDTM, you can see just 88 page document only. This is 88 only, not that much. No need to spend that much time and compare with SDTM. 1 second. Okay. But when we start our project, we have all these documents, guys. 1 second, there is a call from baby school. Yes, madam. Hello. Okay, all these documents, guys. So mostly from tomorrow, Thomas, we are going to the SDTM. Tomorrow or day after tomorrow. Tomorrow we'll discuss all documents order-wise one more time. So then we'll start SDTM. Okay. So this is the way, guys. These are all the teams we discussed. In the clinical trials, mostly what and all teams are involved and what kind of documents we are going to get, all the things we discussed. Just I will repeat, guys, 10 to 15 minutes, everything what we discussed, and tomorrow we'll start about SDTM. Okay. Now, see, all stages, study data reported by or processed by SDTM implementation guide and analysis data sets as for the SDTM, ADAM and TLF'S that's been created by, based on the mock cell. All these SDTM implementation based on the SDTM implementation get generated datasets, analysis datasets, and TLFs, tables, listing figures, everything will be submitted for the regulator to the approval, guys. Once the approval is done, if it is a phase 1, for example, then trial can be moved to the phase 2. There will be no change. Usually the same process will be continued. Phase 2 also will be submitted to the regulatory. Once phase 2 approved, then again same process. Phase 3 will be submitted to the regulatory, will be approved. Phase 3 approved, then it's a different step. Sponsor can start manufacturing the drug, which is nothing but production department, and then it goes to the commercial launch, right? And thereby it will become in the retail market. Behind any new drug, guys, before gets launched into the market, usually this is the process, like preclinical studies, phase 1, phase 2, and phase 3. And after phase 3, once approved, commercial launch, then post-marketing surveillance. Like sponsor, followed by the protocol team, should be recruited. So they have to do the document, followed by the CRF design team. They are going to design CRF document, and followed by the site management team. They recruit the participants, and they will create awareness to the public. Whoever is willing to uh, join this that particular study, come forward. They need to check their eligibility. Once the eligibility are confirmed, they will participate in the trial. As for the CRF document, all the data gets recorded and collected. It is going to store in somewhere, like EDEC, electronic data capture. It is going to store in the databases. For all the data, clinical data management team, there will be several people. Again, there is a database related people, data management people, data architecture people, all of them, data entry people, data validation people, mid-level programmers, senior level. There are different teams will be there, should be working in that particular data. So they are going to organize the data. It's organized, okay, from the CRF document to database, guys. Okay, once CDM team work is done, then data analysis team. Again, several people will be working in data analysis team. What will be the several people? Biostatistician, and it's us, nothing but SAS programmers. Well, they will work together. Team of the biostatistician and team of the SAS programmers has to work. All the work gets done. Then it will be forward to the next team, medical writing team. So all the study as for the SDTM, analysis datasets as for the ADM, so TLF'S, and final CSR report, guys. See, what we generated, SDTM datasets and analysis datasets, ADM, AM datasets, and TLF'S, tables, listing figures, everything will send to the next team. What is the next team? Medical writing team. So this is the team, they will write the synopsis based on your analysis, based on your reports. Finally, they are going to generate CSR report. Final report, clinical study report. So then it is going to be submitted, guys. So this is the whole process should be done behind launching the new drug into the market, guys. There will be different phases. All the phases we discussed in detail already. Phase 1, phase 2, phase 3, and preclinical studies. That's nothing but animal clinical studies, all these. Up to this clear, guys, what we discussed, everything. Introduction. Any questions? So tomorrow onwards, we are going to discuss industry workflow. See, I will show you. This one I will update anyway in the drive. So just we discussed all the phases, sponsor, protocols, here of design, site management, and data analysis team, medical writing team. See here, industry workflow. So SAS programmers work in the clinical industry. What and all documents we are going to get, and what is the workflow from where to start, where to end. Okay, so all these SAS programmers work we are going to discuss, guys, and followed by SDTM standards and ADAM standards. There will be different steps. The work need to be carried out in the sequence, guys. There should be a proper manner, systematic manner we should follow. What and all steps are involved to create SDTM datasets, to create ADAM datasets. All the steps and workflow we'll discuss first 30 minutes. So then we'll go to the SDTM implementation, guide, SDTM concepts. Tomorrow onwards, definitely we'll start our SDTM classes, guys, once we discuss industry workflow. See here, same. CRF annotation. So already I said yesterday, SDTM specification, mapping specification, spec document creation, and creating datasets. Analysis datasets and TLF'S. We discussed, right? After creation of the analysis datasets, TLF'S. That's it. Yeah, I will update right now. CRF and the mock cell protocol, SAP. All these I'm going to update in the trial. Okay, guys, let me wind with the session for today. That's all about today. We'll continue tomorrow, guys. Industry workflow. Okay, I don't want to miss the flow tomorrow. Industry workflow we'll discuss first, then we'll start with SDTM. Thank you. See you tomorrow, guys. So last 1 week classes, what we discussed, just I will repeat in just few minutes, like 3 to 5 minutes, then we'll continue the session. So we're discussing in detail about clinical trial process. Whole clinical trial process we discussed along with all the teams gets involved in this particular whole process. And what are the responsibilities of the each and every team? What kind of work gets carried out by each team? And we discussed all these points, right? So let's take all the points briefly one more time. And then we'll move to the next topic, what is going to be industry workflow and how SAS program is going to work with all the documents. What is the first document we are going to start, and everything we'll discuss in detail. Now, sponsor. Sponsor is a team, who funds for the complete clinical trial process. The very first team, once sponsor is ready, he looking for the recruit to the protocol team. So sponsor is the one legally everything he is going to take care about the complete clinical trial, budget and resources and teams involvement and everything. Okay. So next he is going to recruit the protocol team, the team which is responsible for mainly planning for the complete clinical trial process, clinical trial execution, how it should start and how it should end. So that is about protocol team work. Once protocol is approved, protocol team is going to generate the protocol based on your study requirement, all the procedures, what you are going to do, what kind of people you are going to recruit, and what kind of geographies, location people you are recruiting, everything. So we are going to protocol team, they are going to generate the protocol document. Once protocol document is ready, once sent to the regulatory. So while generating the while doing the protocol document, along with the protocol team, there will be biostatistician also. He is going to interfere. Okay, because as for the analysis plan, he will suggest some suggestions. Okay. So once protocol is approved by the regulatory, then sponsor is looking forward to the next team. That is nothing but case report form team to generate the case report. Who design the case report form? So there will be, to generate the case report form, there will be various external vendors nowadays. Or in-house itself, if it is a well-established organization, well-established pharma sector, pharma company, they usually try to maintain all the teams in-house itself. Or I'd say, mid-scale or small scale of organizations, they always look forward for the different vendors to get this particular work done. CRF document. Okay. So, but based on, based on protocol document, CRF is going to be get designed, and case report form will be used by the site management team to do the data collection, collect the data when they are going to recruit the participants, and what is the start date and everything. Site management team, they will take care about the participant, how to enroll and everything based on the protocol, and they are going to check all the criteria, like whether the person coming forward to the voluntarily to the site, and what kind of inclusion criteria, what kind of exclusion criteria should follow to participate in the trial. Once eligibility confirmed, then they are going to enter into the trial. Then, like whatever the data as per the or as per the study requirements should be collected based on your CRF document, right? Or okay, so all the data gets collected by CRF document, and usually next team, clinical data management team. Clinical data management team will be always they are like CRF form, it's like it looks like front end like a form, but back end there will be tables, right? The table of the structures, CDM team mainly responsible for doing the database design, database architect, creating the tables, doing data validation, checking for the data consistency, and everything. Once study data is ready, study data, raw data, metadata, you can call anyway. So then the data analysis team will come to the picture. Data analysis team. So for the protocol document and CRF document and study data, all this become as the input for the statistician. So data analysis team, guys. Protocol, CRF, and raw study data. These three will send to the next team, data analysis team. Okay, so where we find the biostatistician and SAS programmers, this particular team only. So biostatisticians are mainly responsible for doing the statistical analysis and what statistical analysis can be done with this particular data, explaining all the information that will be nothing that SAP document is going to create it. In the previous sessions, all SAP documents and CRF document and protocol document in detail we discussed already, right? So followed by that, TLF mock cells, like preparing like reporting. This will be done by biostatistician, usually mock cells, TF mock cells. Sponsors sometimes they will provide, looking forward for the different vendors to do these particular mock cells. So once mock cells is get prepared, so TLF tables, listing figures, what kind of tables, what kind of plots, what kind of figures to generate based on the analysis. Okay, so all the teams, guys, well established, and when sophisticated organizations, they will try to maintain each and every team in-house itself. Okay, there will be team who is doing for the TLFs. Now coming to the SAS programmers, so study data which is known to submit to the regulatory will be processed by the SAS programmers following the SDTM standards. Whatever the as per the statistician analysis plan requirement, whatever analysis data sets to be created based on the ADAM standards. So the SDTM standards and ADAM standards are developed by CDISC organization, maintained by CDISC organization. As for the TLFs and mock cells, tables, listings, figures will be produced by the SAS programmers. That is the main role of the responsibilities of the SAS programmers. That's what we discussed already. Okay, so this is about SDTM, ADAM, and TLFs. Based on this whole study, data analysis reports and TLF reports, all statistician and medical writing team will be sitting together in order to develop a report called clinical study report. So this is the one, medical writing team. Last, CSR, clinical study report is statistician and some medical writing team. They both will sit together to generate the clinical study report. So all these clinical study report, raw data collected from the patients, and CRF document and protocol document, all these we are going to submit to the regulatory for review and approval purposes. So this is complete clinical trial process we discussed. Now let's talk in industry workflow. Coming to the industry workflow. what is the main job for a SAS programmer? Okay. So as of now, we discussed about teams, and along with the teams, what is your work? We discussed anyways. Here we'll go a bit detail. Okay, whenever you talk about the team, within the team there will be, always there will be hierarchy followed, guys. Like a junior programmers, senior programmers, lead programmers, managers, lead managers, project managers. Like there will be always a certain hierarchy gets followed. So based on the experience, how much exposure do you have for the particular project? Accordingly, the work will be allocated to the programmers, guys. It's not done, complete end-to-end work going with only one programmer. Not like that. Like within the SAS programmers also, within the SAS programmers team also, within the statistician team also, within the CDM team also, protocol team, and CRF design team, whatever we can call as a team. Within each team, there will be different hierarchy based on the experience, guys. How much exposure you have, based on that, they will provide the work to you. Just keep all these things in your mind. Okay. So these are the, see here. Coming to the industry workflow. These are inputs we'll get from the teams, guys. So already what and all teams will be involved, and which team, what kind of document they are going to provide. Already I said, see, from the protocol team, protocol document, and CRF design team, CRF document. From clinical data management team, all the study data or raw data or trial data, whatever you can say. And then we'll get some of the study data specifications, guys. Specification is nothing but, I will explain what is called specification. While discussing CRF document, I think I said, anyways, I am going to explain in detail. Spec document. Some of the spec document and any other supporting document necessary for the understanding the study data and tables. All these documents will say to the SAS programmers and biostatisticians. Because when you work to the different organizations, within each organization, there will be different terminology, guys. Although it remains same, but still, like, based on the study, data will change. So we should just get used to team environment, whichever the team we fall in, and what should be your work. All the things you should know. And all the documents, yes, especially you can see all the documents, these documents you are going to get, guys. And again, within the SAS programmers team also, listen, there will be set of 15 to 20 SAS programmers are going to work, guys. Not necessary every programmer should be provided with all these 5 documents, with all the data. They will not be having complete access with all the documents, okay? Not all the programmers. Say like lead programmers or managers, they will be allocating a task for you. You have to do so and so work. In order to do that particular work, how much input is required? Usually that much input will be shared to you. Don't expect all the documents all the time should be shared. Usually, generally it never happens in the real time. Suppose if you are allocated to, for example, if you are doing with the dataset DM domain, in order to do the DM domain, how much inputs were required? Only that much data is shared with you. That's it. It's not like, the protocol document and CRF document, side data table should be shared with you. We shouldn't expect that one, okay? But usually the managers or lead programmers, they will having complete access. Most of the documents, like all the documents, they will be having access. And as for the work, as for the teams, as for the programmer, the work gets distributed among the other programmers. Like senior programmer, what kind of work they need to do, junior programmers work. Huh? So like the required amount of the input, how much is required to be shared to that particular part, that particular person. Okay. Now let's see what is the sequence of the workflow. Very first work that will be allocated to the SAS programming team is CRF annotation, guys. What is meant by CRF annotation? First work. CRF annotation is nothing but mapping columns as for the SDTM implementation guide. So CRF is nothing but, to collect that data we are going to use, right? CRF document to collect all the patient's information like inclusion criteria, exclusion criteria, so start date, gender, and everything. We discussed about CRF document detail. So each and every column information, each and every value, it is going to store in one of the table in a cell, right? In one column. So which table and what is the column as for the implementation guide? That is called CRF annotation. For example, I will show you. Okay. Let me open SDTM implementation guide. Not updated yet. Okay. Mapping is nothing but, as for SDTM implementation guide, we need to uh, allocate the columns. Which column, which data set, which domain. For example, one moment, guys. Where it is, section 5. DM. Sex variable should be in the DM dataset. Okay. So while doing the annotation for the CRF, this is here like this. We'll do mapping like this. This is for DM domain. Or something like that. Sex variable. Okay. So this is RAM. This is DM domain. DM dot race variable. DM dot age variable. DM dot birth DTC. Like this, based on your values, we need to do the mapping, and it's nothing but just mapping, guys. Like that, like this, all what and all columns will be there. What and all values, which data set and which under which column. Very first job. Okay, CRF annotation. Where we need to start the work is nothing but CRF annotation. CRF annotation is nothing but mapping the main source of the study data based on your SDTM implementation guide. Okay, just map where to store, which table, under which variable. So my inclusion criteria, exclusion criteria need to be stored which variable, like that. Mapping each and every field of data should get submitted through which data set, through which domain, as for the SDTM implementation guide, through what variable. Okay, so this is about CRF annotation. All, so 400-page document, right? Not only one programmer will do all this annotation. So there will be team. Okay, first 10 pages, first one 10 to 20 pages, one programmer, next 20 pages, one programmer, next 15 pages, one programmer, like that. Complete CRF document they are going to annotation as for the SDTM. Okay, every research happening, different different kinds of data going to be collected. As for the SDTM implementation guide, how many data sets we need to create? Okay, so guys, actually, whatever data you are going to collect from your study, definitely you need to submit these dataset format only. Here you have around 48. As for your study, for example, you collected 30 tables data. 30 tables data only we are going to submit to the regulator for the approval. Okay, so like that only. SDTM implementation guide is backbone, guys. So we need to learn each and every important domain thoroughly. For example, if you are good with the DM domain, for example, I'm explaining one project with the DM domain, guys. One DM domain, one project with the TA dataset, one project with TV dataset, concomitant medication, CM dataset. So around 25 datasets we discussed. If you are good with these domains, all the variables and all. So in the DM domain, for example, there will be, let's assume, if I say RF SDTC, start date and time of the study treatment. First exposed of the study. You can see when subject was first exposed to treatment. If I say RF SDTC, once you learn it, so whatever study it is, every study have start date. The date column value only will change. Okay, so remains everything it will be. For example, simple terms I can say that adding 2 numbers, adding 3 numbers, adding 4 numbers. There is no difference, right? If you know the sum, okay, if you know the concept of the addition, it doesn't matter adding 2 numbers or 3 numbers or 10 numbers. Learning the concept of the addition is important. This is all the same, guys. Okay, each subject data, what we are going to collect. So always try to consider yourself as the participant. So what data gets filled while during the screening, all the data it is going to store in table format, right? All these tables of data should be submitted to the regulatory by using specific data set, specific domain. By using our programming terminology, we are going to convert your raw study data into these kind of tables, guys. Domain is nothing but, so everywhere it will say domain, domain, domain. Domain is nothing but data set. Because in implementation, we always use term called domain. So based on which place we are talking, based on where we are using, the name itself different. For example, guys, in the SDTM implementation guide, instead of dataset, we'll call DM, domain. But whereas coming to the SAS terminology, we'll call SAS dataset. Simple example, I can say that in SQL, SAS table we can call just table only. But whereas coming to the base SAS we'll call SAS Dataset, right? 1 second. Okay. In SQL, tables, rows, and columns will call. Whereas coming to the basis programming, variables, observations. But both are same. When we are talking about implementation guide, we always call domain. Consider domain is nothing but, guys. Okay, as first time leaders, when what I'm trying to say here, if you find something anywhere in implementation guide domain, simply just remember like dataset only. It will be, SDTM implementation guide, it will be around a 400 page document, you can see here. So no need to learn all 400 pages, guys. When you are reading this particular guide, if you feel this is something important, if you feel like that, just underline. Hmm? I will show some of the example. Now you can see like this. Just make highlighted specific text based on your importance. This is important to me to remember like that. So what is the main purpose of this document? As I explained before, SDTM implementation guide. So what and all data we are collecting from participants through CRF document, all the data, which table we need to store what column. So by using SDTM implementation guide, we are going to mapping. That is called CRF annotation, guys. So first work, CRF annotation. So in general, as I said already, CRF annotation not a beginner's work. It's a senior level programmer work. CRF annotation. Once CRF annotation is done, see guys, this is the workflow. Without CRF annotation, we are not able to get spec document. Without spec document, you are not able to create a SAS tables. Okay. So then next work is going to be mapping. SDTM mapping specification document, spec document, you can say. Okay. So, spec document we are going to create. For example, already you are going to annotate the CRF document, right? So this field, this is the value for this particular column, this particular table, like that you are going to map, right? So based on that, we are going to create, there is a next team, they are going to create a spec document. I will show you spec document, how it looks like. So this is domains metadata. So each and every domain, how many variables should maintain CDM dataset. This is, we are the one created, guys. This is the one. CDM domain variables, CO domain variables, SC, SV, all, around 46 domains, all the variables, list of the variables here. And each variable table, this is as for the SDTM implementation, this table. Spec document is nothing but like this. For example, this is the data. This is the DM domain spec document, for example. Study ID, is it available in your raw study data or not? If it is yes, it is available. Yes, of course, every study there will be ID, unique ID. What is the domain? So if it is a value, it will be, see here, is it available in the dataset? In dataset. and length. What is the length we need to put? And what is the source table? From which table I need to pick the value for this particular variable, and source variable, from the table which variable, and programming instructions. Okay, like that we'll do the specifications, guys, in spec document. Okay, so for example, RF ENDTC, from where, what is the raw study data? So from the CRF document, what and all data we collected, it will be stored in a table format. I said right here. Like either Excel sheet, database file, or CSV file, some of the table. Which table? What is the column? Here we are going to mention source table and source variable name. Is there any programming instructions? For example, um, mm, for example, there is a race, guys. So, or else I will show you a real spec document instead of doing this one. There you can understand easily. This is raw study data and spec document. DM. You can see here what we discussed. Same thing here is available here. Is this available? Yes. What is the length? What is the source table? DM table. So in the source table, what is the source variable? Subject is the variable. What is the programming instructions? So by using study ID and subject variable, we need to concatenate to get the unique subject ID. Okay. How to get subject ID? So in the DM table, there will be subject. Just open the raw study data. Study data. So in the study data, there will be DM table. You can see DM table. There you can see subject. This is the value for that particular variable. Okay. So this particular information, guys, this spec document, how to do this? As for this spec document, you can perform based on your annotated CRF. With the help of annotated CRF and the study tables, one of the programmer. So this is the one also. Specifications document also, like 6 years or 7 years programmers, they are going to perform, guys. 5 years above programmers only, not junior level programmers. Doing specifications also. Okay. So next RF SDTC. So to get RF SDTC, just pick the data from RAN table first to DT. RAN table. So just go and check your RAN table will be there. In the RAN table first to DT column. So this is your RF SDTC value. Okay, first import the data from RAN table, what and all columns, DM table, what and all columns, all the tables with help of one of the reference variable, either subject or site ID or unique subject ID, any one of the reference variable, just take and combine. After combining, so just set the attributes. What is the length? And what is the label? All these attributes set to that particular data set. That is fine. For example, I will show you one of the sample SAS program here for DM or any other, how we are doing. For example, you can see here, to get this particular DM data set, from DM table, these columns, RAN table, these 3 columns, DS table, these 2 columns, around 5 different tables, guys. 5 different tables, around 12 to 15 columns. 5 tables, each table 2 to 3 columns. All these columns we need to import, we need to merge. So import the data, keep the required variables. Check for the duplicates. Is there any duplicates by subject ID across the datasets from DM and RAND and DS and INCO and TRT? Sort the data by subject and site. And once sort the data, so without sorting, we are not able to do any merges and right? Just merge the data. Okay. And after merging these four, next finally merge TRT also. Finally, raw data set to be created. So this is the program. Attributes. See here attributes. What is the variable and what is the length? So retain, to set the variable order, retain statement. This is these and all attributes for what is the length and what is the label. Retain statement, yes, we should follow particular order only, guys. As for the spec document, as for the SDTM implementation guide, this is the order we need to put. That's why we are using retain statement. These are the labels and length we need to put. By using attribute statement, we can assign the labels and length and everything. Okay, conditional processing. For example, you can see here, there is a condition. If race 1 equals to 1, then race equals to white. If race 1 equals to 2, then race equals to black or Asian African. So this kind of information you can get from the spec document only. Just you can copy and paste. I will show you. See here you can see programming instructions. Are you able to see here programming instructions? If race equals to 1, then race equals to white. If race equals to 2, so like that. Here coming to the arm CD also. There is a condition. If arm CD equals to A, then arm equals to drug A. If arm CD equals to B, then arm equals to drug B. Same data we copied here. See, to get this particular column. Okay. So each and every variable, see here, study ID, we copied. Guys, when it comes to the programming part, no need to assumption yourself. No need to identify any variable your own. Either variable values, either assigned values or corrected values or derived variables only. Derived values only. Okay. Assigned values means subject ID, study ID. That is already defined. No need to do your own. So sponsor is the one going to provide study ID. What kind of domain? Unique subject ID. With help of study ID and subject while concatenating, we'll get unique subject ID. Okay. Same like that, site ID. So site ID you will get from the site variable. Birth date etc. There will be column called birth date. Based on that, you are going to get birth date, birth date. Age. To get the age, no need to no need to set your age by your own. So first date of the treatment and birth date. So both just subtract and do the calculations by 365 days. It will return the age after doing the round. This particular functions and what and all program instructions we are using, there you can see. In spec document it will be there. Where it is age variable, you can see round of RF SDTC and birth DTC plus 1 minus RF SDTC minus birth DTC plus 1. There will be instruction. All the instructions, all the study data, all the spec document they are going to share with you. By using spec document with help of raw data, you need to create a SAS dataset as per your programming instructions. Guys. That is going to be your work. Okay, SDTM mapping. So once mapping is done, so mapping means first annotation, guys. As for the annotation, which table, which column and all. So by doing spec document, and we are going to create the spec. So most of the spec document, it will be in Excel format only. Okay. So once spec document is ready, annotated CRF and spec document is ready, then we are going to create the datasets. Creating datasets. Once the datasets are created, will be shared with the concert team in the form of XPT file, guys. XPT. So this is also very important to know. So we never say datasets as it is like a datasets, guys. We'll convert into XPT file. That is not a big deal. Very easy to convert XPT files as dataset into XPT file. Why, Balu? What is the need of XPT file? XPT file, all the teams, all organizations, or all the how can you say, everywhere may or may not use same operating system, same SAS software. Somewhere they are going to use Enterprise Guide. Somewhere they are going to use SAS Studio. Somewhere they are going to use SAS Studio version. Okay, with to avoid the migration issues, we'll convert into all SAS datasets into XPT files. XPT. That XPT file we are going to share to the regulator guys. Okay, XPT file. So I will show you XPT file how it looks like. So this is XPT file, as you can see. SAS export to transport file, XPT. Just go to the properties and there you can check. .xpt file. Of course, this one open with SAS only. Dataset you are not able to open directly, but XPT file you can open directly. Okay. To avoid the migration issues, we are going to convert into XPT file. So once XPT file is ready, then analysis team, creating analysis datasets. This is also SAS programmers work only. So based on the SAP document, followed by the ADAM guide. Okay, analysis dataset model, ADAM guide, ADAM implementation guide, and with the help of a SAP document, what kind of statistical analysis we need to perform on that particular your study data or raw data. So, as I said right yesterday, so first we'll create the SAS datasets. To create the SAS datasets, we will use reference SDTM implementation guide. With the help of SDTM implementation guide, we are going to create the SAS datasets. And then to do the analysis, we are going to use ADAM guide. Okay. So ADAM guide with SAP document. Once analysis is done, once analysis datasets is created, will be shared with the concerned team in the form of same XPT files only. Okay. So again, once analysis datasets is created, so still reporting not done, right? So here also, after doing analysis, we are going to create the SAS tables only. So now coming to the TLFs, tables listing figures or tables listing graphs, anyway you can call. So based on mock cell, so they will send some mock cells. These kind of graphs are required. So sponsor is the one, guys. Sponsor is the one they are going to sponsor and biostatistician. All these teams will create TLF document. So TLF is nothing but a template, or I can say a mocked, mock cell. Anyway, you can call. In this particular document, it will show you, examples. So this is the table. These kind of minimum values or maximum values. This particular text, this particular color, this particular font. So everything they will mention in that particular mock cell document. What kind of graphs you need to generate? Each graph, what color? Okay. So TLFs. Based on the TLFs, tables, list, and figures will be created for the submission to the concert team. This is guys. This is our going to be SAS programmers work. Okay, so as I said, there will be separate teams, guys. CRF annotation, separate team. Yes, most of the time, CRF annotation creating SDTM datasets, SDTM mapping, spec documents. Senior level programmers work only. Let's come to the creating datasets and all these things. Remaining analysis datasets and everything reporting. So junior level programmers like 1 year programmers, 2 years as just 3 years programmers. This is not a big deal, easy to do, because all the spec documents and raw data, once you have with you, then you can do your programming to create that particular data set. That's what we learn, right, in technical training. Simple. So this is going to be a workflow, guys.
Yeah, Balu, so just I want to give small input. Like currently we are working, so just one point I want to add in that. Particular, can I tell?
Yeah.
Yeah. So we are discussing about the SDTM dataset creation, right? So once that
Mm.
SDTM datasets are created and we are converting into XPT. So we are sharing anyway that XPTs define SDRG to the regulatory authorities. So once the XPT is created, there is a other team, case report tabulation team. They will
Yeah.
validate. They will put that XPTs into zip folder. They will validate in the Pinnacle tool. So that
Yes.
Pinnacle tool will, that Pinnacle tool will identify all the data issues, any data entry issue or something is been defined. So it will identify all the issues so that it will help to improve the quality of the data. So once that is
Not done
done,
yet. Not done yet. I
yeah,
am going to explain Pinnacle and all the things. Yeah. Of course.
yeah,
You are right.
yeah, that data for workflow just I thought of adding this input. So this also we are doing.
There will be presentation. I will show you. Not done yet. Okay. So till Pinnacle, starting with the clinical trial, till submit to the data to the regulatory, what and all documents and what is the flow. There will be picture. I will show you. I say somewhere. One second. I'm going to explain. We'll discuss, Thomas. All the things what you said, everything we'll discuss.
Yeah, sure, sure.
Mm. Okay, that's all about today. Tomorrow onwards, I will go through the SDTM implementation guide. Tomorrow we have session. I will introduce what and all things like what is core, what is the role of the variables, or how many types of the variables will be there. Okay, SDTM implementation guide, we'll start learning from the tomorrow, guys. So we are done with all the introducing classes. So as you said, still left with only so complete flow from one team to one team to what and all documents. So finally, what is the validation tool we are going to use while submitting data to the regulatory? What and all documents we are going to send to the regulatory? I saved somewhere. Oh, anyways, I will explain tomorrow if time permits. Okay, done. Thank you, guys. That's all about today. So today we are going to start with the SDTM workflow and SDTM guidelines. What and all guidelines we should follow as per the SDTM. Everything will discuss. So before going with that, I want to complete flow from till from starting of the study to submit the document to the regulatory till approval. What and all phases will be there? I will mention here. Okay, so just 10 minutes. Then we'll go to the 10 to 20 minutes. We'll discuss SDTM workflow. Then we'll move to the SDTM guidelines. Okay, now very fast. As you know, once study start, then what and all documents we are going to prepare, review documents. Come on, guys. What is the first document? Protocol and case report form, CSR, design team, CSR. Then data management team, they are going to collect the data, raw data sets. Okay. Then what is the next document? As for analysis requirement, the analysis team, they are going to prepare SAP document. All these documents we are going to get from your sponsor. Okay, so listen carefully. First, once study start, these are the documents we are going to prepare. Of course, there is a team, there is a certain hierarchy. Based on the hierarchy, all these documents is ready. Once all these documents is ready, what is the first step we discussed? What is the SAS programmer's first work? Yesterday we discussed.
So annotation.
Annotation. Create a CRF. Annotated CRF or CRF annotation. Anyway you can call it. Okay, once the documents is ready, so we are going to start with annotation. Annotation is nothing but just mapping. What and all data you collected from the participants as for the CRF document, as for the SDTM guidelines, as for the SDTM standards, as for the SDTM compliance, which all these values, which table, under which variable value it is. So just annotation, CRF annotation. Once annotation is done, once annotation is done, this is going to be validated by your senior level programmer or else team lead. Okay, so annotation is right or wrong, so there will be validation. Always there is a validation team, guys, everywhere. So team lead, he's the one going to validate. And who is going to annotation? As I said already, so most of the cases, senior level programmers, 5 years, 3 years or 5 years above experienced programmers only, they are going to do the annotation. Once annotation is done, CRF annotation, once you created a CRF and validated by team leader, then what is the next thing? SDTM mapping, right? SDTM mapping specifications. Okay, we are going to create mapping spec, SDTM spec document. What is this spec document? Already yesterday we discussed about spec document. Spec document is nothing but, in SDTM guide there is a table, right? As for that particular table, with help of your raw data and answered CRF, which table under which column, how to derive. See guys, some of the variables derived variables, some of the variables collected variables, some of the variables will get by default. Sponsor is the one, he's the one going to provide directly, like subject ID, sponsor ID. So some of them, so we never enter values manually your own, guys. Either we are going to enter the values by collected based on during the study, collected values, or else assigned values, or else derived values. With help of few variables, some of them variables we are going to derive. Okay, so by using RF STDTC, RF ENDTC, and all these study days and duration, all these variables we are going to create, we'll discuss. Okay, mapping. First, once annotation is done, mapping. We'll map. Okay, so each table, how many columns will be there? Each column, what is the value? The column value is available from which table? From the raw data. Okay, definitely it will be there in the raw data only. Okay, 60 to 70% of the values it will be available in the raw data. Maybe in the raw data the column name will be different, but the column name we need to convert as for the SDTM implementation guide. but value only same, but column names only, names and attributes, labels and everything as per SDTM guide we need to change. So complete instructions, complete specifications, we are going to create this particular spec document. And based on this particular spec document, you will come to know how many data sets need to create for this particular study. Okay, so you have around 40 to 40, 45 to 50 data sets. Is it, I am going to create 50 data sets? No, not like that. As per your study requirement, as per your correction of the data, based on that. So once annotation is done, based on the annotation, and you are going to come to know once you create the spec document, okay, these many data sets I need to create, like that. 1 second, there is a call. Okay, SDTM mappings, specification Excel once ready, then you will come to know programming plan, guys. Next thing comes to the picture is, so once annotated CRF and spec document, raw data such as SAP document is ready, the next thing comes to the picture is programming plan. Okay, as for the spec document, you will come to know, right, how many data sets need to create. So all data sets, not only one programmer is going to create, okay? So there will be, senior level programmers, junior level programmer, inter level programmer, beginners. Based on that, they are going to split the data. Based on that, they are going to split the tables. As for the tables required data, also they are going to share. Okay? So we'll come up with the programming plan. For example, there is a 28 data sets to create around that particular organization. 15 SAS programmers are working. So based on their experience, for example, for DM dataset for one particular programmer, DM and comments dataset, both combination of the datasets one programmer, concomitant medication, interventions, all interventions related data one programmer, trial design one programmer. Like that, they are going to split. They are going to make a plan, programming plan. So he's going to make this programming plan. So the team lead RHR, any one of the senior level programmer, they are going to do this particular programming plan. Once programming plan is ready, they are going to split work along with the documents. Then you are going to start your, then you are going to use your SAS programming skills. Once you start your, see here, that's the reason why we learned already SAS software. Okay. So to create all these data sets nowadays, it's been around 60 years more. We are using from raw data sets to create SAS data set. As for the SDTM implementation guide, we are using SAS tool only, SAS programmers only. Maybe in future, next 3 to 5 years. So along with SAS, they are planning to do the R programming also. With help of R also, they are trying to create the SAS data sets and all. So some of the organizations nowadays also they are expecting. So if you have R skills along with SAS programming, you may get the job very easily when compared with only SAS programming skills. But not all the organizations. Few of them are, guys. Few organizations are expecting along with SAS R as well, but not mandatory, as I said. If you learn, that is added advantage. Like simply, I can say that if you have certification credentials with you, there is a person there is no certification, you have certification with you. Definitely the first priority going to be you only, na? Certification is nothing but valid proof for your SAS skills. Certification gives you confidence, right? So here like that, SAS programming we are going to use here. 1 second. Nani evaru? Okay. Okay, so by using your SAS programming skills with the help of SAS software, we are going to create what we are going to create, guys. So by using spec document, by using annotated CRF with the help of raw data, what we are going to create? We are going to create SDTM and ADAM standards or compliance datasets. Am I right? So first SDTM datasets we are going to create with the help of raw data by using SDTM implementation guide. So just that doesn't yield any report? No. So first we'll create just tables only with help of SDTM, guys. What kind of analysis is required based on SAP document analysis datasets model? We are going to use ADAM implementation guide. So finally, by using SDTM and ADAM, we are going first SDTM, next ADAM. Then as for the SDTM ADAM standards, we are going to create SAS datasets. Then, so we'll never send SAS dataset as it is to the regulatory. So these SAS datasets, we are going to convert like an XPT file. We are going to create export. XPT is nothing but export. XPT files. Like all the data like dm.xpt, cm.xpt, ta.xpt, sv.xpt. Once create a SAS dataset, within a moment we are going to create XPT. What is the use of XPT? To avoid the migration issues. Okay, to open with any environment and any Windows, any operating system. Simply I can say that. To open anywhere. Okay, there we are going to create XPT file. This is mandatory, guys. Even if you check your SDTM implementation guide, every domain under there will be, it will display like dm.xpt, cm.xpt, co.xpt, tv.xpt like that. Okay. So we are going to create XPT files. Once XPT files are ready with all these data sets, then there is a validation tool. Okay. So validation tool, they are going to use Pinnacle 21. There is separate team. Of course, might be SAS programmers also. Nowadays they are checking. So validation tool, guys. So as for the SDTM implementation guide, ADAM implementation guide is everything is all the reports what we generated, it's clear or not. See, for example, if it is a one table, if you take a reference, for example, SDTM implementation guide, there is a domain called DM. DM dataset, if you want to create a records, observations, per subject, per record, guys. Each subject, there will be one record. So there is a SV dataset. Every visit, one record. So for example, one participant, one subject entered 6 different visits. 6 records should be there. 101, one record, 101, second visit, 103, sorry, 101, third record, 101, fourth, 101, 5, like that. Only one participant, 6 visits happen. For 6 visits, 6 different records should be there. And there is elements. Elements means screening, running, treatment, and follow-up. There is a flow. So based on the element, each element one record. So like that, we have some standards, right? The particular standards you are following or not. So this is the one validation tool. All the values are as per the SDTM implementation guide, created or not. Pinnacle 21, there is a tool, validation tool we are going to use. Not done yet. Give me a moment. I will drop my picture code. 1 minute. Okay. Then finally, we are going to create final report called define.xml, guys. Okay, so define.xml is the one final report we are going to submit to the regulatory for the approval purpose. With all these documents, along with all these documents, we are going to create final report define.xml. Submit study. Okay, starting from here we are going to submit. This is these and all things will happen. All the documents, all the all the team's work. But guys, so to design the protocol and case report form and all these, we are not involving. So there will be protocol team, CRF design team, and data management team. They are going to collect the data sets, and the SAP statistician is the one take care about the SAP document, and combination of the sponsor and statistician investigator. All the teams, they are going to take care about these documents. But once documents are ready, you are the one need to take care about the SDTM standards datasets and Adam standard datasets, SAS programmers. Okay, and create annotated annotated CRF making and spec documentation creation. What is the programming plan you have to split your work? So once split by using SDTM implementation guide, Adam implementation guide, how to create SAS datasets. So once datasets is ready, we are going to convert into XPT files. Then by using validation tool, we are going to validate. Then final report is going to create. Then we are going to submit that particular data to the regulator. Okay, if you approve, so if you prove your efficacy and safety of the drug, then okay, then they are going to, the same thing will happen for all the phases, phase 1, phase 2, phase 3. So if they approve, then okay, fine. Otherwise, you need to fit this particular trial. It is going to be waste of money. Otherwise, if they recommended some revisions, you need to go for the revisions. Okay, see, out of 100% of the trials, guys, 10 to 20% of the drugs only going to be approved, not 100%. You can Google it. Okay. So this is what we discussed, all the documents, the teams, and everything. Anything else here? Any questions, please? Thomas, is this right? Anything we need to modify here?
No, Balu. Yeah, right.
Okay, then now coming to the SDTM guidelines, guys. There it is. I will keep maintain desktop this one instead of going all the time. Yeah. So coming to the SDTM, SDTM means Standard Data Tabulation Model. Here, I will. DTM. Study data. Study data only now. We are going to convert like a standard. Study data tabulation model. SDTM. So name itself telling study data how to convert a particular table model. See here. Data that we have collected during the study to be stored with proper integrity and as per the standards. Now, coming to the SDTM versions, how to download? I said there are many versions, guys. 3.1, 3.2, 3.3, 3.4, latest version 3.4. I think they have released in 2022 or something. But most popular one, and all of the organizations, they are using 3.2. Guys, versions, you're the one not going to decide. Sponsor is the one, or else organization, they will guide you. Hey, this is the version you need to use to create this particular data set domain. Okay? So sponsor's choice or organization choice. Any version they are going to, they can use. But this is the one most popular and it's been using from the ages. 3.2 version. Few organizations only nowadays they are using latest version 3.4. But as I said, not that much difference, guys. With additional features, with additional tables, they have updated old version with the latest version. Okay, so existing tables will be there with additional tables, either 2 or 3 tables or 1 or 2 tables. So nowadays there are a lot of trials and there are a lot of data they are going to collect, right? So what and all data we are collecting from this trial study, definitely we need to submit the data. So maybe, 48 data sets may be not enough to submit the data, or else within the 48 data sets, that particular data to submit, there is no way, there is no chance. For example, guys, each and every data set, there is a unique purpose. Visits information in one data set, adverse event information in one data set, elements information in one data set, complete patient information in one data set. Each and every data set unique purpose. Okay, all that particular unique purpose may be not enough to send complete clinical trial study data to the regulatory. Such cases, what they are going to do is every 3 to 4 years, they are going to update the versions with the new tables. Okay, so whatever version it is, almost it will be same only. Okay, they will not change, columns, names, and attributes and all. It will be the same. So we are going to learn 3.2 version, guys. So 3.2 version, as for the 3.2 version, these are the data sets we are going to create by using SDTM standards, SDTM standard domain models. Okay, so today just I will go with one model, one domain will go. What and all key sections will be there, we'll see. See guys here, if you want to create a SDTM domain data set, there will be, see here, dm.xpt file, and that too, what kind of version it is, and what is the structure of this particular table. Table or domain or dataset, all are same. Domain is nothing but dataset. I said yesterday, one record per one subject. If you want to create a DM dataset, each subject one record only should be there. In case if you have any duplicates, that is completely invalid. Of course, there is a validation time. They are going to validate. If there is any duplicates, they are going to remove the duplicates. We are the one while doing the programming also, every time we are going to check duplicates. If we have duplicates, based on the duplicates, we need to do something else to remove the duplicates. Some of the options we need to put extra. Or else to derive the sequence variables, we need to go with first dot and last dot. In case if we have same dates, two different dates are there for one participant, that is first date comes under the starting date, last date comes under the end date. So based on the first dot concept and last dot concept, we are going to do. So we'll discuss later. But so in case sometimes, as I said, examples already I said, right? One for element per subject, for widget per subject, like that it will return. Each element one record like that. Now you can see here variable name. So if you check all the variables as per the SDTM implementation guide, it will be below 8 characters names only, guys. Yeah. Complete all the data sets what we are going to discuss, all the variable names, 8 are below 8 characters names only. That too always we need to display in capsulator formats. And again, so as for the SDTM implementation guide, each and every domain name should be 2 characters long only. Just abbreviation, call it 2 characters. So concomitant medication CM, trial visits TV, subject visits SV, exposure EX. So like that, if you want to create a final dataset, the dataset name should be just a 2 characters abbreviation only. So first thing, variables, 8 characters or below 8 characters. All the variables from all the tables, all the domains. And whereas coming to the label, you will come to know, guys, all the variables, attributes, you will come to know. But remember, so dataset name should be 2 characters, just abbreviation. And again, variable names, 8 characters or below 8 characters names only. All are capsulators. Now come to the variable label. So every variable there is a label. Well, because USUBJID. So how we can understand what is this particular column name? As per the SDTM implementation guide, they may understand what is use subject ID. Okay. So we need some proper label for each and every variable because we have limitations, right? Each and every variable we have 8 characters names only, below 8 characters, up to 8 characters only you can use for the variable name, not more than that. So what kind of variable it is? Labels. Labels gives you always detailed description about your column names, right? Yes, labels. Labels also 40 characters mostly, guys, not more than that. That to prop case. All labels should be 40 characters, up to 40 characters only we need to put labels, variable labels. So these are important questions. Just keep in mind. Every domain, if you want to create a data set, for example, once your DM program is ready, if you want to create a final dataset, the dataset name should be DM only, 2 characters DM only. And after creating a SAS dataset, as I said, we are going to convert into dm.xpt. DM means demographics. Okay. So variables, variable name, variable label. And again, you can see here. So ID, so domain, use subject, USUBG ID and RF ST So here we have some code names, guys. DTC means what, ST means what, RF means what. It will be there somewhere at the end of your SDTM implementation guide. Each and every code values it will display. You can see here. ADJ means adjustment. AD means analysis data set. For example, ST. Start. For start, we are going to use ST. See here, ST, start. And DTC. DTC means date, time in character format. Where it is, D variables. Here it is. DTC. Date, time in character variables. Okay, ET element, EM emergent, OBG object, like that each and every one. So no need to worry about all the variables, guys. All the variables is like very simple only, na. So ST means start, DTC means date, time, character format, RF means reference. Huh? For example, where it is RF, REF, here RF or REF both will be reference. Huh? REAS reason. So shortcut names only we are going to use everywhere. Okay, so each shortcut code, what will be the description? Here you can see fragment, keywords, and each keyword fragment. Around 383 page it will be there. Okay, see here, RF means reference, END means end, T means DTC means date and time in character format. EN and DTC means date and time character. RF reference, start date, ST start, DTC date and time in character format. So in complete SAS programming, all the date columns we are going to send as a character variables only. And that too, we should follow some control terminology. The control terminology is nothing but ISO 8601. About ISO 8601 control terminology, we have separate class. There I will explain. ISO 8601, almost all the dates it will display like, YYMMDD only, guys. First year, next month, next day. Okay, that is the way it is going to display. ISO 860 format means. So we are going with one by one, right? First variable name we are discussing. Here variable names, how it will display. So shortcut names. Okay, you see here RFX STNDTC, RFX ENDTC, RFIC DTC. IC means informed consent. Reference informed consent, date and time in character format. DTH DTC, death, time, date and time in character format. DTH FL, FL means flag, death flag. Yes or no? Site ID, INV ID. INV means investigator. ID means identifier. So like that. So every day we are going to discuss, guys, same repeated variables only, na. There you can easily remember. No need to by heart blindly, guys. What is called FL, what is called DTC, and everything. Okay. So similar values only it will be there most of the domains. Okay. So like this, ASU units, and all this. This is about variables, guys. Now variable labels up to 40 characters, and we should maintain the prop case for the labels. Prop case means you know that, right? First word, first character in the word, capital letter, remaining everything small letter. Mostly I'm going to use prop case. And next type, what type of variable it is? Is this character variable or numeric variable? And these two type of the variable we have, right, in the computer science. Yes, it will display either character or numeric type. Up to this, okay, we are familiar. Names, labels, and types. Whereas coming to the control terminology, some of the variables we should follow control terminology, guys. Control terminology. Control terminology means while submitting the data, as you like, you are not able to submit the values. For example, here, few of the variables, see here, DTHFL, death flag, there is a control terminal. If you click on this particular link, it will go to that particular website, CDISC organization website. There will be, there is a document called SDTM terminology. In that particular terminology, it will display this particular variable value. If you want to fill, either you can use yes or null. Okay, or else come to the age units. Age units, if you want to submit through regulatory by using three variable, either years or months or days, like that only we need to submit. Not like as you wish. We should follow some control terminology. I will show you. You can download this one also from CDISC website itself. Yes, you can see here. If you want to submit age unit CDISC submission values, either days or months or weeks or years, this is the only format you can submit. These are the values. These are the terminology values you should follow. Nothing else. Okay, what is the CDISC submission value? The control terminology. We have separate document. This is the one I will share with you. All the terminology, what and all we need to follow. Everything it will be available in this particular document, guys. So this is not created one, just download one only. You can download from the CDISC website. How to download, I will explain later. Okay. See, if you find any domain after the variable, this kind of hyperlink, that means not like your ways. We should follow some SDTM standards. Control terminology we need to follow, submit that.
Well, and also that control terminology for each version we have different year of control terminology, I guess. Like 3.2 means some 2018 or something, some date of control term.
Yeah, based on the versions they are updating terminology values also, Thomas.
Yeah, yes.
Okay, so as per the 3.2, 3.2 only we are going to use. If it is 3.3, based on 3.3, you can download from the CDISC official website. Now, so see guys, some of the variables there is no control terminal. That means you can use any number. Okay, see here, empty, empty, empty. And ISO 8601 means I will show you ISO 8601. Maybe next session I will explain ISO 8601 detail. It will take 30 minutes class. And blue color means definitely we should follow terminology document, and there will be star somewhere. You can see star means maybe in future you can expect a control terminology for this particular variable. Right now there is no terminology. Star means you can expect based on the versions, maybe coming version after 3.2 version, maybe in the 3.3 version, maybe it will be there for control terminology for this particular variable. Star means you can expect, but this version, for this case, there is no control terminology. Okay. See here, star indicates variable may be subject to control terminology. It is displaying, you can see at the end. Star indicates variable may be subject to control terminology. Okay. This is about control terminology and formats, what and all terminology we need to follow, what and all formats we need to follow while submitting this particular variable data. Next, role, notes, and core. I will just explain brief here. So each and every role we'll discuss in detail tomorrow session, next session. Tomorrow session in the sense, next session. Role. What kind of role it is? We have 5 different type of role variables, guys. Identifier variables, timing variables, topic variables, rule variables, and qualifier variables. Under qualifier variables, again, record qualifier, synonym qualifier, result qualifier, variable qualifier, grouping qualifier. Okay, so based on that particular variable role, what kind of role it is. So just to understand only, we never display what kind of variable it is in the data set. Okay, so only for the interview purpose, only for the understanding purpose, what kind of variable it is this topic variable. Topic variable is the main variable for every domain. Okay, so like that topic variable or else identifier variables. Identifier variables will help you to identify the distinct values of the dataset, distinct values for the records. See, these are the identifier variables: study ID, domain, unique subject ID, identifier to identification, to identify the distinctness of the dataset. Distinct values means non-repeated values. Okay, and topic variable. Topic variable means, see here, this is the main topic variable because based on this topic variable only we are going to create dataset per subject per record, right? So subject ID. So each subject ID one record only. So this is the main topic variable. About what is called topic variable, what is called identifier variables and record variables, I will explain in the next session, not now. So about role, we need to discuss. Not done yet. And coming to the CDISC notes. CDISC notes just notes description about this particular column, how to derive. So for example, if you want to derive unique subject ID, concatenation of study ID, site ID, subject ID. So if you with the dash we need to concatenate. Then you will get unique subject identifier value. Okay. For example, to get RF SDTC, how to get RF SDTC? So this is the collected date only. Record qualifier means recorded one. No need to derive this one. Okay. So recorded one. So during the screening, we are going to record, right? Date and all. So every date is going to be collected, guys. Once you enter into the clinical trial site, so while signing the informed consent during the screening, once start the actual trial in the sense study treatment, study drug, main drug, which drug we are doing clinical trial, that is called study drug. Study drug treatment once you expose, that is first date of the exposed study drug. So this is the one. RFSDDC. This is most popular variable. Every domain we are going to use this variable to derive different variables. RFSDTC means reference start date and time in character format. When subject was first exposed to the study treatment, first treatment start date, and that means RFSDTC. I'm not teaching about the variables here, just explaining notes. Notes will give you complete information about how to derive that particular variable. Okay. So like this, RFA and DTC. So first doses of the exposure to any protocol specified treatment, RFXST DTC. Yes, guys, there is a lot of confusion between RFST DTC, RFXST DTC. There is only X only difference, but two different variables. This is study treatment starting date. This is any protocol specified treatment. In the protocol, there will be different treatment also we are going to mention. Not only study drug. To prove the efficacy of the study drug, we are going to use another drugs also. Okay, for example, there is already existing drug you are using. It is curing in 30 minutes, but your drug is curing in 20 minutes. You can prove your efficacy, right? So for that to prove, we are going to use existing some of the FDA approved drugs also. We are going to mention the protocol document. That particular drug, if you expose first time, that is called RFXTDTC. Study drug exposure date, RFSTDTC. We'll discuss. So this is about notes, guys. Notes will give you complete information. And remember, what and all notes and this complete table is available for all the domains. Each and every domain, this kind of table will be there, guys. The tables we created in Excel format, guys, for easy for all the students, to make specification documents easily. This is the one. All the domains. See, same table I will show you here. For example, you want to go with the DM, right? This is the table. Same table. Variable name, label, type, control terminology, role, CDISC notes, and code. From the PDF to Excel file. For easy mapping, for easy specifications to mention here. So here, variable name, label, type, role, notes, and code. This is from SDTM guide on. So here you can simply do the mapping. All domains, SC domain, SV domain, CM domain, all 48. And coming to the core, guys. Core means, what type of variable it is. This is role, what kind of role it is playing. Core means, is this required variable, or expected variable, or permissible variable? Three different types of core variables, guys. 1 second, guys. Yeah, core. Core is coming to the three three cores, guys, mostly required variables, expected variables, and permissible variables. So here, let me explain in a simple way. Required variable means that variable presence is mandatory. Variable should be there. Value also we need to put. Null value is not available. Required variables means variable presence mandatory. Null values are not available. Variable also should be there, and value also we need to put that particular table. And expected variables. Variable presence is mandatory, but values, you can use null values. In case if you collected the values, just assign the values. If values are there, if there is no value, just use missing value. But variable we need to put, definitely. Variable presence is mandatory in the dataset, expected. Whereas coming to the permissible variable, these variables you can skip. If you no need, not mandatory. Presence is not mandatory. If you collect the data in that particular case, just mention with values, just assign this particular column with value. If you are not collected, no need to mention even with null values also. No need at all. Just you can ignore permissible variable. Okay. So whenever you want to create the data set, guys, first we are going to apply the filter. How many of them required variables are there? Next, how many of them expected variables are there? I need to mention. I need to process for this particular data set. Then last, we'll check for the permissible variables. Okay. While creating the program, I will explain anyways one more time. So these three are the core, guys. Requested, expected, required, expected, and permissible. Required means variable value, both should be there. Expected, variable should be there. Values or null values, both are available. But permissible, not mandatory. Variable presence is not mandatory. If you collect the data, then only we need to put the value. Otherwise, just this is just introduction only about core variables and role variables with the notes. I will explain in the next session. All the SDTM definitions, guys. Done? That's all about today, guys. Continue the session Tuesday only. As we discussed Saturday session, next class will be Tuesday. Okay? Thank you, guys. That's all about today. Where we are? So SDTM workflow and SDTM definitions we are discussing here, or SDTM guidelines, I can say that. Study data tabulation model. So what and all data we are collecting from the clinical trial, that particular study data, as per the SDTM standards, we need to standardize and we need to organize. Then that particular data, we are going to send it to the regulatory. So as for the next procedures things. Now coming to the SDTM guidelines, guys, here, as I said, we have 3.1 version, 3.2, 3.3, 3.4. There are many versions, but most popular version of 3.4, sorry, 3.2 we are using nowadays. So as I said, there is not that much difference between the versions and versions. A few more added domains you can identify when compared with 3.1 and 3.2. For example, I will show you. So here, there is a version 3.1 version. This is 3.1 models, domains. These are the domains. See, you can see here around 35 domains only, not more than that. Whereas coming to the 3.1 version, now I will go to the 3.2 version. You can see here the difference between both. Special purpose domain, you have 4 domains here. Special purpose domain only 4. Okay, fine. Whereas coming to the interventions, here you have 5 domains, right? Concomitant medication, exposure as a collected exposure, and substance uses and procedures. Here you can see, you can find only 3 in 3.1 version. Concomitant medication, exposure, and substance uses. Procedures and EC. Exposures as collected. PR and EC is not there. Right? PR and EC. when compared with 3.1 version, they have updated in 3.2 version, 2 more domains they have added. This particular prior information also it will display relationship to prior CDISC documents. Here you can see, just go through. See here, guys. So here is coming to the interventions, exposure as collected and procedures. These new domains when compared with 3.1 version. Yes, added right here. And let us come into the events. Based on the versions, guys, they are going to add few more domains, nothing else. Okay. So here, let us come into the next one, interventions. So interventions done. Now coming to the events. Events, you can see 6 domains are there in 3.2 version. 5. One domain added. Which domain it is added? I think where it is prior to. Which one? We're talking about interventions, right? Yeah, interventions. No. Events. See, HO dataset got appended. So HO dataset got added to the 3.2 version. Okay, so just go and check here. See here. So 3.1 version 5 domains only, and 3.6 version, 3.2 version 6. Here there is a 5 domains right here, 3.1 version, and one domain added, healthcare and counters HO domain. Like that, each and every classes, observation classes, few more domains added. Here you can see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and around 22 domains will be there in the findings. Here not 22, around 12 or 14, around. I think 12 only we have here. 10 domains got added for the new version 3.2. So like that, based on the versions, they will add few domains, but a lot of changes when compared with 3.1. We have 3.2, guys. Okay, it's a lot of domains they have added. When compared with here, around 30 domains will be there, I think. Below 30 only, but when it's coming to the 3.2 version, we have 48 and 50 domains will be there. Okay, so but once learn 3.2 version, that is enough to work in industries, guys. Any project. What is the difference between previous version and next version? It's same. They have mentioned in the SDTM implementation guide 1.3 section. Okay, so this is about the versions. So we are going to learn 3.2 version next. Few points to discuss. As for SDTM, each domain name should be 2 characters only. That is called abbreviation. Okay, each and every domain, each and every data set or else domain, only 2 characters. 2 characters long only. Okay, either adverse event, substance usage, medical history, demographics. The name itself we are going to call, but when we are going to create a SAS dataset, just 2 only. Example. See here. Adverse events AE. Why it is not copying? Demographics. substance. Right, substance use means, substances. What are the substances using the particular participant? Like, like coffee, alcohol, or cigarettes. Is there any substance usages are there for participating subjects? All the information they are going to collect and save in this particular data set called SU. Okay, concomitant medication, CO. Like that, every domain, 2 characters names only, as per the SDTM. And again, and every domain should have proper labels also. How many characters labels we can use max?
42?
40.
40.
Yeah, up to 40 characters. And prop case only. Okay, and variable names should not exceed 8 characters. Wrong. Up to 8 characters only, maximum. Okay, examples. Use subject USUBJ ID RF SDTC reference start date and time in character format. Like this. SUSTDTC substance uses start date and time in character format. Like that, so not all the time 7 characters, 8 characters. Sometimes, for example, subject ID SUBJ ID 6 characters or 5 characters. That is count the sex variable, just sex only directly. Age only. So these are the variables, but all the variable names should not exceed 8 characters, and all 8 characters should be in caps only. Okay, uppercase letters only, all the variable names. And while creating the datasets, SDTM datasets, guys, so we should follow the order, same like SDTM implementation guide only. Variable order should be as per SDTM IC. Okay, and for example, concomitant medication. Here you can see first subject ID, next domain, next unique subject ID, next CM sequence. This is the order we need to put. Okay, not like as you want to set the order. So what is the order they have mentioned in SDTM implementation guide? Same order we need to go through. Okay, so this is about order. Whereas coming to the, as I said, control terminology and ISO 8601 format. See, control terminology, there is ISO 8601. So about control terminology, we discussed there is a control terminology document will be there, SDTM terminology. So few of them variables, we should follow the terminology. So based on the document. So if there is no terminology, the value should be anything you can use. Okay, values can be anything if there is no control terminology. But if you have control terminology like blue color text permalink, if it is highlighting, that means we have control terminology for these variables. Okay, so for example, there is a dose form, either tablet or lotion or else syrup, whatever it is, the form, there will be control terminology. We should follow that particular control terminology while assigning the values. Frequency. So twice a day, daily twice, or every 12 hours, or per day. So for example, if it is a daily twice, there is a value we need to put BID. Every 12 hours, Q12H. So these terminology values we can find in this particular SDTM terminology. See that particular document I'm going to update in the drive, SDTM terminology. Copy. You can download it from website, guys. Okay, so this terminology, while doing the programming itself, there I will discuss again. Whereas coming to the ISO 8601, guys, this is very important to know because we learned a lot of formats and informats in while doing the technical training. Informats and formats, date dot week date, DMM DDMM by MM by what date and all. But whereas coming to the clinical sector, in all the domains, wherever there is a date variable, so date variable should be character variable, guys. All date variables in clinical, especially all the domains, what and all domains we are going to discuss as written Adam and all the data variables should be in character format only, guys. So while discussing technical training, date values, definitely we need to read as a numeric variable, then only we can do the mathematical operations we discussed, right? Yes, of course, that is true. So here also, sometimes we are going to convert character variable to numeric variable whenever there is a calculation required comes to the picture. Calculation, there is a requirement, calculation. So by using one date to another date, you want to do some calculations, then we are going to convert that variable into numeric variable. But again, while submission, once you want to finish the data set, the variable, finally, we need to convert into character variable. Character only. Okay. So I will mention here. All dates in SDTM will be stored as character data type variables. And again, all dates not only date and time should store ISO 8601 format only. What is this ISO 8601 format? We'll discuss, guys. We'll spend time here because you should learn this particular concept. So this format is nothing but YY MM DD. First year, next month, next day. If it is a only date, along with the date, if we have time also, then including time. T, guys. T for time reference. Time indicates. Okay, time designer, whatever you can say. T. T we are going to use after the T. Hours, HH, colon modifier, MM, colon modifier, SS. I will give examples. There you will come to know. Okay, if it is a only year, only date, YY, MM, DD. Date with time if you want to display in that particular column, YY, MM, DD. And in between day and month, usually, we are going to use colon modifier, right? Date and time column, but ISO 8601 format, not colon modifier. So date and time between, we need to put the T. T means followed by this particular date. This is the time. Okay, to indicate. So few of the characters we are going to use here while going with ISO 8601 format. Hyphen. two scenarios we are going to use, guys, to separate year, month, to separate year from month from day. So year, month, and day. Between we are going to use dash only. And again, one more use is also there, guys. This particular icon to indicate, to represent missing date component. Date component in sense, either if you have missing year, missing month, missing day, there also we are going to use hyphen character. I will give examples for that. Let me explain what and all characters we are going to use. Next, colon modifier, you know that colon. To separate hour, minute, hour from minute from seconds. Okay. Next, this is one slash. This one to separate date and time intervals. Yeah, of course, Thomas. I will update. Sure. Okay, guys. One moment. Let me drop my kids. Okay, slash we are going to use in case there is a 12 p.m. to 12:30 between this particular date. In case if we have date and time to separate intervals, we are going to use. I will give examples also. Next, P is the one we are going to use, guys. P means period. Okay, to represent duration. Okay, so these characters. We'll go with examples. For example, if this is the date, January, for example, 07 Jan 2025. Okay, this date if you want to display like ISO 8601 format, as for the format, 2025 dash month, right? 01 dash 07. Okay, so in case if you have only month and year, there is no day. January 2025. Then if you want to represent 2025 dash 01. Okay, and again. And day and time, date and time in case if you have. This is the date and time, for example, 15, 25, 30. Okay, So as for ISO 8601 format, if you want to display date and time, so remember I said right, followed by T we are going, this is the format exactly. Just apply 2025-01-07 T right, followed by the T time. So 15 colon modifier, 25 colon modifier, and 30. Okay, as for ISO 860 format, date and time if you want to display, this is the way. So date, dashes, and time colon modifier between the T. And again. So here also, in case if you miss something, for example, there is no seconds, we have only time, hour and minutes in case if you have. Then this is common. 2025-01 and after this, like this, you can skip. Okay, if you have only hours without any seconds, then just T15. Okay, even if you miss month, even if you miss day, so you can skip like this. Or if you miss hours also, yeah, it is fine. Next, whereas coming to the slash, how to use slash. Okay, we'll go with missing value. Hyphen, I said right, we are going to represent to missing date component values. Date component means, for example, if you have missing, for example, January 2025. So for example, this is the date and time. Here, for example, this is missing. Don't know. How minutes? Don't know. Okay, so to indicate this kind of date, this kind of value. There, instead of minutes, just put dash. If you find dash, guys, don't confuse. That means they are not collected that particular date. Sorry, that particular time or minutes or seconds. Okay, and next. 25th. For example, like this, in case if they have collected data, 25th of some month in 2024. So then, what is the meaning of this one? Month is not there, so but day is there, year is there. Such cases, 2024. Come on, guys, anyone of you tell me how to indicate month missing here? 2024.
Then double hyphen.
Double hyphen.
Yeah, 25.
No.
Hyphen 25.
Hyphen 25. Exactly. Okay, one hyphen indicates one missing. These two are common, right? Between month and year to separate. Got it? In case if you have, for example, 5th month, guys, 05, remains everything same. Here 0 instead of month, just dash on. Alright. Are you able to understand, guys, where we are going to use dash to indicate missing? Okay. For example, 8:30 of some unknown date. Then, how many dashes you are going to use? To indicate time, okay. T 8:30. 8:30 you are going to use. Here date is missing. To indicate days, years, month, and day. First year indicate missing. Next dash. Month indicate missing. Next dash. Next dash for. So guys, this is for year. This is for month. This is for day. Between two dashes is common. Every month and year and day, it will separate the dashes, right? So total 5 dashes. 5 dashes T 830. And one more thing, guys. Here, SAS will not allows AM PM in time values. Not SAS, SDTM guide. Okay, as for SDTM terminology, AM PM concept is completely not available in SDTM implementation guide. Okay, our minutes 24 hours format only we are going to use. Are you able to understand here where we are? We are discussing about hyphen. So to indicate year, month, day, as well as to indicate missing values also, we are going to use. Okay, I will ask you then. January 5th in 2025. And 15, I don't know minute. Sorry, I have minutes. 30, and I don't know seconds. So to indicate this,
2025 hyphen
2025 hyphen
01 hyphen 05 T15 colon
15
colon 30 colon dash.
Yes. Wonderful. That's it. Got it? This concept, are you able to understand, guys? This concept where we need to put the dash. If you find the dash, that means what is the meaning? So there is no value for that particular either month or day or year or hour or minute or seconds. This is one scenario. Next, coming to the duration. Coming to this character. So colon, you know that to separate only hour, minute, seconds only, we are going to use colon symbol. Coming to the slash, where we are going to use. So coming to the slash, as I said, duration, to calculate the duration, to display the duration, to indicate, to represent duration. For example, between 11 and 11:45 on the morning, January 5th, 2025. Okay. So between 11 to 11:45 on the morning, January 5th, 2025. To indicate this kind of between, okay, duration. Sorry, not duration, like interval of the dates, right? So here, guys, first, as usual, first year 2025 slash 01 slash 05 month, sorry, 05 day. Then just give your time. What is the time? 11. 11 slash 11 to 11:45, right? So duration, just give slash, then as usual 11:45 after the slash. Okay, just put 45. Are you able to understand what scenario we are going to use? If you find any between, between this year to this year, this time to this time, this is this date to this date, if you find. Okay, for example, between 1st and 10th of April 2024. Now 2024 04 01 slash 2024 04 slash
10.
10. That's it. One second, guys. Okay, where we are? So like this, between we are discussing, right? So like this, either if you have time to time as usual, followed by the T, just use the time with including minutes and seconds if you want assign just display. Or else if you have date to date between, in case if it is displaying, just use slash between the date and date. Okay. So what is remaining? Coming to the duration, P, to indicate P, to indicate duration. So this is also very important. For example, guys, especially during calculated, during collection of the, for example, it's how many years it's been you are using this particular substance, alcohol or cigarettes or coffee. So duration, there will be duration variable common for the most of the domains. Duration, DUR, the variable based on the domain. So duration, for example, if they say from it's been around 2 years, okay. So not no need to mention. Don't follow directly 2 years, guys. Some of the variables we should follow ISO 8601 format duration. As for the ISO 8601 formats, if you want to display the duration, you know, P to Y. P means period. 2 years. Years Y. Days D. Weeks W. Month M. Okay, I will show you one of the example here. For example, CM only, I think CM duration it will be there. CM DOS, CM form, category, X and DC. You can see here duration. Duration of the treatment ISO 8601 format. Exposure domain. So from what is the duration you are using this particular medication, that particular treatment or therapy? It's been how many years or how many months, how many days, how many weeks? Duration, if you want to display, definitely you should follow ISO 8601 format. As for ISO 8601 format, if you want to display the duration for that particular column, if it is a years, for example, 10 weeks. P common, guys. Whenever you want to display the duration, P means period. Period, 10 weeks. W. 15 days. Uh, 6 hours. 45 minutes. Okay. This kind of days, hours, and minutes. P common. Whenever you want to indicate duration, P comma, P, 15 days, right? 15, D. So you want to indicate time. So definitely we need to follow time. So T common guys here. Okay. After the date, if you want to display the T, either duration or else here, whatever it is, the column. So T, we need to put T. Let's come to the time, 6 hours, right? 6 H, 45, 45 to indicate minutes. Just M only, guys. Okay, 15 days times 6 hours, 45 minutes. Okay, tell me, for example, 5 months, 25 days. How we are going to display as per ISO 8601 format? 5 months, 25 days. Come on, guys. P?
Yeah, P5M. For days like same, no? P25D.
No need continuation P. Just 25D is enough.
Okay.
Okay, P indicates period. Okay, months and days and year continuously. For example, you have 2 years. Next 2 and 1. Okay, 5 months, 25 days, 2 years. Or else if you have years first, definitely we need to put years only now first. 2y, 5 month, for example here. 2 years, 5 months and 25 days. P2y, 5mn, 5m, 25 days, 25 days. No confusion, right? I will ask you. Just for 3 days. P?
P3D.
P3D is enough. Okay? Like this. And again, 1.5 hour. Half hour, 30 minutes. To indicate 30 minutes, usually we are going to use time, right? Directly P time 0.1 by 2, right? Half an hour means 0.5 hours, guys. 0.5. 1 by 2, which is equal to 0.5 hours. 1.5 hour. Okay. So 5 days.
But 0.5 is like half an hour, right?
No, 0.5 is half an hour. Yes, half an hour only I am talking about.
Yeah.
1.5 hour. So 1 hour, not like 1 hour 30 minutes. I mean 30 minutes. I mean it. Okay. So in case if they mention directly 30 minutes, then PT 30 m you can use 1/2 hour. So if they mention like that, then only. If it is a direct 30 minutes directly you can use. Or else, for example, 10 days, 12 1 by 4 hours. So 4 hours means quarter, or around 50 minutes. 1 by 4 means. How to indicate? Or else, not like this. Yeah. 12 and 1 by 4 hours. P 10D followed by T 12 point 1 by 4, which is equals to 12 point
1.5.
2.5, not 1.5.
2.5.
2.5. 100 by 4, 25, right? So 2.5. 0.25, like this. So here, for example, 20 days.
Well, 0.25 hertz should be there, right?
Oh yeah, of course. This is hours, right? We want to display like hours. That's why H. If it is a minutes, 30M we are going to use. We want to I didn't see yesterday class. One second.
Sir, actually one doubt. 1.5 hour, right?
One moment. 1.5 hour no, means 1/2 hour. 1/2 hour, not 1.5 hour. 1/2 hour. I'll simply half hour. Okay. If it is half hour, then how to display? So PT 0.5. It's the last one. 20 days. 10 years, 15 days, 15 months, 9 months, 25 days, 18 hours, 45 minutes, 30 seconds. Come on, guys, how to display?
Yeah, P10Y,
Mm,
9M,
mm,
25D,
mm,
T,
T.
18H,
18H,
45M,
45M.
30S.
Okay, this is called ISO 8601 format, guys. Depends. If it's a direct date, in case if you have directly, you can mention DD Y by MMDD. Date and time in case if that particular column is available. Guys, always there is a spec document. Based on spec document only we are going to display. Okay, so like this. So in that particular column, there will be duration is there, or else intervals are there. If it is an interval, this is the way. Duration. So I will mention here like this, or else. If it is duration. Duration example. Intervals. Missing date and time components. This is directly original record. Date and time recorded. Guys, all this information which is it is available in your implementation guide also. So files, save as, desktop, this PC, SDTM. Not done yet, guys. We started, okay? So there are a lot of things to discuss. SDTM guidelines save. Okay, just today we discussed the control terminology ISO 8601 only, and about role and core we are not done yet. Tomorrow we are going to discuss what is the meaning of the core and what type of core variables will be there, what type of role variables will be there. core and role tomorrow we are going to discuss, guys. Then you will come to know about this particular table structure of the SDTM implementation guide domains. Okay, then that's all about today, guys. We'll continue tomorrow. I will update video. Thank you, guys. Okay, guys, if you take any domain in SDTM implementation guide, so you can this kind of table with all variable list labels and whatever variable and all these things it will display. Okay, so as of now what we discussed, I will repeat one moment. SDTM guidelines. Whereas coming to the SDTM implementation guide, there will be, first is variable name, variable label. So variable name, every variable name, 8 characters or up to 8 characters only. So not more than that. Labels, up to 40 characters labels. Next, type. What type of variable it is? Is this character variable or numeric variable? So under type, it will display either character or numeric. So you know that type, only we have two types of variables. One is type character variable and numeric variable. So these three we are familiar. And whereas coming to the control terms or format. That's right, control terms and format also we discussed. So whereas coming to the format, we have only one format, which is nothing but ISO 8601 format. ISO 8601 format date values, how it looks like, and everything we discussed. So some of the variables, it will follow control terminology. So what is the terminology we should follow? There will be Excel sheet, Excel document is there. Based on that, we are going to follow. So they will size, guys. Simply you can go, just if you click on this particular link, it will go to the website. There also you can download. So this particular SDTM terminology document, no need to download. Already I have downloaded and updated in the drive. So control terminology and formats. So these four variables as of now we discussed. Now what else remaining? Role and CDISC notes and core. Role, CDISC notes and core. These three are also very important to know. Okay, so what type of role variability is? Topic variable or identifier variable or record variable or group of grouping variable, synonym qualifier. So we have different types of roles, guys. Okay, roles I will explain. These two are small topics. These two topics, series notes and core. Okay, So coming to the CDISC notes, the CDISC notes will help you to how to derive the variable. That variable is derived variable provided by the sponsor, or else recorded variable, or else with any variable reference you are going to calculate. For example, guys, some of the variables no need to derive. Directly we are going to get the values. In complete SDTM programming, guys, we never mention the values for the variable by your own. Either assigned values or else derived values only. Okay? Assigned values are derived values or recorded values. So based on the notes, you can easily identify. So for example, so there is a unique subject ID, guys. SUBJ ID. Example. This particular variable, if you want to derive, so this is derived variable. Okay, with the help of few variables we need to derive. So what and all variables you need to put to derive this particular unique subject ID? Their CDISC notes will define detailed information. Concatenation of subject ID, site ID, and one more variable is there. Study ID. What study it is? You can see unique subject ID, concatenation of. See, notes only giving all this information. Concatenation of study ID, site ID, and subject ID. Yes, well, because same site, multiple trials may conduct. Same site. Or same participant. He is participating in the two different studies. Let's assume like that. Okay, for example, if I go with only subject ID, just a subject ID, okay, there will be a lot of confusion, guys. Along with subject ID, which side he is participating and which study. Study ID and the site ID and subject ID, if you concatenate, this is unique subject ID. Okay, so like this, just I'm giving example like this, how to get the variable values. Okay, see here. So study ID, unique identifier for a study. Yes, every study there will be unique identifier study ID. So they will provide. This is provided by sponsor only. Domain, 2 characters abbreviation for the domain. So just which domain it is. If it is demographics DM, subject widgets SV, subject elements SC, like that. Subject ID, you can see subject identifier which must be unique within the study of an ID of subject as recorded on CRF. So this is a recorded variable. On CRF document, it will be there. Of course, in spec document, they are going to mention. And next, RFSTDTC. When subject was first exposed to the study treatment. Okay? Yes. Like this, CDISC notes will help you to how to get that particular variable value. Is this recorded variable or as derived variable? If you want to derive, what and all variables, existing variables you need to put all these. Okay. So this is about CDISC notes, guys. CDISC notes. Every day we are going to use CDISC notes to derive the variables. Next, coming to the role. Role means core. Role I will explain at the end. Let me finish first core. So this can be categorized into three types. Actually, three types of core variables. Based on the core only, we are going to create SAS dataset. Okay. So first one is, REQ, required. Second one is expected. Third one is PR, permissible. These three different core variables will be there. Score is very important. Required variables must be included within the domain. These variables must be included in the domain, or else you can say dataset. One second, guys. Same idea only. These variables must be included in the domain. Yes, required variables definitely we need to mention. So otherwise there is no meaning of that particular dataset, guys. Okay. So, and that too, these variables cannot have null values. Null means missing. Yep. Example. So guys, what and all required variable they mentioned based on that particular domain, all the variables should be there in the data set. So required variables gives you, meaning of that particular data set. For example, let's say, so there is a demographics data set. Demographics data set, there will be variable called subject ID. Subject ID. So this is required variable actually. So if you skip this particular variable, if you mention, like sex variable, age variable, race variable, ethnicity, ethnic. So whatever data it is, if it is, this is required variable. If you skip, so age, which person age it is, which person race it is, which person gender it is. How can you identify? Huh? So this is the one gives you, yes, this particular subject ID. So if you have subject ID, the particular ID will correlate all the variables. Yes, 1001 participant, sex, this is gender, this is age, this is race, it is ethnic, join date, end date, start date, whatever it is column. Am I right? So required variables, that's why we need to include the variables in that data set. At the same time, we should not miss the values. The variables should have values only, required variables, guys. Okay, so study ID, study ID, example, study ID, unique subject ID, and EC sequence, EM sequence. There are so many sequence related variables, any sequence related variables. Like if I use 2-s, 2-s means, for example, if it is a CM domain, CM sequence. If it is a EM domain, EM sequence. If it is a EC domain, EC sequence. EX sequence. So 2-s indicates domain name, guys. Don't confuse all these. So simple examples. Okay. So domain, every domain have some required variables. Those variables very first we need to include, guys. See, if you want me to show you, see here. Unique subject ID, required variable. Study ID, yes, of course, required. Domain, required. Subject ID, required. Remaining all expected and something permissible and all. But you can see sex is required. Again, arm series, what kind of treatment it is, what kind of treatment group it is. Okay, we'll discuss later all this. Okay, but anyways, what is the core? 3 different cores will be there. One is required, another one is expected, another one is permissible variable. Coming to the expected variables. These variables also, guys, expected variables also must be included in the domain. But the difference is between, one moment. Okay, these variables also we need to include in the dataset while creating the dataset. And yes, expected, guys. So missing values allowed. Okay, so expected means yes, variable should be there. In case variable value is corrected, then okay, we need to put the value. Otherwise, you can set the missing. But variable presence is mandatory. Okay, coming to the examples. For example, guys, there is an adverse event dataset, adverse event domain, AE. In the particular data, there is a column called DTH, DTC. Death, date, and time in character format. Okay, if there is an adverse event, unexpected data. So may or may not. Okay, all the participants may affect with adverse event is nothing but side effects. Okay, some of the participants only will affect with side adverse event. Some may not, right? So even if it is affected with adverse event, if that particular participant is died, then we'll fill that particular information in the DTH DTC. Death, date and time in character format. Otherwise, you can simply put it as a missing. But because this is expected, we don't know either that a particular subject died or not. If it is a died, then only we are going to count the data. If he is not died, that is just missing. Column will be there. Got it? Expected. You can see RF RF STDTC, RF XSTDTC, RF ENDTC, all these are expected variables. RF STDTC. So first exposure treatment date. Okay, if they collected, okay, fine. The data will be there. If they miss that particular data, so that is going to be missing. Okay, so expected variables can have null values. Expected name itself telling, right? Just we can expect the value. If value is there, just we can fill. Otherwise, you can skip. You can ignore. Next, permissible variable. These variables not mandatory. I'll simply I can say that these variables are depends on study or else sponsor. So based on the study or else based on sponsor. If sponsor is say, hey, these variables data we collected, just include in the domain. Okay. So as for sponsor requirement, if they want to include the variable, then. But as for the SDTM standards, these variables no need to include, guys. If you have collected the data, such cases, okay, if sponsor decided or collected data, then include. Else, we can skip them. Okay. Sponsor. Please do ignore my spelling mistakes if you find. So here, guys, missing values also allowed. You can set missing also for these variables. Examples: investigator ID, INV ID, or else let's see, investigator name. So every domain have few of them are permissible variables, guys. So these are okay, and that to see here. So we discussed the three different core variables, right? Required variables, expected variables, permissible variables. Required variables are mandatory. Presence is mandatory. We should not allow null values. Expected variables must include the variables, but you can use the missing values. If data is correct, data is available, okay, then otherwise you can skip. Let us come to the permissible variables. These variables depends on the study or sponsor. If the data is corrected or sponsor decide to include that particular variable, then we are going to include, right? So these three. See, while creating the data set also based on your spec document, very first, guys, see here, For example, I will go through one spec document. One second. So there you can see PM specifications document, concomitant medication dataset document. So here these are the variable list. Okay. So this table is from SDTM implementation guide only, guys. You can see variable name, variable label, type, control terms, and roles, CDISC notes, and core. So these are assigned by programmers. So specifications. I said right, spec document we are going to create. Senior level programmers, they are going to generate the spec document based on your annotated CRF, which table. So to see, for example, to create this particular variable, from which variable you need to pick the input data, which table, or what is the column name. All this information they have mentioned in the spec document. So you are going to get spec document and raw study data. Based on that, you are as per by using your SAS programming skills, you are going to create a domain. That's what I said, right? So this is spec document. For example, you can see here in dataset. Yes, no. What is this yes and no? Means to include dataset or not. See, very first we are going to check how many of the variable I need to skip. Just after the filter, if you select no, all the no. So no means no need to include that particular variable in the dataset. That too, data is not collected. Okay, in dataset no. If you select OK, now you can see all the no variables. Is it permissible variable or not? Permissible variable, then you can simply ignore. No need to bother about that particular variable. See here, all these are permissible variables only. No need to include. No need to include. Data are not collected at the same time. Permissible variable. Permissible variable, even if you skip, not a problem. But whereas coming to the yes, these variables we need to include. See, that two permissible variable, CMINDC, okay, permissible variable, but there you can see they have collected data, so we need to include. So permissible variable, CM category, permissible variable, but the data is there, so we need to include. So in dataset, yes, these variables only we are going to derive. These variables only we are going to create while creating the dataset. Guys, only dataset include variables only, not permissible variable. That to no variables. Okay, so based on dataset only we are going to identify filter based on not core. Okay, core after the dataset, if it is no, is it permissible variable or not? Am I able to skip or not? Then you are going to check the core. So first dataset only. In dataset, I need to include or not. Clear? So this is about core, guys. Am I clear? Okay, once we start programming, there you will come to know in detail. But anyways, not that much difficult to understand core concept, which is very easy. Next, role, guys. So CDISC notes and variable name, variable label, what type of variable it is, control terms, all these we are done with all this, and core also. So what is remaining? Role is remaining. Let me go through role. Role also very important to know. Okay, during the interviews, they may ask what type of role this variable is. Is this record qualifier, or topic variable, or is timing variable, or identifier variable, or else for DM domain, what is the topic variable, or SC domain, what and all identifier variables. If they ask, So until and unless if you don't know what is called identifier variables and record variables and timing variables, topic variable, we are not able to answer, right? So you should thorough with all these role of the variables, guys. So SDTM IZ define role for each variable for all the domains. Okay. So what and all variables are there? What and all role will be there? Identifier variables, I will explain guys one by one. Identifier variables and topic variables and timing variables and rule variables qualifier variables under qualifier variables. There are 5 more variables, guys. Record qualifier, result qualifier, grouping qualifier, synonym qualifier, variable qualifier. Yeah. So these are the variables: identifier variables, topic variables, timing variables, rule variable, qualifier variables. Qualifier variables again 5 types. All these information, guys, you can find in SDTM implementation guide. Where to check, I will explain. I will show you later. But coming to the identifier variables first. What are the variables? Identifier variables. See, guys, here you can see these are. See here, role, record variable, record qualifier, or variable qualifier, synonym qualifier, or topic variable, identifier variable. You can see. So on what basis we are going to categorize into identifier variables or topic variable or record variable? That's what we are discussing here. So coming to the identifier variables. So one moment, there is a call. Okay, coming to the identifier variables. Yes, identifier variables, guys, name itself telling to identify which study it is, which subject it is. Okay, so these variables use it to describe the values of an observation. Or else I can say that. One second again. So these variables gives you identification, enough, identification information. One domain can have one or more identifier variables. Okay, examples: SUBJ ID, subject ID, study ID, site ID, domain. All these guys, all these are you can say identifier variables, which will give you identification information. Through these variables we are going to display. Okay, and again topic variables, very important. So topic variable will decide the nature of the dataset, what kind of dataset it is, how to record the these variables gives you structure of the dataset, guys. Simply I can say that. And every domain can contain only one topic variable. Simply remember, topic variable, every domain can contain only one topic variable. Topic variable gives you, or else topic variable focus focus on the meaning of the dataset. Okay, for example, every domain have one topic variable only, as I said. See, so demographics, DM dataset. So completely to submit subject information only, guys. Complete subject information. Subject age, subject gender, subject demographics location, subject ethnicity, and when he entered into the study. So when he started the study, informed consent date and start treatment date, which group it is, which treatment group, all the information about the subject. So as subject information is, we need to submit through DM dataset. Subject ID is the topic variable. SUBJ ID. This is topic variable. For example, concomitant medication. Okay, other than study drug, if any participant are taking any other medications. For example, guys, nowadays medication is common, right? Some of the people, they are going to use some illness, based on the illness. So if any participant are using any medication other than study drug, so what and all medications they are using, what and all therapies they are using, what and all tablets or syrups, whatever it is, that complete concomitant medication other than study drug, any other medication if they are using, that particular data we need to submit through CM domain, CM dataset. So then Your topic variable going to be CM TRT. Okay, CM treatment, concomitant medication treatment. What is that particular treatment? What is the therapy? What is the medication they are using? Like that. Okay. So for example, TV trial visit. All the visits information we are going to submit by using TV dataset. All planned protocol defined visits information. Okay. So visit is your topic variable. Okay, simply I can, that's why I'm saying topic variable will decide the nature of the dataset or will focus on the entire meaning of the dataset. Okay, every domain has unique purpose, right? To send death details, disposition domain, to send adverse event AE domain, to elements information, subject elements domain. So widgets information, widgets domain, procedures information, procedures domain. So like that we have every domain has unique purpose in the SDTM implementation guide, and every domain has one topic variable. Okay, topic variable very important to know. Next, rule variables are timing variables. Timing variables describes the timing of an observation. Okay, if any variable data collected time, guys. Okay, so time means that can be start date or end date, or else mostly all the domains it will contain SDTC and ENDTC. SDTC means start date and time in character format. ENDTC means end date and time in character format. For example, if you started medication this particular date, what is the medication? Starting date, ending date. Treatment starting date, ending date. Substance usages, okay, starting date, ending date. Element start date, end date. So that particular column, start date and end date columns, we can call timing variables, guys. Okay, date values and time values it will display. Okay, timing variables. Examples: SDTC, ENDTC. Start date, end date. See here, date and time of collection, timing variable. DMDY, timing variable. Okay, so like that. Some of the DTC variable, guys, not all DTC variable. Some of the DTC variable timing variable, some of them are like record qualifier. For example, I will explain record qualifier later. Where we are coming to the rule variables. Record qualifier means, guys, while entering the study, so while signing the informed consent, they are going to collect your dates. When you signed, that is recorded date, right? When you signed informed consent, that is informed consent date, they are going to record. That is called record variable. Okay, if you derive, that is one kind of variable. So now coming to the rule variable. So name itself telling rules, guys. Every few of the domains, especially trial elements, trial widgets. Okay, if you want to enter into the first widget, you need to finish informed consent form, or you need to finish the screening, then only you can enter. This is start rule, this is end rule. Okay, so every TE dataset and TV dataset, elements and widgets. So these kind of domains, mostly we have some of the rule variables, guys. So these variables provides start rule and end rule information. Okay, examples. If it is a TV dataset, TV STRL start rule and TV EN RL. Okay, ST indicates start, RL indicates rule, EN indicates end, RL indicates. If it's a TV, T, guys, all these domains we are going to discuss, no worries. Okay, just examples. T STRL, T EN RL. So rule variables. Now coming to the qualifier variables. First, record qualifier. Okay, recording or getting or taking measurements. Okay, either if you record or if you collect or if you take measurements from the participant or any other drugs information, that is called a record qualifier. Examples, there's an example you can easily understand. For example, birth date. How can we define birth date? They are going to explain, right? BRTHDTC, birth date and time and character format. Age. So based on few variables, we are going to calculate age variable and sex variable. All these record. Then, so guys, like this, we have a record qualifier and result qualifier, grouping qualifier, and synonym qualifier. So as of now, we discussed up to record qualifier. Remaining variables we'll discuss tomorrow. Then we'll start open SDTM implementation guide and what and all important points we need to know. So we'll underline. Then we'll start our programming classes, guys. Done. So that's all about today. We'll continue tomorrow. Thank you, guys. So we are discussing about role variables about SDTM implementation guide. What kind of role variables will be there in each domain? Okay, not mandatory, guys. All type of variables should be there in a single domain. Okay, either 2 or 3 combination of the variables, either identifier variables and topic variable. Every domain must have topic variable and identifier variables and timing variables, guys. Rule variable, some of the domains they can have rule variable and record qualifier and all. So depends, but these 3. So especially these 2, topic variables and identifier variables, are most common for all the domains. What and all domains and datasets are available in that particular SDTM implementation guide. Okay, so identifier variables for identification purpose and topic variables and nature of the dataset. What purpose the dataset we are creating? So to focus main nature, enter meaning of the dataset, topic of the topic variables we are going to use. Okay, every domain, every dataset, there is a unique purpose. So what purpose you are creating that particular table? The purpose of the dataset. Okay, so topic variables it will reflect. And timing variables to describe the time. Okay, either start date or end date or whatever. So timing variables it will display the time along with the date also. And rule variables, some of the rules we are going to follow. Okay, and so if you have any rule related variables, there we can call rule variables. And rule variables few domains only you can identify, not that much. Like especially trial designs, protocol defined treatments and elements, and which is what and all will be there. That we are going to use rule variables. What is the start rule before? What is the start rule to start the treatment? What is the end rule to end this treatment? What and all things you need to finish and everything. Start rule and rule variables we are going to use, and the qualifier variables. Qualifier variables based on the uh, based on the type. There are different qualifier variables will be there. Record qualifier and grouping qualifier, synonym qualifier, variable qualifier. What else? Result qualifier. So record qualifier to record while getting uh, during the screening itself. Very first, guys, screening, right? To check the participant eligibility by using inclusion and exclusion criteria, signing the informed consent. All these very first, right? Okay, so there we will collect some of the uh, information from the participant based on the study. Okay, so like is there any substances uses? Some of these studies, guys, they are going to check substances uses information, also they are going to collect from the participant. Okay, so substances uses may impact on that particular study drug. Okay, but not all the studies. Okay, but not all the studies. So if study requires, they are going to collect the data and they are going to create data set called SU dataset substance uses. If, for example, kids related any syrup or any medicine, any therapy they have identified. If it is a kids means it's like drinking coffee, tea, or alcoholic. So not possible, right? So they will not do that particular. They will. You can skip that particular data set. Okay. So depends based on the study what kind of data we need to collect. So collecting the information called record verifier, record qualifier. Okay. So record qualifier, under record qualifier variables, we are going to assign the correct information. So next to result qualifier, name itself telling results. Okay, lab results or test results or any other results related variables, results variable you can say. These variables contains results. Examples, LB OR RES. Okay, lab original results. OR means original, RES results. Shortcut names, the code. What is the code? What is the element and element description? So we have an implementation guide. There also we are going to discuss. And yes, like this, we have some of the original results, vital signs, originals like that, VSOI. So these are the result qualifier. We have so many, as we are not started yet SDTM implementation guide domains and all. So if I explain something that is completely new for you, okay. So I'm not going that much detail. So just concentrate on how many types of the variables are there. Core variables and row variables. So both are very important. Every programmer should know. Okay. So result qualifier and ROUPING grouping qualifier. So certain group, specific records. For example, there is a treat, there is a therapy for this cancer. So cancer related therapies, how many, or else cancer related medications, how many of them you are using? For example, there is a disease. For that, you are using some medications. Okay, how many types of the medications? For example, two different diseases are there. So, for example, For diabetes, you are using some kind of medications, and cancer, you are using some kind of medications. So all the medications, so maybe 3 medications or 5 medications comes under one category only, one therapy only. For remaining everything, one category. Okay, category, subcategory. Okay, so combination of the records, if it is comes under one category, that is called grouping, guys. Group the observations together. Okay, multiple observations. If it is a grouping, to display the value, grouping qualifier. Set of observations based on category. Okay, so grouping qualifiers, examples. Any category variables, guys. CMCAT, EXCAT, okay, ECCAT, like that. Subcategory. Instead of domain name, what I'm going to do is I will use Datasets. Any category and subcategory. Related variables comes under grouping variables. You can say grouping variables. Next, synonym qualifier. Similar names, guys. Synonym qualifier is nothing but synonym means same name, same meaning, right? Okay. So some of the variables, so while collecting the data, so they will give same simple names, but whereas coming to the synonym qualifier in this particular variable, so related name, is there any scientific name based on the dictionary? So based on the medical dictionary or from MEDRA or from WHO. Okay, what is the detailed name? For example, every plant, there will be scientific name, right? Usually we are going to call one name, but there will be scientific names for each and every plant. Same like that. So if you use any term, okay, so as for your convenience, if you use, for example, fever, fever, if it is a variable, as for the synonym qualifier, next variable, definitely every variable, every topic variable, there will be synonym qualifier variables will be there. Okay, so in that particular variable, some medical dictionary name, pyroxia, like that you are going to mention for fever, as for the MEDRA. Okay, so medical dictionary. So based on that particular scientific name. So guys, renowned means similar name with a detailed description. Simply, I can say that. Okay, similar meaning. Variables. Example. For example, guys, element. Element, for example, if it is an element is SCR. Element code, etc. Complete name, SCREING, screening. Or else, AE term. AE term, if it is a fever. AE dictionary related code, ADCOD. As I said, pyroxia. Okay, similar meaning, guys. Simply I can say that cinnamon qualifier. Okay. Next, last, I think, variable qualifiers. Guys, variable qualifiers means like specific. How can I say? There is a dose. How many doses units? So there is a variable called dose, guys. So for example, 20 or 30 or 40. What is that particular 20 and 30 and 40? Units. Dosage units. Okay. So used to describe the some of the variable values, guys. Simply I can say that. Okay. These can be used to describe the value of an observation. Okay. So variables with units. Example. Dose U. Dosage units. There will be dose column, guys. So dose it will display just number. 10 or 20 or 1 or 2. So dosage units like mg for ml or else ng for nl, whatever, or grams, whatever unit it is. So units information, this particular variable it will display variable qualifier. Okay, so I was saying like that age units. Age. For example, like ISO 86201 format, if it is a 10 years, P10Y. So Y indicates years. So age, in the age, we are going to assign only 10 only. But age units, 10 years or 10 days or 10 weeks or 10 months. So units, we should follow some control terminology while assigning the units. All units, guys, there will be control terminology. Based on the control terminology, we are going to follow. Okay, so like that. So any variable, if it is describing with units, simple, I can say that. So with values, so units, guys. So variable qualifier. So these are comes under qualifier variables. So record to record, result, any results, some related records, some category records. If it is displaying good through specific variable, that is grouping variable. Synonym qualifier with the same name. If it is displaying detailed description, synonym qualifier, variable qualifier. So variable description. So these are the variables comes under qualifier variables. One moment. So where we are? Yeah, we discussed about variables. Now, see guys, this is SDTM implementation guide, study data tablets model implementation guide, human clinical trials version 3.2. Okay, so most of the modifications when compared with previous versions, they did with version 3.2 only. I said many times. That's why most of the organizations still they are using 3.2 version only with their projects. Okay, so which version you need to use? So your company may going to be decided, guys, not you. But whatever version it is. So if you want to create a domain or dataset by using implementation guide, you should thorough with implementation guide, guys. What and all? See, for example, here. Always I am going to say same thing, but this is very important. See here how many domains are there in SDTM your implementation guide IC 3.2. So coming to the special purpose domain 4, interventions 5, 9, here 6, 15, findings 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21. 15, 21, 36, 2, 38, 38, 6, 44, 44 and 2. 46 domains are there. So already I said many times, again I'm telling you guys, no need to create all these 46 data sets. Okay, so once you conduct the trial, okay, so as for your trial requirements, you are going to collect the data. So based on that particular data, what is the first document you are going to create? Of course, while if you want to collect the data, you are going to use CRF form. Through CRF form, you are going to collect the data, and then you are going to do first CRF annotation. Once, so based on the annotation, you will come to know somehow around 50% of the, not 50%, most of the data sets, how many data sets I need to create. There you will come to know how many data sets, and each data set, how many variables I need to create. Because one data set have 38 variables, all 38 variables values you are not collected, for example, and that too some of them permissible variable. Okay, so out of 38 you collect only 25 variables records, and 25 variables record only we are going to create that particular data set. Okay, so how many data sets to create as for the study, as for the collected data, as for the raw data? With the help of CRF annotation, we are going to create a spec document and a spec document. Yes, these many data. For example, like this, I will show you specification document template. I will do one thing. I will copy. Maybe I think I have already copied. Not yet, right? You can see here domains metadata. So how many domains? Okay, see guys, this table copied from SDTM implementation guide only. You can see in SDTM implementation guide, I think around 20th page or something. Just scroll down, you can find that particular table. Here, SDTM submission dataset definitions metadata examples. This one also very important to know, guys. Okay, how many domains, and each domain under what class it is, what category, either special purpose domain or events or interventions or trial design. What is the structure of the particular domain? Structure. So this is very important. So one record, for example, there is a DM, one record per subject. Each subject we are going to create one record in the sense one observation only. And what are the key variables? Keys. You can see. So this purpose is establishing as table only. So purpose it will be same for all. Okay. Description means complete name. DM demographics. Under what class it is? Special purpose domain. Structure one record per subject. Structure as a programmer, every domain, what is the structure? Structure or nature or what is the main focus for this particular test? All are same. Okay. So without structure of that particular data set, you are not able to create a data set, guys. Until and unless for record, only one subject, one record. If you don't know how many records, for example, there will be 101 participant, 5 records are there in raw data. So 5 records means, for example, there will be 35 variables. 10 variables you are copying from one table, 15 variables you are copying from another table, another 10 variables you are copying from the another raw table. 3 different tables, 15, 10, 10, 35 variables. Okay. So while combining, if you use vertical combining, you are going to get many records. But if it is a horizontal combining by subject, definitely you will get completely one record only, na? Horizontal combining. One observation only. If you go with through subject. Okay, horizontal combining or vertical combining. I need to go with set statement or else images. That is also very important. And again, what are the key variables? Guys, if you want to sort the data, of course, I said, right, we should follow the same order of the variables. What are the variables? Order is there in the implementation guide, same order. And that too, we need to do the sorting. So on which variable we need to do the sorting? So key variables, guys, always key variables. You can see each domain there is set of some key variables, guys. Key variables are nothing but to identify the distinctness of the record. Distinctness, uniqueness, any way you can call. You know, right? Distinctness means what are the distinct values for the age variable in the class data set? 11, 12, 13, 14, 15, 16. So unique values, right? So like that. So by using these key variables, you can sort the data and you can identify the distinct values and you can remove the duplicates. There are a lot of advantages, guys. Key variables are also very important for each domain. What is the structure of the dataset? What are the key variables? And location. See, once a dataset is created, there we are going to create XPT file to avoid migration issues, to open this particular table with any operating system and any environment in the sense, don't think about other tools. Definitely it will be SAS tool only. Okay. So what is the SAS Studio or SAS Enterprise Guide or SAS University Edition or SAS 9.4 version? If it is Windows operating system or Linux operating system or mainframe operating system, the XPT file you can open. Okay. So this is, for example, here see, as for SDTM 3.2, all these domains, key variables, and what is the nature of the record. Okay. You can see IE, inclusion and exclusion criteria not met. Any of the record, any of the subject or participant not met with inclusion criteria, exclusion criteria, but still they are participating in the trial. So this is also, guys, not mandatory domain. Not mandatory. Not mandatory in the sense, for example, there is 100 participants, all 100 participants met inclusion criteria and exclusion criteria. All they are recruited. Then what is the need of IE dataset? If any of the participants not met with inclusion criteria and exclusion criteria, but still they are continuing in the trial, why they are continuing? What are the what and all inclusion criteria and exclusion criteria they not met? Complete information. See here, one record per inclusion, one record for exclusion criteria. Okay, events. Each event, one record per event per subject. One event, one subject. For example, 5 events are there per subject, 5 records, 1 record only, 1 subject only, 5 different events. So nature is very important. Substance usages. You can see 1 record per substance type per reported occurrence per subject. For example, he is drinking coffee and he is drinking alcohol. He is smoking cigarettes, and coffee and tea or else alcohol. 4 different substances, same subject, just 4 different records, guys, like that. Okay. So this is structure every domain. See, if you go to any domain, I will show you the structure. On top of the table, it will display. That is mandatory. See here, DM dataset. Guys, most of the cases I'm using DM. Why while you are using DM, you may think, guys, this is the one parent domain. I can say that parent dataset. DM demographics. Okay, because almost to create all the domains, we are going to use DM domains, guys. DM domain. There will be study days concept. If you want to derive study days, definitely we need to pick the value from the DM domain RF SDTC. What is the start to date of the treatment? Okay, so guys, there is a combinations. So if you want to derive one domain, you need depending on other domains, other tables also. Okay, there we need to bring this. So once your dataset is created, we are going to save in a library, permanent library, all the domains. So if there is any requirement with existing tables, with any other variables from the existing table, we are going to pick the data. So simply create a libn statement, what and all datasets you saved in your library, import the datasets into SAS enrollment, and you can use those datasets with those variables, what and all variables you want. But whereas coming to the DM, almost all except trial design domains, remaining everything there we are going to use DM dataset. Okay, to derive few variables from other domains. See here, 1 record per subject. DM, CO, comments, 1 record per comment, or else SE, subject elements, 1 record per element per subject. So every dataset, every domain there you can find. So nature of the dataset, guys, or structure of the dataset. Of course, even though there is a detailed table is there with you. See here, and individual domain also there also it will display, guys. Okay, and again, why I'm talking about this table. So very fast, guys, once CRF annotation is done, based on the CRF spec document we are going to create, right? In spec document, okay, as per my CRF, how many datasets I need to create for this particular project. For example, here there will be flag to be created. DM, if it is available in my study data, yes. CO, comments, yes. Subject widgets, yes, these are common, guys. These are subject widgets. And CM, concomitant medication information, subject widgets and the subject elements, adverse events. Okay, for example, disposition. For example, death details information. This is, for example, there is no related information deaths. So you are not created. So there is no related data. So simply no. 1 second, guys, there is a call. Okay, so they will come to know. So how many data sets to create? Medical list like that. So once they identified these many data sets as for the study required, so we need to submit to the regulatory. They will come with plan. So which programmer? I said right, programming plan. For example, so all the domains not possible to go with a single programmer, right? Definitely we need to split the work. There will be separate things. Okay. For example, DM and CO created by ABC, the particular programmer name. For example, let's say, and these three by XYZ. These three domains are this domain PQR. Like that, there will be programming plan. Okay, so they are going to split each domain, which programmer, as per the requirement, required documents. They are going to send to this particular. See, for example, to create the DM domain, what and all study data tables, and spec document. Okay, so specification document need to send, and protocol and annotated CRF. If required, they are going to send all these documents to this particular programmer. Okay, and as for these domains, what and all study data required, and as for the programmer requirement, always guys, all the documents, what and all documents are shared with multiple things. It will be saved in the cloud only, so anyone can access from the cloud. So if you have access with you, not anyone in the sense, you're the programmer, you are working with that particular project. Okay, so if you want to check that particular document, if you have access, you can check it. Okay, so not all the time you don't expect complete access will provide, they are going to provide. Sometimes you may get read access, sometimes you may write access, like that. Different restrictions will be there. Okay, so they are going to split the work like this. One moment. Okay, as for the programming plan, they are going to set their plan, the required documents, they are going to share with the team. But guys, here, so this is, you can see, and domain and variables. So this is the one we created. So maybe you may get this kind of document for easy identification. For example, you are working with few domains, what and all variables you need to create. See here, there is an order. While keeping or while using retain statement to set the order, these variables will help you. All variables if you have in a single line, right? See here, all the domains variables are available in a single This is the one specification documented created by our instructors like Pawnee man. She is the one created this particular one. So and you can see CM domain table. So this is table you can find from as a SDTM implementation guide. And here just you can label. So guys, specifications guys, if you want to go with specifications, you are the one doing specifications, then just use specifications. So is this variable required? Collected data or not? So what is the source table? From that particular table, which column I need to import? What are the programming instructions? What is the length? All the instructions here. Okay, for example, you are dealing with the DM dataset. So as for the DM dataset, you can see DM spec document only you are going to get. DM spec document. This is your DM spec document. So what is the length? What is the source variable? How many variables? This is your source table, guys. Table, dataset. Means Excel file. DM table. So maybe you may receive it from Excel table, or else you may receive from SAS dataset itself, or else you may receive from database table. Okay. So, but these are your source tables, and these are your columns from this particular table to derive these variables. So this is your spec document and programming instructions here. Once you have spec document, see what and all tables will be there. Rand, DS, INCO. Okay, Rand, DS, INCO, DM, TRT. So Rand, TRT, and DM, INCO, and DS, around 5 different tables will be there. Okay, 5 different tables. Each table you can see here. For example, DM, subject variable, Rand, first DT variable, column, DS, LDS, and DT variable. We need to import. For example, go and check your raw study data. So this is the document you receive, specification document. You are the programmer, as well as for they mention in the spec document. So these are your source tables. Var file means, guys, this is source table, variable name. This is table name and this is variable name, column name from this particular table. So definitely they are going to send study data. Contains all these tables. So go and check your where it is DM study data to derive DM and sub DM. Just open. See here, DM table, DS table, INCO table, RAN table, and TRT table. To create DM table, yes, these 5 tables data we need to import. From each table, what and all variables I need to import, there will be spec document. Spec document will guide you, right? So for example, RAND table, first to DT, what else? See here, kit number. These are the variables. From the DM, sex variable, race variable, site variable, birth DT, LSS, not this one, subject. See, if you open my program, there you can find, guys. For example, while doing the program, what we are going to do. Special purpose domain. There you can see from the DM what and all tables I need to import. From RAND what and all value columns. DS, INCO, and TRT. See here we are importing. So 5 different tables from same Excel workbook. Same Excel workbook. What is the Excel workbook? This is the Excel workbook, right? Study data to derive DM and sub DM. Here only we have DM and DS and INCO and RAND and TRT. So see, it's different, but same Excel workbook. So instead of doing again and again, just we created macro here. So based on the macro, see here, we are changing the dataset names. We are changing the seed names like that. And after that, we are merging by using some reference variable. Okay, I'm not going with programming, just I'm telling you. So based on the spec document, how to write a programming and all, there you will come to the picture. Study data and spec document plays major role. So of course, along with the study data and spec document and protocol document and everything you are going to receive. But which data set, under which data set, for example, DM data set for what purpose? And under DM, each and every variable, what and all values I need to mention. For example, RF and DTC. What is the use of R and DTC variable value? What kind of data we need to fill with this particular column? Okay, so as a programmer, you should know what purpose, which dataset we are going to create, and in that particular dataset, variable value, what will be? That is main task, guys, to remember. I will program it. Okay, see here, subject elements. SC sequence, guys. So for example, there is elements 10 elements. So there will be order to do the sorting 1, 2, 3, 4, 5, 5 like chronological order. Element code, topic variable. See here, etcd is topic variable, synonym qualifier, element, description, detailed description. Okay, synonym qualifier we discussed, right? Like that. What I'm trying to say here, under element code, what and all values I need to put by using raw study data. Okay, and what are the attributes? 1 second. Okay, so that is what we are going to learn, guys, once we start the programming classes. So tomorrow I am going to start with, first one, trial design domains, guys. So when compared with all these classes, trial design is completely different because this is not a study data. This is planned data as per the protocol document. So protocol document means around 100 pages or 200 pages document. Okay, so of course we are going to share protocol document to the regulatory along with protocol document. So summary, complete summary in a table format we are going to share, guys. What and all elements you planned in that particular study as per the protocol document, trial elements. What and all treatment groups will be there? How many treatment groups as per based on your trial design? Randomization trial or else parallel study design, single sequence, open label, or whatever it is. How many plans? How many treatment groups you planned? Come on, guys, one moment. Okay, so here this trial design domain datasets completely protocol defined, not collected after the study. So before the study, what is your plan? How many visits you planned? What is your inclusion criteria, exclusion criteria in the protocol document you mentioned? So instead of sharing the document, if you create a table format, so which is very easy to understand, right? Regulatory. So all your protocol document summary, we are going to create in a 6 table format, 6 in 6 table. So these are the 6 tables first we are going to discuss. Okay, so out of these 6, mostly 5 are most popular, guys. Trial arms, trial elements, widgets, and trial summary, inclusion, exclusion criteria. These 5 domains we are going to discuss under trial design. So tomorrow, very first, I am going to explain how many domains we are going to discuss from each category. Trial design, how many domains, and findings and events and interventions and specific purpose domain. So almost around 20, 22 like that. These domains only we are going to discuss. The remaining domains, I will provide raw data and everything with you. Try to do your own. Okay, that's more than enough actually. 20 domains. No one can explain these many domains like EduMaster Training Institute. We are the one explaining these many domains, and we are the one taking these many days training, 90 days more for the project training, guys. Okay, so you are going to learn, no need to worry. So of course, this is completely new for this particular standards and all the terminology. You may feel initial days you may feel a bit confused, but once we done with around 5 domains or 5 to 6 domains, there, okay. So each and every variable, guys, similar variables only. Based on the domain characters only will change, remains everything same. For example, under intervention category, all the variables what we are going to create with these tables, all are almost same. All same variables. Just if it is SU we are going to use before the name. If it is EX only we are going to use. Remains everything same, all characters. Okay? You will come to know. So remember, SDTM implementation guide is very, very, very important. So of course you know very well about your SAS programming. Until and unless if you don't know which domain under which category, what is the nature of that particular domain, and what is the use of that particular column, and what is the value I need to put that particular column. If you don't know, there is no use of your programming. Okay? So this is main concept, guys. And SDTM implementation guide, you should thorough with SDTM implementation guide. Okay, this guide we are going to learn next 3 months classes. We are going to learn this guide only. One by domain by domain by domain. There is an order. What is the order we are going to discuss all the domains? I will list down tomorrow. So that's all about today, guys. Any questions, please? Any questions? Not tomorrow. Tomorrow just I will introduce all the domains with you, and mostly from after Pongal only. You know, from 10th onwards, there is a Pongal holidays. Next week, there is no classes, guys. Simply I can say that. Saturday, Sunday, there is no classes. Next week, Sankranti, Bogi, Kanuma, there is no classes next week completely. And again, Saturday, Sunday. So next class will be Monday only. So tomorrow is the last session. Again, after 20th only. So 20 means, you know, what would be the date? 21st. Next class will be 21st. So 21st onwards, we'll start your programming classes. Done? So meanwhile, just go through all the introduction, what we discussed, and go through the SDTM implementation guide.
Yeah, okay, Balu.
Thank you, guys.